Welcome, everybody. Give us just a few more seconds. We're going to start in just under a minute and let folks roll on in. So stand by. Good day, good evening, wherever you may be, and welcome one and all. My name is Peter Girguis, and I'm the co-director, along with my colleague, Dr. Mike Gilmore, of Harvard's Microbial Science Initiative. The last year has been one of tremendous upheaval, to put it mildly. The world faced terrible wildfires in Australia and the Western United States. Antarctica saw its highest temperature on record. And of course, the murders of George Floyd and Breonna Taylor and many others brought to the fore the persistence of systemic racism around the world. And humankind will be reckoning with these and many other issues for years to come. Of course, the COVID-19 pandemic loomed large over 2020 and 2021, perhaps in some part because the pandemic is caused by a virus that seemingly came out of nowhere. Like other pandemics throughout human history, COVID-19 was responsible for many deaths. It also brought our global economy to its knees and brought our vulnerabilities into sharp relief. 2020 was, on whole, a year in which humanity's vulnerabilities and shortcomings were on full display. Now, conversely, the power of microbes and viruses were, in many ways, also on full display. And if there was ever a year to recognize the importance of microbes and viruses, as well as the people who study them, it's this one. Virologists and microbiologists and many, many other healthcare workers have worked tirelessly to develop vaccines against this coronavirus. They did so by putting the power of microbes to work in the lab, microbes that are workhorses of our research. Microbiology, virology, and molecular biology writ large have made it possible to develop vaccines at an unprecedented, astonishing pace. The Nobel Prize in Chemistry was split between two brilliant pioneering scientists, both women, I might add, who unlocked the power of microbial gene editing systems. Microbes are also responsible for helping humanity, but they make many of the goods that we rely on each day, and advances in microbial biotechnology continue at breakneck speeds, even against the backdrop of a pandemic. But praising microbes for their role in helping humanity really isn't enough, because we need to acknowledge that the biosphere itself, our habitable world, is run by microbes, and that these minuscule microorganisms, organisms that are about 20 orders of magnitude smaller than we are, shape every aspect of our planet's habitats. This symposium is a celebration of microbiology and virology, and the research you will be hearing about today brilliantly shows how Earth is in fact a microbial world. We humans, whether we want to admit it or not, are simply along for the ride. Throughout this symposium, we'll be hearing from extraordinary scientists who were nominated by the MSI microbial community, and whose research crosscuts environmental, applied, and medical microbiology. We'll also be hearing from four amazing artists, each of whom is in their own right an amazing microbiologist, but they also enrich our lives and understanding of microbes through the language of art. Like you, I'm very much looking forward to their talks. But before we begin, we would be remiss if we didn't acknowledge the loss of life due to COVID-19, and I would ask you that you join us in just a single minute of silence in remembrance of those who lost their lives. Thank you very much. Now, without any further ado, I'm going to turn the virtual stage over here to my colleague, Dr. Hannah Gavin, who's going to tell you about today's program. Hannah?
Hi, everyone. Thanks, Pete. My name is Hannah Gavin, and I'm MSI's program manager, and I'm really excited to share some of the logistics of today's symposium with you. So, as you probably know from seeing some of the materials that we've distributed, we have two major scientific sessions today, and those are divided by a lunchtime program and capped off with a happy hour that will occur in the evening. In each session, we'll have 3 research talks and 2 sci-art features, which will be interspersed between those research talks. Now, at the end of a given research talk, we might have time for 1 or 2 questions, but if you don't fit yours in, don't feel worried, because we've set aside 45 minutes at the end of each session where we'll talk amongst all 3 of the researchers who have given presentations in that session. They will be able to ask questions of each other, and you will be able to ask questions of them. So we've got plenty of time set aside for Q&A towards the end of the session. Now, whether or not regardless of the time zone you're on, feel free to join this lunchtime program. Maybe it's more like an evening time program for you, but we've got an awesome family-friendly microbe trivia hour lined up, which will be a really good time. So if you want to grab a family member, grab a snack, and join us for that, it will be excellent before we proceed to session 2, which will be in the same format as the first session. And we've also got a happy hour set up in a separate Zoom call. And you can find the information for that Zoom call in the reminder email that you received, the automated reminder email that you received from Zoom about this webinar. If you scroll down to the end of that, you can find the info for the happy hour. Now, thinking about some tips for enjoying and participating in the virtual symposium today. If you haven't gotten it already, we have available a full symposium program, including biographical information and contact info for all of our researchers and sci artists. And that can be found on our website at msi.harvard.edu/annual symposium. You can also easily navigate there from the homepage. If you'd like to use them, subtitles and closed captioning can be enabled by using a link at the bottom of your Zoom screen, one of the tools in that bottom toolbar. If you have questions for a speaker, I hinted at this before, but feel free to make use of the Zoom Q&A feature as questions occur to you. I'm going to really encourage you to, if you want to type in a question, use the Q&A as opposed to the chat so that we can make sure we log those questions. Now, if you want to get it down on, I was going to say paper, if you want to type it in while it's on your mind, feel free to do so during the person's talk. Just keep in mind that we might not get to it until the panel Q&A session. During that 45-minute panel Q&A, you're also welcome to virtually raise your hand, and at which point we'll invite you to unmute and ask your question aloud, which helps us feel a little bit like we're more in a community too, so you're very welcome to do that. I've already told you about the evening happy hour. So before we get to the first talk, the last thing that I want to do is give a couple of partner spotlights here, which will intersperse throughout the program today. One of our partners who you will be hearing from at our trivia hour is Skype a Scientist. The mission of Skype a Scientist is simple yet impactful: to make science accessible and fun through personal connections with scientists. We are an educational entity with a focus on connecting the general public with scientific disciplines in fun and meaningful ways, making science education available and engaging for everyone. We want to give students the opportunity to get to know a real scientist and get the answers to their questions straight from the source. Skype a Scientist creates a database of thousands of scientists and helps them connect with teachers, classrooms, groups, and the public all over the globe. Scientists, teachers, and groups of families of any size can sign up to participate in Skype a Scientist programs at www.skypeascientist.com. I'll also highlight the Harvard Museums of Science and Culture. The mission of HMSC is to foster curiosity and a spirit of discovery in visitors of all ages, enhancing public understanding of and appreciation for the natural world, science, and human cultures. HMSC works with Harvard faculty, museum curators, and students, as well as with members of the extended Harvard community, to provide interdisciplinary exhibitions, events, and lectures, and educational programs for students, teachers, and the general public. HMSC is seeking scientists who want to share their stories through a wide range of media. Occasional opportunities for podcasts, social media, e-news stories, and live webinars exist. And they're especially interested in highlighting diverse stories, including from bilingual Spanish-English speakers. So to learn more about that, you can find contact information for both of these partners on the annual symposium page at the MSI website. With that, I invite you to sit back, relax, and enjoy today's symposium. We've got a really great lineup for you, and thank you for being here.
Thanks again, Hannah. Now, very briefly, over the past year, as we've mentioned, we spent so many hours focusing on the havoc that can be wreaked upon humanity and even on our biosphere, right? When we throw things out of balance, to be candid. But now let's spend the next couple of hours or so in celebration of the microbes that keep our planet habitable for all life, including humans. So it's my pleasure to introduce our session on environmental microbiology. I'll be introducing the science speakers, and my colleague Julia will be introducing our featured scientist artists. So first, it's my great pleasure to introduce Dr. Arpita Bose, who's an associate professor of biology and earth and planetary sciences at Washington University in St. Louis. Dr. Bose's interests and talents are many, ranging from geomicrobiology and microbial physiology to biogeochemical cycling, gene regulation, and microbial respiration. But as of late, Dr. Bose has also been bringing her expertise to bear on microbial metabolism, or expertise in microbial metabolism, to bear on engineering microbial strains for sustainable biochemical and bioenergy production, as well as to tackle issues in bioremediation and biofouling. Dr. Bose has been the recipient of many honors and awards, including a Packard Fellowship in 2015, the Changing the Face of STEM Mentorship Award from L'Oreal and AAAS in 2017 and 19, and the Department of Defense Mentorship Awards in 19, 20, and 21. I'm very proud to say that Arpita Bose is also an alum of my lab here at Harvard, where she led groundbreaking work on microbes that breathe rust. I learned much from her, and it's wonderful to see her thriving at Washington University. So without any further ado, Dr. Bose, the screen is yours.
Thank you so much, Pete. I am so excited to give this seminar because most of what I present today is all stuff that started in your lab. So I'm going to share my screen. And hopefully everybody can see my slides. Can everyone see my slides?
Looks perfect. Thanks, Arpita.
Awesome. So I'm really excited to give this seminar today because so much of this started at Harvard, and it's like coming full circle for me. And like Pete said, that there's a lot going on, and microorganisms are doing so much for us, and there's a happy side to it. And I hope that this can showcase the happier side of microorganisms. So I'm going to talk to you about work that I've done in the past 6 years or so, discovering and exploring the electric side of photosynthesis. But this is the first talk of the session. So I wanted to go big, and I wanted to ask some of you in the audience, what do you think life is? And I know that this might not be as interactive as I had hoped, but let's just dive in and think about this existential question. What is life? For me, boringly, life is made of carbon, and maybe some of you agree with me. I also know that life requires energy. But then it gets even more complicated. Life requires an electron donor, and it also requires an electron acceptor. And this movement of electrons from a donor to an acceptor and vice versa is what I think life is made of. And at least one person I know agrees with me. It's Albert Girguis. He's no longer with us, but he had won the Nobel Prize in Physiology or Medicine in 1937 for the discovery of ascorbic acid, which we recognize as vitamin C, and also the reactions of the citric acid cycle. He also said that life is nothing but an electron looking for a place to rest. So now let's look at what we do. We think we are very exotic and we have very different diets, but actually turns out that we have a very specialized diet. For energy, we can only use chemical energy. For carbon and electron donors, we use food. We might think that we are omnivores or vegetarian, vegans, etc. But all of the food we eat is organic carbon, and it serves as a carbon source and our electron donor. As an electron acceptor, all we can use is just one thing, which is oxygen. So in the morning, maybe you ate some breakfast, I did, and I burnt that organic carbon in my breakfast. The electrons from that ended up on oxygen, reducing it to water, and that's how I made energy. And I'm very energetic giving this talk right now. But microorganisms are much more exotic with respect to their diets. And this, Pete, is a fish image of an ethane degrading consortium that was isolated essentially in Pete's lab, starting in Pete's lab. And this consortium is so unique, it can actually degrade a 2-carbon, highly reduced molecule like ethane, something that we can never imagine ever consuming. So microorganisms have diverse diets. They can eat complex carbon, they can consume and use simple carbon molecules. For energy, they can not only use chemical energy, but they can also use light energy. In that way, they're like plants in some ways. And for electron donors, I would venture to say that they can use anything that can donate electrons. And that might be a little bit far-fetched for some people, but that's what I believe. And for an electron acceptor, again, I think they can use anything that can accept electrons. So to me, the true diversity of microorganisms comes from this ability to use different forms of carbon, different forms of energy, electron donors and acceptors. And diving into what I have been focusing on with something that started in Pete's lab was this really extraordinary ability of microorganisms to use solid materials, solid conductive substances like rust or proxies of these like poised electrodes as sources of electrons, as electron acceptors and donors. For about 30 years, we know that microorganisms that can degrade organic carbon, much like we do, and produce electrons, when we actually take these electrons and dump it on oxygen to make water, these organisms, however, can use rust and even poised electrodes, practically making electricity. And the mechanisms underlying this process are known, and this process itself is called extracellular electron transfer. Now we know that this is one flavor of how microorganisms can interact with solid materials. And this is the reductive form of extracellular electron transfer. When I came to Pete's lab, I didn't really have a plan in mind, but I know that thanks to the efforts in his lab, I was able to recognize that it's possible that these substances that are solid can serve as sources of electrons, like electron donors for microorganisms. At that time, there was very little understood about this. In fact, people were mostly noticing this phenomenon in the environment. And there were very few organisms that were in pure culture that could do this. Since then, this entire concept where solid materials that are conductive can serve as electron donors has burgeoned into its own field in the past 6 years, 7 years or so. And this process is called extracellular electron uptake, where these solid materials can serve as electron donors for microbial metabolism. So why was I even thinking about all of this? I was thinking about this in the context of a very cool microbial metabolism that's called photoferrotrophy. Photoferrotrophy is the ability of microorganisms to use light. So I mentioned that microorganisms can use light, much like plants, as an energy source, and they can fix CO2 to biomass. But the electrons for this fixation reaction comes from something really exotic, like soluble iron, depicted here as iron 2. And this metabolism happens in the photic zone. When microorganisms do this, they practically surround themselves in rust, and many of them get attached to these rust particles like goethite and eventually magnetite, and they actually sink to the bottom of ecosystems. So earlier their metabolism had to be focused in the photic zone, then they sink to the bottom in the aphotic zone, and they settle in the bottom like microbial mats that you might have seen if you go to some salt marsh. And why is photoferrotrophy so important? many people believe that photoferrotrophy might have given rise to what are called the Precambrian bandadan formations. And to just give you a sense of scale, I can't see myself, but this is like a little show and tell. This is a bandadan formation from Australia. And you can see that there are layers of alternating iron oxides with black layers that are called chert or silica. And you can see that these structures are very exotic because they formed a very long time ago in the Precambrian period. And many scientists believe that photoferrotrophs might have led to the formation of these structures along with other processes. And believe it or not, this is the largest iron ore deposits we have. So this is why I got very interested in understanding photoferrotrophy. But thanks to my introduction to Pete's lab, I also understood that these microorganisms exist in extant ecosystems and that things might not be so simple for them. And I already knew that in the lab when I was growing them, these microorganisms would get surrounded by rust and most likely would sink to the bottom of any ecosystem. And then they would be spatially separated from their choice electron donor iron 2 as well as light, which is their energy source. But we understood that they're reductive ET capable organisms that can use, say, organic carbon that's being released from these same cells and other cells dying and releasing this organic carbon. So they can use this organic carbon as an electron and carbon source. They oxidize this to CO2, much like we do, and they transfer these electrons to solid or rust particles produced by, say, organisms like photoferrotrophs. So they establish a really nice iron cycle. So it all makes sense. You have photoferrotrophs that want to hang out in the photic zone. You have reductive EET capable organisms that can make this iron 2 available to them in the photic zone. It all kind of makes sense. But there's one problem. Reductive EET capable microorganisms don't want to hang out in the photic zone. They want to hang out in the aphotic zone where there's lower oxygen, and they tend to be almost in the bottom of ecosystems. So now this creates a problem. Photoferrotrophs want to hang out in the photic zone. They also require iron 2. But reductive EET capable organisms want to hang out at the bottom. Now what? And so this is where this idea of perhaps a process like extracellular electron uptake became even more attractive to me as I was continuing my postdoc in Pete's lab, where it became apparent that if organisms like photoferrotrophs want to hang out in the photic zone, which is where their choice energy sources, but they don't have access to their choice electron donor, they can still have access to these electrons through this process of extracellular electron uptake if there's a conductive matrix around them, and these electrons can be taken up by these microorganisms. So they can continue to have access to these electrons to fix CO2 to biomass. So this was this idea that I had when I came to Pete's lab, but then how do I really go about answering this question? We were just scratching our heads trying to figure out if there are even organisms that can do this. And so this is where I think I got lucky, because I had started my postdoc in Diane Newman's lab, where I was working at MIT while she was at MIT, and I was working with this really cool organism called Rhodopseudomonas palustris Taiwan, and it can do photoferrotrophy. So it uses the energy of light, hangs out in the photic zone, and fixes CO2 to biomass using electrons from iron 2. But I was growing this in the lab, so you'll start with not rusty cultures. You'll inoculate them with this organism, and the bottle will become rusty within a few weeks. And practically, these organisms would surround themselves with iron oxides like you see right here. So I was seeing just in the lab that there was spatial separation between the microorganism and iron 2, which was their choice electron donor. So I knew that this organism faces the same challenge that I just outlined to you earlier in the talk. And when I moved to Pete's lab, he was going to the bottom of the ocean, bringing rocks and mud and whatnot, and bringing them to the lab. And with a lot of energetic grad students, we were putting together these bioelectrochemical systems and studying whether these the microorganisms in these rocks and mud could be used, could use solid materials as electron acceptors or donors. And we were using these bioelectrochemical systems where you had an anode and a cathode. They were separated by a proton exchange membrane. And I knew that the cathode is where electron transfer would occur for an organism like Taiwan. So I thought that maybe if I inoculated microorganisms in the cathodic chamber and only provided light as the energy source, CO2 as the electron acceptor, perhaps an electron donor, the electron donor being this poised electrode, perhaps the only thing that could happen for the organism to grow is for it to do extracellular electron uptake from this poised cathode. And the potential that I used was exactly these minerals that these organisms produced. So it was an experiment that we tried with some other postdocs in the lab. We had no real expectation, but very quickly we saw that we were correct, that Rhodopseudomonas palustris Taiwan was able to accept electrons from these poised electrodes in the presence of light. And if we took light away, we lost 70% of this ability, and only 30% of that activity remained. And in Pete's lab, this became a nice paper for me, and I started my lab at WashU using this idea. And since then, we've used Taiwan as an excellent model to study not only the molecular mechanisms of how electrons come in from soluble iron as well as electrodes and rust, where these electrons go, because this is a photosynthetic system, it goes to the photosystem, and eventually how these electrons allow the organism to fix CO2 to biomass. And in the midst of it, we have had a couple of PhDs from the lab. So this Dinesh Gupta, now a PhD and a postdoc in UC Berkeley, and Dipti Nayak's lab, who's also from Harvard. And he was able to show, with the help of Bob Krantz and Molly Sutherland, the mechanism underlying how these electrons come into the cell. And so we got clues from an organism that's a reductive EET capable organism called Schuonella onidensis. It's a gram-negative organism, so it has a cytoplasmic membrane, a periplasm, and an outer membrane. And it uses this 3-protein complex where you have a porin that holds together two decaheme cytochromes, one that sits in the periplasm and another that sits extracellularly. And electrons that are produced by, say, degrading organic carbon are passed from the cell through the periplasm, through this conduit, to, say, iron oxides or rust, to reduce them to iron 2. We also knew from Diane Newman's lab that there's an operon called the PIO-ABC operon that she had shown was important for iron oxidation, and that there were similarities between these two systems, where you had the porin, you had the periplasmic decaheme cytochrome, but you didn't have the extracellular outer membrane decaheme cytochrome. Instead, you had this high potential iron sulfur cluster protein that might be important for bringing the electrons into the cell instead of pumping them out. And to really ask if to test whether this system might be important for extracellular electron uptake, we had to move away from the bioelectrochemical systems that we were using sort of in Pete's lab, and we had to make miniaturized versions of these bioelectrochemical systems. And this was work done with Mark Meacham at Mechanical Engineering at WashU and my postdoc, where we literally miniaturized these electrodes. And now we have microfabricated them into 9612 format, which is essentially a small little plate that can sit on a plate reader. And the way that this system differs from the bioelectrochemical systems I just described is that here you have the cathodic chamber and you have planktonic cells, as well as cells that are attached to the electrode. And so you have the contribution of both the plankton as well as the cells that are biofilm attached. However, in the bioelectrochemical cell, or the microbec, we can actually attach the cells as biofilms. And because there's fluidic control, we can wash off the plankton, and the only contribution we see is from cells that are attached to the electrode, which is indium tin oxide in this case. And using this, we were able to show that indeed the wild type is able to take up electrons. And here the gray bars show dark conditions, and you don't see any electron uptake in the dark, but you do see under light that there's electron uptake, shown by the blue lines here, that the PIO-ABC mutant and other mutants that we had made in this operon are absolutely not able to do extracellular electron uptake. And there's a mutant where a small portion of the PIO-A protein is deleted, and that has a defect. And so using this bioelectrochemical system, we were able to show that indeed the PIO-ABC system was absolutely essential for extracellular electron uptake. And now we know that we can use the bio-AB complex as a hook for environmental organisms. And there are at least two other environmental organisms that we're easily able to find in GenBank. And these are Rhodomicrobium odeperense and Rhodomicrobium vanellii. And just the presence of the bio-ABC system in this showed us that we can actually predict that these organisms would be able to do iron oxidation. So here we are tracking iron over time. So these organisms have bio-ABC. Just like Taiwan, they're also able to do photoferrotrophy. And just like Taiwan, they're also able to take up electrons from a poised electrode. And this got a graduate student, Eric Connors, in the lab very excited about studying these organisms because they have been implicated in bandedite formations, the same structures that I showed you here. So we kind of come full circle, going from not knowing anything about these structures and how they might have come about, looking at extant organisms and connecting the dots with Earth history. So microbes are super important. And now you can see that using Taiwan as a model, we have been able to show that the bio-ABC proteins are produced inside the cytosol of Taiwan. They're transported through the through the inner membrane to the periplasm. The bio-B protein gets inserted in the outer membrane. The bio-A protein is truncated. A 200 amino acid region is lost. It makes a complex with bio-B. And then with the help of bio-C, it actually takes electrons eventually to the energy heart of the energetic system in this organism. And this was work done with another graduate student who's the first graduate student to go to college in his family, Michael Guzman, who published his work in Nature Communications. And he was able to use the same microbac that I described to actually probe the photosynthetic system in this organism. And again, there were two other PhDs that were involved in this as well. And that's Michael Binkley and Andrea Stoica, who's in my lab with Marks as well. And so again, just as a reminder that in the microbac, we are looking at the response of only the cells that are attached to the electrode. And here, if you have a no cell control, you can see that there's no electron uptake. But if you have wild type Taiwan and you expose it to either dark or light conditions, you can easily within seconds track electron uptake happening in these microbac systems. And using that, we probe the photosynthetic electron transport chain of phototrophs like Rhodopseudomonas velustris. It's a complex system, and I don't want you to focus too much on what you see here, except the fact that if you were to track the energetic changes that were happening to the photosystem of this organism in response to light, you see light and this photosystem are friends. The moment light shines on this photosystem gets very energized because it has a molecule inside it. And then this highly energized electron wants to move back to the ground state. From basic physics, we understand that. And biology has come up with ways of sort of taking advantage of this electron drop, and it has inserted all these different proteins in the way, and these proteins can generate a proton motive force that allows the cell to make ATP, and we do exactly the same thing in our mitochondria. And here, using different kinds of chemical inhibition approaches, especially using antimycin in this example, where it inhibited one particular component of the photosystem called the cytochrome BC1 complex, we were able to show that if we actually inhibit the cytochrome BC1 complex, we are completely able to stop electron transfer from occurring, suggesting that the electrode, which is poised in this case at plus 100 millivolts, probably allows the electrons to enter at the BC1 complex. So we were able to do biophysical experiments using this system that we created. And we now know that the electrons come through the bio-ABC system in Taiwan, and they enter the photosystem at the level of BC1. And Michael also showed that these electrons can move even further to allow these organisms to do primary productivity. They're actually making biomass using these electrons from solid conductive materials. And so he actually was very brave, and he went about deleting a gene called rubisco that's part of the Calvin-Benson-Beauchamp cycle. Rubisco is an absolutely essential gene, and Rhodosudomonas palustris has 2 copies of it, Rubisco form 1, which is called CBBLS, and Rubisco form 2, which is called CBBM. And through many years of effort, he was actually able to delete these essential genes. And what he found was spectacular. What he found was the wild type was able to take up electrons over time, but the Rubisco form 1 CBBLS mutant had very little defect in electron uptake. The Rubisco form 2, which is CBBM, also didn't have a defect. But when he deleted both of these forms of Rubisco, he prevented carbon fixation from occurring using the Calvin-Benson-Basham cycle, and this organism could no longer take up electrons anymore. So in summary, what he showed was that Rubisco was essential for extracellular electron uptake, and that the inner inside of the cell is connected to the outside of the cell. If you don't allow the cell to fix CO2 through the CBB cycle by deleting Rubisco, it can no longer do extracellular electron uptake. And to me, this was remarkable, that there's such a huge connection between what's happening inside a microorganism with what's happening outside. They're extremely smart creatures. But as someone who's passed through Pete's lab, there's no way I was satisfied with doing all this molecular work, genetic work, biophysical work. To me, none of this meant anything if it didn't matter ecologically. And thanks to my training in Pete's lab, I was able to move to Woods Hole as a teaching faculty the summer before I came to WashU. And together with students, we isolated organisms from this really cool ecosystem called Trunk River, which is an estuarine environment. And a fun fact is that it's very close to Albertson's Girguis home. So I'm very connected, very attached to Woods Hole. And we were able to show that in these ecosystems, we can isolate photosynthetic organisms and that they can all do photoferrotrophy and they can all do extracellular electron uptake. And this work is going to come out soon in ISME-J. And being in WashU, we're very close to the Mississippi River Valley, and we identified a little cool ecosystem called Ellis Island. And every year, twice a year, we go to this ecosystem, we sample this wetland ecosystem, and we have been able to isolate many, many photosynthetic organisms that also can do photoferrotrophy as well as extracellular electron uptake. So this process isn't just limited to Taiwan. You can find it everywhere. And we, over the course of 6 years, have focused on photoautotrophs that can do extracellular electron uptake. But here, Michael and Dinesh, together with me, reviewed that the field has come a long way, and it shows that extracellular electron uptake, this ability to use solid conductive substances as electron donors, is all over our planet. It happens in photic zones, in oxic zones, anoxic zones, in the deep sea. It's pretty spectacular that in a very short time, the community has shown that this process is so common. But why is this process so common? I think it brings us back to the same original idea that we had while I was in a postdoc in Pete's lab, where microorganisms face the separation from their choice electron donor all the time. It's happening in the test tube and when we are growing Rhodosudomonas, but it's happening in the environment all the time. And the environment is extremely conductive. Not only does it have minerals, but it has other small molecules that make the environment very conductive. And as long as an organism has this ability to do extracellular electron uptake, it will be able to survive and perhaps even grow. And it can be spatially separated from its electron donor, but as long as it has this ability, it's fine and can hang out wherever it wants. And now we have been able to show that not only is it happening in wetlands like Woods Hole and Ellis Island, but that it's happening in our bodies as well. And we're looking at this phenomenon in human pathogens. And there are hypotheses that are being tested in my lab that maybe this process is important for the first eukaryotes that might have come across as well. And so without any further ado, I want to thank my lab. My lab has been spectacular, and we have created a very diverse ecosystem. And when Pete brought up racial inequities in every aspect of our lives across the globe, I have been fortunate that my lab has never felt any racial inequity. Look at my lab. We have people from all over the world. We have people all shades of shades, and we have gotten amazing support from the community to help build their careers and explore this really interesting phenomenon as well. And with that, I just wanted to say thank you for your time, and if you have any questions.
Thank you very much, Arpita. Boy, I have questions, but first, let's in the interest of time, let me remind folks in the next few seconds that we do have a session dedicated to questions immediately after our environmental speakers. So let me ask you to jot your questions down and stick around and ask Arpita the questions, because I know you have them. And Arpita is fantastic about answering whatever questions you may have. But thanks again. I'm going to turn this over now to my colleague Julia to introduce our next speaker. Julia.
Thanks, Pete. Hi, everyone. My name is Julia Worcester. I am MSI's program assistant, and I'm really excited to introduce our scientist artists to you during this morning session. So first up, we have Dr. Vineetha Zacharia, who is a postdoc at UC Berkeley. She studies the multicellular life cycle of the soil bacterium Streptomyces silicolor, which produces a diverse array of natural products, including antibiotics. She dips her paintbrush directly into antibiotic-secreting colonies to make her actino art. I'm now going to roll a short video that talks more about Dr. Zacharia's work.
Hi everyone, my name is Vineetha Zacharia, and I'm a postdoc at UC Berkeley studying the soil bacterium Streptomyces silicolor. This microbe is amazing in that it has a complex life cycle and makes a lot of medically and industrially relevant natural compounds, many of which are pigmented. I study the factors that drive these complex processes in Streptomyces silicolor. My sci art is inspired by this bacterium because I actually paint using the secreted pigmented antibiotic called actinorodin from growing colonies. My origin story begins when I was bitten by a radioactive spider. No, that's not right. Actually, it began when I noticed the beautiful blue actinorodin droplets being secreted from Streptomyces silicolor. Out of curiosity, I wanted to know what it would look like on paper, and as soon as I realized that it resembled watercolor paint, that's how I started creating what I now call actino art. My interest in science started at a very young age with the children's program Reading Rainbow with LeVar Burton, which featured a book about microbes. I was fascinated by these tiny organisms, and it is amazing that I get to work with them every day and now make artwork with bacterially derived paint. I particularly enjoy drawing animals, and I think I was largely influenced by my mom's incredible college zoology notebook, which contained her intricate animal drawings. I also enjoy drawing and painting people's pets. For my artistic process, I focus on a specific animal and I find a reference photo, do a quick pencil sketch to build muscle memory, and then finally paint the piece freehand by dipping my paintbrush directly into a bacterial colony and transferring the pigment to paper. Also, the bacterial colonies that I get my paint from are actually plates that I've already done an experiment on. So I not only derive my scientific data from these growing colonies, but I'm also able to use it for my art. Science is a great subject for my art because there is inherent beauty in science. Being able to tap into the all-natural bacterial paint to make my art speaks volumes, because not only is Streptomyces silicolor a source of precious antibiotics, but also as a source of sustainable pigment. I actually have two favorite pieces of my art. I'm particularly fond of my tiger and blue jay paintings, which personally for me symbolize strength and peace, respectively. My audience is everyone and anyone. I've gotten great responses for my art from the wonderful folks on Science Twitter, and also people from a non-science background, so it's been really rewarding sharing the wonder and benefits of bacteria.
Great, now we're gonna show a slideshow of Dr. Zacharia's work, and this will loop for about 5 minutes. So folks are welcome to go get some coffee or run to the bathroom if you need to, and the art will be here when you come back. to introduce our next speaker.
All right, thank you very much, Julia. And thank you, Dr. Zacharia, for sharing your amazing art. I was just commenting that I can't really even draw a circle with a pen. So watching you produce such amazing images with microbes is amazing. And thank you again, Dr. Bose, for a wonderful opening for our symposium. Now, it's my pleasure to introduce Dr. Oded Beja. Dr. Beja is a professor at the Technion Israel Institute of Technology. And he's well known for his amazing discovery of the first bacterial rhodopsin, naming it proteorhodopsin, during his postdoctoral fellowship in the laboratory of Ed DeLong at the Monterey Bay Aquarium Research Institute. Today, Dr. Beja's lab focuses on the role and diversity of photosynthetic organisms even cyanobacteria and the like in the oceans. And he continues to pioneer the use of functional metagenomics in the discovery of new light-sensing proteins. Now, in 2018, Dr. Alina Pushkarev, Dr. Beja, and other members of the Beja lab discovered a new family of rhodopsins, naming them heliorhodopsins. They possess an inverted membrane topology, and they are found broadly among bacteria, algae, algal viruses, and archaea. Oh, and I should also note that Dr. Beja's lab really looks at the role of viruses in moving these gene systems around. Now, I'm also very fortunate to say that I got to work alongside Dr. Beja in Ed DeLong's lab. Many years ago, when Oded was there as the lab's most experienced postdoc, and I was a very green, very new postdoc, and quite new to genomics. And I learned a great deal from him. And watching Oded do his work sort of blew my mind. At the time, Oded and Ed were, of course, working together to use large insert libraries like back libraries and the like to make these discoveries. And amusingly, I was on this, I was just sort of learning cloning, just to put it in perspective. So felt like Oded was on this information superhighway and I was on this little dirt road. But Oded was a wonderful colleague and taught me a lot and kept me out of a lot of trouble, not the kind of interesting trouble that one can get into, but the really kind of mundane trouble one can get into in the lab. And so I'm very grateful for Oded's mentorship and equally grateful for Oded's many contributions to science. And I'm really thrilled that he's able to join us today. So now I give you Dr. Beja. Oded, the screen is yours.
You see it?
Yeah,
okay.
There it is.
Okay, so I'm going to talk today about microbial rhodopsins, and Peter did ask me to really introduce this subject. So I'm going to start by introducing metagenomics really briefly, and then how we use it to find new rhodopsins. And I'll give you things already from the 70s, but then we'll move to nowadays. So as Peter said, I'm a member here in the Technion in Israel. I established my lab in 2001. And ever since I'm working on photosynthesis, but also on rhodopsins and the differences between them. Okay, so the main problem for us trying to understand what are the roles of microbes in the environment is the problem to cultivate most of them. So it depends whom you ask. It would be 98%, 99% uncultured. And the problem is if those are not cultured, how can you study them in the lab? So here you can see a picture from a drop of seawater. So you can see those microbes in the microscope, but perhaps only one of them, let's take this one, could go into culture. And most of them you will not be able to study easily in the lab. So luckily, these days we have metagenomics, and we have all kinds of metagenomics to study them. And the idea, I would say, is simple. If you cannot grow them, let's collect them from the environment and try to look at the genomes and predict what they're doing. And then perhaps, if we are lucky, we can really close a cycle and then go back, in my case, the oceans, to study them. So I'm going to show you two kinds of metagenomics. What you see now on the screen is the one that me and Ed DeLong and others also at the same time, already in 2000, perhaps also before, developed. And this is, you go to the environment, any environment, you collect your microbes of choice, and usually the entire population, using filtration usually. We collect the entire DNA of the entire population. In this method, we try to take whole genomes, entire chromosomes, and then we clone them into special vectors. It's BACs, bacterial artificial chromosomes, or other vectors. So you can see here, this small black line is actually the vector, and the inserts could be even 200 kilobase. So 200 kilobase could be, in the case of marine bacteria, 1/5 of a genome. And then, of course, we sequence it and trying to understand what they're doing using bioinformatics. Nowadays, metagenomics is done a bit differently. So again, you go to a certain environment, you collect the DNA of the entire population, but you chop the DNA to small pieces. You can clone them into regular vectors, but these days, we usually use the new sequencing methods, so mostly Illumina, but other methods too, and we sequence directly those pieces of DNA, let the computer assemble, and then try to understand, based on those assembled genomes, what you have. Okay, so why do I show you the Dead Sea? And this hypersaline environment is from Israel. And actually, I was in the Dead Sea a week ago while traveling from Eilat up north, and it's very hot and it's a very hostile environment. I'm showing you this because I want to describe to you the first microbial rhodopsins that were found in the 70s. And they were found in an archaea called Halobacterium salinarum. Nowadays, this is the name. And in this archaea, we can see different pumps that are actually rhodopsins. So before I describe rhodopsins, I just want to mention that you all look at this lecture using your own rhodopsins in your eyes. So those are animal rhodopsins. The structure is similar, but there's no connection between the two families. So it was a big surprise in the early 70s where when proteins resembling the rhodopsins we have in our eyes were found in archaea. And this was going on for 30 years because for 30 years, all we knew were those archaeal rhodopsins. So what do we have here? We have actually one protein plus a chromophore, in this case, retinal, a derivative of, you can say, vitamin A, not exactly the same, but very similar. And then you have light, or actually photons, hitting those retinals, changing the structure, and this entire structure is then moving the entire protein. So we have in those archaea, we have proton pumps activated by light. We have chloride pumps, and I'll show you in a minute, also sensory rhodopsins. And this is actually, I would say, the simplest way we know these days to harvest light. So if you compare it to photosynthesis, here we have only one protein and one chromophore. So it's a very simple system. So I'm still with archaea. So what we have in archaea, or in the early 70s, we knew about those two proton pumps, about the proton pump and the chloride pump. And we also have in those archaea sensory rhodopsins. So to sense light, enabling those archaea to approach light if they want, or if they see UV light, to go away. So this was known in the 70s. And when I joined the EDS lab, preparing all the metagenomic libraries that Peter mentioned, we were surprised that we can actually get pieces of DNA from many, many uncultured bacteria. I would say uncultured in 2000. Nowadays, whatever you see here, this is a phylogenetic tree of bacteria and also archaea, and all in blacks were uncultured groups back then. Now we have SAR11 in culture and others. But anyway, we had different pieces of DNA from those uncultured bacteria. And actually the second one I've picked for sequencing was a piece of DNA from SAR86. SAR86 is a gamma protobacteria. I understand that this year it was cultured back in Hawaii. So I'm waiting to see the publication. But anyway, so sequencing the genome or pieces of SAR86, we came upon a very big surprise. And this is actually the first bacterial rhodopsin, not archaeal, that was found. And this was really a big surprise. You can see here actually the original tree that we made in 2000, where you can see bacterial rhodopsins. Those are the archaeal proton pumps that were known, the chloride pumps, and the sensory rhodopsins. And here is sitting in this protein tree, this protorhodopsin. So it's actually impossible to say what protorhodopsin is doing only based on this tree. But luckily for us, because it's coming from gamma protobacteria, it's very easy to express it in E. coli. So what you can see here is E. coli. Those red cells here are actually E. coli expressing those rhodopsins. And of course, we add a retinal because regular E. coli could not produce retinal. So very easy. And based on this, we can really could show that those are actually proton pumps. And again, it was a big surprise because not only was it the first bacteria that was known to carry rhodopsins, it's actually a very abundant bacteria in the ocean. So in the photic zone, it can reach 15%, which is a lot. So this is 2000. So we knew about those pumps coming from archaea. And now we have a bacterial member, protorhodopsin. How abundant is this? Quite abundant. But you see here are different stations at the bottom. And then we check abundance of rhodopsins compared to chlorophyll-based photosynthesis. So we have oxygenic photosynthesis, we have anoxygenic photosynthesis. And what you can see here with the warm colors is that actually in most environments, you have more rhodopsins. I'm not saying this is harvesting more energy compared to photosynthesis, but it's more abundant in the photic zone. We now estimate that about 70% of bacteria in the photic zone would have the means to harvest light using rhodopsins. Okay, so this was in 2000, and what the picture I'm showing you now By the way, I forgot to mention this very nice slide was prepared by Keiichi, a collaborator I have from Tokyo. So we have those rhodopsins, or in this slide, I'm showing only the pumps, not only in archaea, we have them in different bacterial groups, in algae, in fungi, even in giant viruses. Most of them do pump protons, but not only. A recent find is a rhodopsin that can pump sodium, which was really a surprise. So we have a big variety of those pumps in microbes. So until 2000, more or less what we knew were the pumps, the rhodopsin pumps, so the chloride and the proton pumps. In 2000, 2001, a new family of rhodopsin was found, and those are actually channels in Chlamydomonas. So we don't have only pumps, we have also rhodopsins, which are channels. Moreover, we now have a new family of rhodopsins, which are actually enzymes. So you have a rhodopsin module connected to some enzymatic part connected to it. So, for example, histidine kinases. So histidine kinases that are activated by light. So we have a variety of different rhodopsins. So whatever I told you until now was found using metagenomics and then bioinformatics, looking for new rhodopsins based on homology. And in 2016, a student of mine, Peter mentioned her name, Alina Pushkarev, decided that she doesn't want to find rhodopsins based on homology. It's actually looking at under the torch, you say in English. So you're looking for something that is similar to something that someone found before. So we started functional metagenomics at those days, and the idea was very simple, looking for rhodopsins based on function. So one function is, for example, the ability to pump protons. So what you see here is a very simple set, 8 narrow pH electrodes, and here you have a 96 well plate, and below we have 96 leads. And in each well, you have a different clone from our BAC libraries or Fosmid libraries. And we're looking for positives. So a positive is whatever, when we shine light, is changing the pH of the medium. And this is an example of 2 positives from, plate 80. And you see here, it's a very simple assay. You measure pH in the dark, then you open the light for 2 minutes. You see acidification here, you close the light, it's coming back again. And then all those positives, you just sequence the back. And I must say that this was a bit of a disappointment, because what we found are things that we knew from before. So you can see here, this is a phylogenetic tree of, at that time, known rhodopsins. So you can see most of the positive clones that we had were all proto-rhodopsins, very similar to those that we knew from before. But one was a surprise, because this came from a gram-positive bacterium, and it was expressing the rhodopsin in an E. coli system, which is saying that E. coli are good for our screening. But again, whatever we found were rhodopsins that we knew from before. The surprise came when we decided to sample in Lake Kinneret, or the Sea of Galilee. It's a big freshwater system in north of Israel. And you see here Alina actually concentrating water from this lake. And we used another functional screen. And this is even simpler than to the screen that we had from before. And this is just to look for E. coli that would become colored. So E. coli, regular E. coli, whitish, yellowish, and if it does express rhodopsin in the presence of retinal, you would see those nice colors in E. coli. And actually, if you take this positive that she found in Lake Kinneret, here are membranes isolated from this clone. So this is membranes without adding retinal. And the minute you add retinal to it, you can see this, we call this deep purple. So it's really, I like this color very much. But at that time, we thought that we found, okay, another rhodopsin. The surprise was when we sequenced this clone, we couldn't see any rhodopsin in this piece of DNA. And then we had to go slowly over each open reading frame to see that actually what we had is a completely new family of rhodopsins, which we named heliorhodopsins. And actually those are so different to the regular rhodopsin. It's less than 15% on the amino acid sequence. So regular BLAST that we usually use would not pick it. So what did we find? So the other rhodopsins that I told you about from before are called type 1 rhodopsins, okay? Here, type 2 rhodopsins is actually the rhodopsins that we have in our eyes. And you can see the topology in the membrane is very similar between those two families. As Peter mentioned, the new family is inverted in the membrane, completely different. This was the first rhodopsin family that is sitting completely different in the membrane. And of course, only 15% to the type 1 microbial rhodopsins. So this was really a big surprise. I mean, no one expected this, and it was a big surprise. Moreover, if you force alignment on those proteins with the type 1s, so you can see here a tree, so you can see here the other rhodopsins I mentioned. So you have here protorhodopsins here. Where is bacteriorhodopsin? Bacteriorhodopsins here. And you can see the entire new family. Now we can find them using BLAST, and they are everywhere. Everywhere in a lot of microbes that we did not suspect could sense light. So methanogenic archaea, for example, Asgard archaea, gram-positives, hyperthermophiles, even giant viruses. So they are everywhere, and we were just waiting for the functional metagenomics to find them. So I'm mentioning only two groups. I've picked them because they are it's more interesting to mention them. Of course, it's found in others too, but surprisingly, Asgard archaea have heliorhodopsins, and also giant viruses that attack algae would have those rhodopsins. We have no idea what those rhodopsins are used for. We know that you can sense light, but that's it. We don't know yet. Okay, so I'm wrapping up. I want to acknowledge my lab and my collaborators. Here, by the way, you see Alina collecting water. In this case, it's from a pit lake up north. It's called Lake Hula. We can see a lot of birds over there. So I would like to thank my students and my collaborators, and of course, the money sources that we've used. And this will be my last slide, and this is actually to tell you that when you do metagenomics, it's like opening a treasure box, because you don't know what to expect. You have some hypotheses, but sometimes you can really find things that were unexpected. Peter, back to you.
Thank you very much, Oded. That's fantastic. We do have time for a few questions. And Dr. Gavin, am I correct in saying that our participants should go ahead and post those questions in the Q&A?
Absolutely.
All right. So go ahead and send those questions on in, and we'll pose them to Dr. Beja. Don't be shy, folks.
While we're waiting, I'll pose one, which is, have you found rhodopsins from microbes in inhabit environments that don't seem intuitive that they would be sensing light, or you can't predict what this would be used for?
Yeah, we see a lot of heliorhodopsins that are coming from microbes, for example, 2 kilometers deep, 3 kilometers deep. So they don't see regular light, I would say. We have now some speculations about perhaps infrared light, or perhaps things that are not really connected to light, so sensing, how would you say it? It's connected to light, but sensing heat, for example, but perhaps without the retinal. We are still checking this.
Wow.
But then, you know, most methanogens are not necessarily in environments that would see light. For example, Asgard archaea usually are found deep or inside sediments. So another mystery of those heliorhodopsins. We have no idea.
Wow, thanks.
Excuse me. That is very cool, Oded, and I have a number of other questions for you on that. But let's turn to our audience. From Jeff Gralnick. Very exciting. Do you think there are other types of cofactors that could be used by rhodopsins beyond retinal?
I did not mention, but there are some rhodopsins called xanthorhodopsins. They use retinal, but they also use an carotenoid antenna from the outside. So you can really see energy transfer from the antenna into the retinal. But what we know is that all rhodopsins that are known until now would have retinal inside. And actually, I did not mention that this retinal is covalently linked to a lysine in transmembrane domain 7. But the minute this would not be a retinal, then you wouldn't call it a rhodopsin.
Rhodopsin, yeah.
But yeah, I mean, and we are looking all the time. The microbial rhodopsins would have all transretinal. In our eyes, we have 11 cis retinal, and then depends on the photocycle. In microbial rhodopsin, it would cycle from all transretinal to 13 cis retinal, and then closing the cycle, going back to all transretinal. But no, only retinal within those rhodopsins.
Oh, that's very cool. Thanks, Oded. So Hyman Hartman asks a question: how similar are heliorhodopsins to the rhodopsins in the eye, presumably the human eye?
Okay, so there is a connection between the type 1 and the heliorhodopsins. There is no connection between the microbial rhodopsins as a whole group to what we have in our eyes. So it's only, the similarities are only in structure. So 7 transmembrane domain protein, a lysine in transmembrane domain 7 in both families, retinal connected covalently to this lysine. But if you look at the primary sequence, no signal. So it could be that this is convergent evolution, or it could be that it happened so many years ago that we lost the signal. So it's actually an open debate whether those had a common ancestor protein and we lost the signal, or whether those are two completely separate families.
Outstanding. Thank you. Any other questions for Dr. Oded Beja about rhodopsins, SAR86, functional metagenomics, life at Technion, if you want a job?
If I may add, is that, for example, the different rhodopsins are found in so many microbial groups. So, for example, SAR11, the most abundant bacterium on Earth, would have rhodopsins, proto-rhodopsins actually. So I would say, as I mentioned, 70% in the photic zone of bacteria are using those very simple way of harvesting light. And I found it amazing.
Well, just to add a little bit to that, I think it's, if we look at, if we remind ourselves that all living organisms have to harness energy from the environment in some way or the other. And if you think about the tremendous amount of energy that hits the Earth as solar irradiation, right there, it would make sense that you would see strong selection for an evolution of systems to harness that, right? And I think the ubiquity is what really I think is amazing. And says a lot about the connectivity of organisms in our biosphere, right? And the prevalence of gene transfer and the like. But switching gears, there are some two more questions coming in. Rashid Sabahi is asking, is there any biological activity of rhodopsins? I'm not entirely sure what that you mean by that, Sabahi. If you could try and clarify a bit. Let's come back to that. Hyman asks another question, Hyman Hartman, why is there no photosynthetic bacteria with proteorhodopsin? So let's answer Hyman Hartman's first, and Rashid, please expand on, is there any biological activity? But go ahead, Oded, for Hyman.
So nowadays we know about some bacteria that are doing anoxygenic photosynthesis, and at the same time, they would have also rhodopsins. Why? We have no idea, because not regular, but anoxygenic photosynthesis is much more efficient than the rhodopsin system. So why would you have those two different light harvesting systems in the same bacteria if you have photosynthesis available? Could be, I don't know, availability of iron. I mean, this is perhaps a question to the previous speaker. If you don't have iron, I would presume you cannot have regular photosynthesis. You can have rhodopsins, perhaps.
Interesting. We have, so before we leave Rashid Sabahi's question, is there any biological activity of rhodopsins? Dr. Beja, do you have a sense of what that question's about?
As I mentioned in one of the slides, all we know are, the different rhodopsin pumps, usually for energy, but not only. So for example, and now I move to channels. Channels are used for sensing light, so Chlamydomonas could react to light using those channel rhodopsins. Enzyme rhodopsins are also used to sense light.
And so Rashid Sabahi clarifies, asking about antimicrobial activity or antioxidant potential.
Not that I know.
Not that you know of. One last question, briefly, please, Dr. Beja. Alex Hall asks, what function do you think the viral rhodopsins might be having?
So in those algal viruses, some of them use channels, not rhodopsins, which are important for the entry, because you have the fusion of the membranes. Those viruses would have also membranes around them, so it's not only a capsid. So it could be that those rhodopsins are actually on the particles. We are now trying to tackle this, but also we know that some of them, after infection, they start to express some of the rhodopsins, and we don't have yet an idea what exactly those are for. I guess for changing some of the light sensing of the host, changing it. Because I can mention, Peter mentioned it, that we also work on cyanophages, which do carry photosynthetic genes, for example. So we also already showed that those are actually changing the photosynthetic apparatus of the host. So it could be that those rhodopsins are used similarly to change the sensing of the host, the light sensing.
Excellent. Thanks, Oded. Appreciate that. We're going to hold off on questions for now, and we'll save them for our upcoming Q&A. Just a reminder for those of you out there who are on Twitter as well, our hashtag here is hashtag MSI Symposium. So feel free to tag us as you tweet. Let me turn it over now to Julia to introduce our next featured scientist artist. Julia, it's all yours.
Thank you, Pete. Our next scientist artist is Dr. Tal Danino. He's an associate professor of biomedical engineering at Columbia University. The themes of his work and art connect the hidden microscopic universe with the impact it has on our macroscopic society as a whole, and convey where we are today in an increasingly digital and biotechnological world. So now we're going to run a quick video featuring more of Dr. Danino's work.
My name is Tal Danino, and I'm an associate professor in the Department of Biomedical Engineering at Columbia University. Aside from leading a research lab, I also create artworks using living materials such as bacteria. And the themes of my work connect the hidden microscopic universe with the impact it has on society and conveys where we are today in an increasingly digital and biotechnological world. As a student, I saw beautiful images and videos of engineered bacteria under the microscope that seemed like they could be works of art. I used this in science communication and outreach efforts, and it wasn't until I was a postdoc that I collaborated with a few artists, such as Vic Muniz and Annika Yi, and art started to inspire the science I was doing. And in addition, allowed me to explore more philosophical themes such as identity and commodification of nature and genes in the human body. From then on, both art and science became part of how my work evolved in these areas. The birth of a concept starts with thinking about how something is connected to microbes. There's almost always a connection. And we go through a collection of bacteria step, we grow bacteria, and then we play with their arrangement, preservation, and the format that they're displayed in, whether that's prints or objects or whether they need light boxes. Images have always played an important role in communicating and inspiring new ideas. Art is a tool that we use to explore abstractions more freely, and living bacteria in the work constantly responding to the environment helps rethink the type of work that we do. And so this generates new questions, visualized concepts, and connects the dots from our own research to the larger narrative that's relevant in today's world. In some cases, many of our projects have even shown direct examples of how artistic processes could help develop techniques for scientific inquiry, such as spatial printing and patterning methods. One of my favorite projects is Transcrobial, and in this project we used machine learning and generative adversarial network scans and incorporated them to mimic and expand upon bacterial evolution and pattern formation beyond what is realizable in the physical world. We like to share our work to as broad an audience as possible, and using visual arts allows us to communicate science in a very robust and universal way that transcends language and the complicated jargon of science that is often difficult to understand. And while scientific research is often very impactful, it doesn't always connect with the everyday experience. So create art to bridge scientific themes and engage with people at a fundamentally deeper level. And ultimately, the hope is that these works can allow for inquiry and self-reflection, connection with the human experience, and help shape dialogue and future policy for how science integrates with society.
Now we'll be sharing a PowerPoint with more of Dr. Danino's art, and this slideshow will loop a few times. So again, you're welcome to stretch, get some water, coffee, go to the bathroom, and our next talk will begin after the slideshow. right, back to you, Pete, to introduce the next speaker.
Thank you, Julia. And I mean, the scientist artists here are blowing my mind. And so I really appreciate Dr. Tal's work. And I'm excited now to introduce our final speaker for this morning. It is Dr. Melissa Duhaime. Dr. Duhaime is an assistant professor of ecology and evolutionary biology at the University of Michigan. Her laboratory studies microbial interactions in the ocean and in lakes on particles to better understand their consequences for ecosystem function and environmental health. In the ocean, Dr. Duhaime's lab studies how virus-microbe interactions influence the fate of marine snow particles, and in lakes, they investigate how microplastic biofilm activity influences the transport and fate of plastic pollution, as well as carbon and nitrogen cycling. Now, I say I've never had the pleasure of working alongside Dr. Duhaime, but her work is making waves, and I just love all the work that they're up to. So I hope you'll pardon the pun. But it is also cool, if I may say so, to see Dr. Duhaime and others have such a neat sort of presence in their sort of outreach and public-facing work. And she's also got a great Twitter handle, Envirophage. I mean, that's pretty cool. But I am really excited to hear her science, and I'm going to turn it over now to Dr. Duhaime to share with you all the great work that she and her lab members have been up to. So Dr. Duhaime, the screen is yours.
Thank you, Pete. Right. Bear with me as I sorry, tiny screen.
No worries, it looks good.
I think I have, all right, I think we're ready to go. So yeah, thank you for the introduction. And indeed, we do different types of outreach, but the heart of what I'll focus on today is the research. So there are two arms of the work that we do in our lab, and today I'll talk about the work that we do looking at the microbe and virus interactions and what that might mean for ecosystem scale processes. So we'll look at the virus's guide to infecting bacteria, how to reprogram a host to meet your replication needs. So we'll start with the Earth is our major ecosystem. And we've come to appreciate that microbes rule the world. I think many of you heard in several seminars and readings that the microbes are driving major redox reactions that are essential for life and different biogeochemical cycles. And also, as we look more closely at different classes of holobionts, we're beginning to appreciate the extent to which animals and plants are essentially biomolecular networks that are composed of the hosts and the microbes. So I think more and more we're becoming familiar and comfortable with this idea that microbes rule the world. But what we think about in our lab is the fact that viruses are ruling the microbes when they interact. And the rest of the talk will be an inquiry into how and why that ruling of the microbes happens and what the implications might be. also a general framework for how we're thinking about this. So these are some of the first studies that we've been doing in my lab group, and I think have become pretty foundational in the things that we are interested in or guiding us to our future interests. So we know that, due to, or as a result of decades now of research looking at viruses, first it was like, wow, viruses are there in particular ocean systems. Most of this talk will be referencing. And then it was, wow, there are a lot of viruses there. So we know viruses are incredibly abundant in most habitats that we have looked in. In fact, in most habitats, they outnumber other microbes, sometimes by several orders of magnitude. So that could mean that in every drop of seawater, there could be tens of millions of viruses or more. So this is a big number, staggering astronomical numbers. I think something on the order of 10 to 30 viruses on the planet has been estimated, and that's just really, I have no way to really appreciate what that scale means. It's a big number. And that number has inspired researchers for decades now to ask questions, kind of following up on that observation of numbers, such as why are the viruses there? Why are there so many? And then what are the population and community and ecosystem level consequences of those viruses where they're found? So really, when you put that together, this notion of relationships, it implicitly invokes the principles of viral ecology. And so in other words, thinking about how viruses interact with microbes in their communities, as well as with the environment in which they are found. So these three entities, knowledge of the mechanisms that underlie the interactions between virus, microbe, and environment, I think really underpins our ability to determine whether it's necessary to, for instance, include knowledge of viruses and their impacts in ecosystem scale models, the way that folks are striving to do and succeeding in doing with their microbial hosts. If you think back to the introduction, microbes rule the world, but viruses rule the microbes. How much of that ruling of the microbes do we need to know in a qualitative and quantitative sense in order to best model ecosystem processes? So that's at the heart of the work that I'll show the beginning of today. So if we tease apart, I can give a few more examples of what these interactions actually look like. So between microbes and viruses, you have interactions, so between viruses and other organisms, such as microbes, that could involve horizontal gene transport for the replication process itself, which results in lysis. So that's obviously an effect of viruses on microbes. Diversification of both the viral and host populations. Lysis generates lysis products, and it's been estimated that in some habitats, the lysates of a viral infection can serve as food, comprising up to 50% of the diet of heterotrophs that are found in that system. And viruses, while they're infecting their hosts, are also known to metabolically reprogram their host. And I'll talk more about that in detail. And these other interactions between the virus and the environment itself includes processes that influence the abundance and the distribution of viruses, for instance. There are abiotic processes that influence the abundance and distribution of viruses, such as photodegradation and different hydrodynamic transport processes. But then the impact of viruses, so this is free viruses, virions we call them, impact of those viruses on the environment include things like the ecological stoichiometry of the system. So virus particles themselves are reservoirs of carbon and nitrogen and phosphorus, and have also been implicated in kind of how iron moves through marine systems too. And this kind of final interaction I think we know most well about between microbes and environments elemental cycles, environmental engineering that microbes are able to do to transform and create their own microhabitats. And the environment provides energy and substrates and really defines the limits of life. But what happens then in a specific instance where that microbe is the microbial host? Well, an infection ensues. And so and this new entity is created that's been termed a virocell. So a microbial host, well, the virocell itself is a cell unit whose function is no longer to produce two daughter cells, but rather to produce viruses or free viruses we call virions to propagate viral genes. And this was a notion introduced 10 years ago now by Patrick Forterre that has really been transformative in the way that I think we think about the possible implications that viruses could have on the environment. So a microbial host that's infected by a virus is distinct from an uninfected microbe. So this new cell, the virocell, is then, as a result, a combination of both a host and a viral genome, so a new novel genotype, and it therefore also has new resource needs, metabolic functions that are possible, so new possible phenotypes that were not there before. And it's really the magnitude of these interactions with, in the end, the environment, so virocell environment interactions that has, I guess, led us to come up with some new thoughts. So virocells interact with their environments in a few ways that we are interested in exploring. So they themselves, the virocells, are organic particles with their own elemental stoichiometry that's distinct from that of a free virus or a host. But they are also still using energy. This reminds me to our first talk today about the concepts of life. So we often also delineate those. What is energy doing? What is carbon doing? Is this thing still alive? What are electron donors and acceptors? And how is that different from that former uninfected host? So just to tie it back to that first talk. So yeah, they use energy to transform organic matter. They also may release and excrete compounds into the environment. And that's another key point for us that we're, I think, far from fully understanding, but the data I'll present today start to bring us towards hypotheses about what that might look like. So I mentioned viruses can reprogram their host metabolism during infection. That means then that in the form of virocells, a substantial portion of microbial cells in any given habitat could be serving the mission of viral replication rather than cellular replication. And this is important in environments like the ocean, where there are estimates that nearly half of the surface microbes are infected with, or may be infected with viruses at any moment. Although, give the asterisk that that is a number that certainly needs better parameterization, I think, but it's a possibility. And so what is the impact of this virocell on its environment? Well, we think of that in terms of an environmental footprint. So I mentioned the possibility for substrate transformation, nutrient uptake and release. And so really recognizing virocells are where this ecosystem action happens, and to better understand the virocell and perhaps build better ecosystem models, there are several questions that arise before first embarking on that pursuit. And I think the first order question is, should viral infection even be included in ecosystem models? So I think we don't know that so far. We've talked about possibilities, possible implications, but it gets complicated very quickly. So if we do include them, for instance, would we need to model each virocell? So that means each distinct virus and microbe interaction. Would we need to model that in every possible environmental situation? Or rather, might we begin to see certain universal features of infection footprint begin to emerge? And what are the bounds of the universality of that? So does that environmental footprint differ from virus cell to virus cell and across environments? Well, those are the types of questions that we're interested in pursuing. And so today I'll focus on the first of a series of experiments that we began, where to better kind of to approach a question, we sought to bound and describe the variation in a virus cell footprint that one might expect given, in this case, varied phage host pairs. So in this case, we tweaked who is the virus infecting a single host. We then followed up on that experiment to look at another parameter to kind of explore the effect space to get at the impact of the virus cell. I won't talk about the results in detail of that experiment. It's unfolding currently. But with these changes, we tweaked these kind of different parameters and then track the molecular responses with multiple omics data types. And so to summarize the first question, how do virus cells differ? Specifically, how do changes in virus cell genotype lead to changes in their phenotype? And by differ, specifically, we think about differences in terms of metabolic reprogramming strategies, work, I'll talk more about that, measures of fitness, viral fitness, and altering their capacity for altering the environment. All right, so first I will introduce you to our model system. Well, now I will introduce you to our model system, which we work with a host called Pseudo-Ultramonas, which I'll abbreviate as PSA in the talk. It's a gamma protobacterium. We're interested in it in ocean context because it dominates ocean particles, which are important to the biological component of the carbon pump, which brings carbon out of the atmosphere into the deep sea. And it's also been observed through omics data that Pseudo-Ultramonas is the heterotrophic genus that's most correlated with ocean carbon export. So that's one more motivation for why we're attracted to this system in terms of ecosystem impacts. And so we look at PSA with by infecting it with two unrelated phages. So one HS2 on the left, a cyphovirus, and one HP1 on the right, which is a potovirus. And importantly, these are very, very fundamentally different viruses. They're genetically distinct. The only area of overlap in their genomes they have is a small domain in a tail spike protein, which we suspect is involved in host recognition, given that they both infect this host. In the genome itself, there's no obvious metabolic genes that might connect it with nutrient metabolism, but there are the canonical phage proteins involved with invasion and replication and assembly and lysis. So those are our players in the system. And one note on another trait that differs between the viruses, when you infect this host, early on we noticed that there are HS2, its burst size is over 60% larger than that of HP1 when infecting the same host. So this to us suggests that HS2 has a higher fitness under the same conditions of infection. Those are all important points to carry on into the experiment. So our experimental setup first addressed the question of how do virus cells differ when we change the virus of the virus host pair. That's what it looked like. In a phosphorus-rich environment, we grew up the PSA host and allowed it to grow, and then we infected it with the two viruses, and in a time-resolved fashion collected transcriptomic data and proteomic data. And by time-resolved, I mean that we captured the discrete stages of infection. We sampled along a time series to try to capture those infection stages for both. So now into the results from the transcriptomics and proteomics data. I'll show two sets of data. The first, remember, collectively, the Vero cell is the viral genome and the host genome, but I'll first show how the host component of the Vero cell genome compared across these three classes, uninfected and the two viruses. And then we'll look specifically at the viral component of the Vero cell genome. So first, in the host, let me see this one. There we go. So overall take home is that the results show that the Vero cells differed dramatically, and that specifically we observed that HP1 here, the red, immediately shut down host transcription. And we were able to observe that over time by looking at the counts of transcripts fitted to a linear effects model. So in the host transcriptome, HP1 immediately shut down host transcription, while the HS2 Vero cell really deviated from the host transcription profile only at the end of infection. And in the host proteome, what we observed was that HP1 translated more host proteins at a faster rate compared to the uninfected. And then at the viral gene component of the Vero cell, we saw that HP1 genes were immediately transcribed, whereas HS2 lagged, and the HP1 proteins were translated at a much faster rate. So what are we seeing here? We see HP1 Vero cell seems to be in some ways working harder for its infection, though we know it's a less fit infection. It produces fewer viable viruses at the end. And so we next wanted to look more at what is the nature of this work. Those are just global perspectives of transcripts and proteins. Was there some Vero cell specific metabolic programming happening? So to answer that, major takeaway is yes. I'll give that off the bat. And what you see on the right are the ways in which kind of elements of sulfur metabolism were reprogrammed differently from the host and also differently from what was observed in HS2. Important for us is that we saw evidence that HP1 Vero cell may be importing sulfate based on the transcription profiles, and that given overexpression of other genes in this pathway, it likely reduces that sulfate to cysteine. And then moreover, we observed evidence to suggest that that cysteine was being used by the HP1 Vero cell via a specific glyoxylate bypass of TCA. So in some ways, I think of this as HP1 bringing in sulfate to kind of harvest in its garden the cysteine, which then could be consumed or used later for energy. So that was the major observation, observed an HP1 that was not seen in HS2. And so why, so this is more drastic reprogramming. Why does HP1 seem to work harder with this drastic reprogramming? So to approach that question of why and understand the concept of work, we thought more about what the currency of infection might be. And that led us to the path of nucleotides and amino acids, and thinking about who has more and who needs to get more and who has more of what they need to begin with at the start of the infection. So we know that phages, for instance, are master recyclers of nucleotides and amino acids, but as such, they will be constrained by what's already available to them. So we found that HP1 has a greater mismatch. So here on the bottom axis, you see increasing difference between a given phage and its host codon usage. And along the y-axis are the different amino acids encoded by the codons. So you see the cloud of orange spreading all to the right tells us that HP1 has the magnitude of difference in its codon usage is much greater than that of HS2. And so given that translation is the most energy costly task in infection, this led us to the notion of this kind of mismatch burden of evident in the codon usage. And then further evidence to support this is that we found that the HP1 virus-cell overexpresses host tRNAs, so the 43 host tRNAs from the very start of infection, and that's represented by this like warming here. And that's in contrast with the HS2 virus cell that does not overexpress tRNAs, in fact, any. And presumably it's because it's able to use its host's tRNAs. And so this brings us to a notion of the HP1 virus cell needing to compensate early for the burden of its nucleotide and amino acid mismatch by some form of work that we observed in rates of protein synthesis and just taking over the host machinery. And as well, in doing so, influencing the metabolic reprogramming, such as through sulfur import, carbon cycling, and energy and redox elements. And thus, overall, has the potential for a different environmental footprint of infection. So again, remembering that the only thing that differed here was the virus itself. And so this was really exciting for us because by looking under the hood of this virus-cell activity, we could start to build inferences about what the ecosystem impacts could be of an infection, what underlies variations in that impact, and then possible mechanisms that could explain the differences between the virus cells. And as well as a possible vision for how to connect some of the dimensions of viral ecology. And so what I mean by that, for instance, are life history traits. So we talked about burst size differing between the HP1 virus cell and that of HS2. We then observed that there's this metabolic rewiring happening to meet the energy and resource demands that was evident in both, but more drastic in the HP1 virus cell. And then we also identified the potential for an intracellular energy resource trade-off, whereby increasing the phage host intracellular resource complementarity, you then will decrease the metabolic effort that's needed to carry infection to completion. And based on the inquiry into the codon usage, we were able to offer this phage host genome complementarity as a possible driver of the magnitude and the nature of the ecosystem footprint. So how do virus cells differ? We've shown that the physiology and metabolisms of the virus cells is distinct from that of the uninfected. I'll add, though, that this is largely supporting evidence to suggest and demonstrate the same. That was not a surprise. But what was a surprise was that, that was further supporting the notion that we may need to consider those two infection states separately, infected versus uninfected. But also we observed that, like viral fitness, right, the different burst size on the same host, the nature of the metabolic reprogramming, so that reprogramming itself is a phenotype of a specific phage host pair, and our data support the growing evidence for this. But still, we recognize this doesn't bring us much closer to our first order question of do viruses and these interactions and virus cell broadly need to be included in ecosystem models? And for that's where we will get our a little bit more of an idea about the answer of that by changing that environment parameter. So let me see where I didn't include this, but so we start to recognize that there are some universalities in the virus cell response to P, the phosphorus limitation that we impose in our experiment. So there's some universalities. And even early evidence is suggesting that the environment may matter more than the virus cell type. So it's in terms of reprogramming, but what matters are really the ways that reprogramming interacts with the environment. So for that, we need to, and have explored changes in the exometabolites of the cultures. So now we're looking at the signs of the transformation of organic matter in the media to evaluate whether the observed changes in reprogramming, what it means for differences in that environmental footprint of the infection. And so the next task will be to kind of look at the relative impacts of these changes. And I'll say, one of the drawbacks of our current data set is that it's not quantitative. So I see the future of much of this work also pushing the limits of what the omics data can do in terms of absolute quantification. So we can begin to look at fluxes and rates more quantitatively. So I'll leave it at that and give massive hats off and acknowledgements to the lab members and collaborators that have pushed this. And largely that is Christina Howard-Verona from the Sullivan Lab at Ohio State and Morgan Lindbach as a graduate student in our lab at Michigan, who have really spearheaded both of these stories, which are incredibly complicated to piece together between the metabolisms and multiple omics types, and making sense of finally not just describing what the patterns are, but trying to fit it in an ecological framework to answer, like, so what at an ecosystem level. And we're not there yet, but this is how we think about these problems, and this is where we hope to go in the future.
Wonderful. Thank you very much, Dr. Duhaime. That is very, very, very cool. I appreciate your, I was a soft spot for those linkages going from observations at a genomic or even transcriptomic level to function. So it was really great. I also have to confess that my kids dropped a handful of fried bacon on my keyboard as I was introducing you. So, so work from home. What can I say?
That's amazing.
So forgive me if I fumbled your introduction at all. But
no problem.
The good news is we are now at our question and answer session. And for you all as our viewers, this is your chance to ask our amazing speakers, all 5 of them, anything and everything. So please don't be shy. We have quite a nice chunk of time here. And the idea is just to give you all a chance to kind of have a dialogue. As I've said before, there's really no such thing as too simple or complex or dumb a question, right? We all come from different perspectives and we're at your disposal. So please send them on in and we'll hang out and field your questions. So please type them in the Q&A box there. As a reminder,
also, if somebody's brave enough to want to ask a question out loud, or if it's particularly difficult to type, you can raise your hand and we'll see that, and then I can invite you to ask it aloud also. Yeah, absolutely. Yeah, and it's it's your choice, right? And you don't even have to be brave. Just raise your hand. I mean, we're all harmless. So here's what we have so far in our Q&A, and I'll give the rest of you a chance to chime in. So Triveni, you started by asking a question, but it looks like a phrase, and maybe it got cut off. If you're still on, Triveni, you were asking a question presumably to Oded about rhodopsin, the biofilms. So if you want to chime in and elaborate a bit, that would be great. So we'll wait on that. Alex Hall asks, do you see any evidence for phages evolving their codon usage over time to become more aligned with the host, or does the difference in codon usage confer an advantage to the phage? That means that they maintain a different repertoire of tRNAs. That's a cool question. And Alex says, thanks for a great talk, Dr. Duhaime.
Sure, I can start to tackle that, though I'm not the expert in that, meaning that not from our data have we observed that, but there's evidence for phage host kind of coevolution that is observed in like amelioration of phage and host genome, which is presumably driven by codon usage. But yeah, so on the one hand, the fitness of a virus and its host are kind of inextricably linked, as well as that of their evolutionary trajectories, but they're not obligately interacting with one another and exclusively interacting with one another. So you have mixed into this the need to think about host range and kind of the balance between, in a given system, viruses with broad host ranges and the trade-offs that might confer and how they may manifest at the genome level of both, as well as the virus host pairs or viruses that are very host specific and how that might manifest at the genome level. So there's a spectrum, or there are multiple axes along which things are operating that can, I think for those viruses and hosts that are specifically aligned, you will see amelioration and codon, yes, evolving similar codon usage patterns, but it's complicated.
Very cool. Thanks. Often is complicated. Let's see, Dr. Gavin, do we have any hands up?
Yes, actually, from Amanda. So I'll just allow you to talk here and feel free to voice your question.
Yeah. Hi, this is Amanda Fotenhauer. I'm from the University of Michigan. And my question is for Melissa. So you were talking about how these viro cells differ maybe in the metabolites that they're able to make or produce. I was wondering if you can also look at just individual interactions. So if you're able to kind of grow these cells and look at like cross-feeding with one other species that they normally interact with or compete with, and just kind of get like on a micro scale, like a single interaction and see how that changes it.
Yeah, and you're thinking about this in the context of virocell?
Yes.
Yeah, so that's really interesting. I was teasing apart kind of the details of this as I was putting this talk together, because sometimes I realized we were referring to the virocell ecosystem footprint, and other times I was querying whether we need to be more precise by saying environmental footprint. And I think that what you're asking hits upon that distinction and how I think we'll try to use it in the future. So when I think of ecosystem, I think of all the interacting parts, which includes the environment and the other organisms. And so then so what we've observed so far, I think, is more environmental footprint, like those abiotic components of the environment. How are they being affected by the infection as it proceeds? And so then I thought, well, cool, the next experiments should be looking at the ecosystem. And they were pretty much as you just proposed. What if we make a mock community and infect just our, put in our model phage host pair and look at the impacts that they may be having on other community members. I think there are a number of ways you could ask that, also probably in situ, but so not necessarily an answer, but pointing out the distinction between ecosystem and environment, I think, is important to then incorporate that idea.
Well, thanks.
Thanks, Amanda. That was great.
Yeah. Now, I have a couple other questions for Melissa, but let me start with a question from Heather Olens that I'd like to pose to all of our speakers this morning. Heather asks, what are your favorite environmental micro or microbial ecology investigations to carry out with novice undergraduates? So let's go from Arpita to Oded to Melissa, if that's okay. What are your favorite environmental micro or microecology investigations?
Phototrophs. Always phototrophs. They're so beautiful. There's so many shades and they're so easy to isolate. You don't have to go very far. You can go in your backyard. So definitely phototrophs.
I would say bioluminescence.
Oh.
This is also so easy to isolate them, and then you can ask so many questions. Worm sensing, and others. Why do they need it? So bioluminescence, I would pick.
Yeah, well, those are great. So we got phototropes, bioluminescence. Melissa, what's your take?
Well, I was thinking back to like the most memorable aha moments in some of my lab classes. One was learning how to use a reference manager and that you can automatically generate citation lists. Not necessarily explicitly microbiology, but that is something that wows them every time. And I think also in silico, there's so much 16S amplicon data available freely, and empowering the undergrads with the skills to manage that. So getting them into R and working with those data, that has been another place where I've seen really immense potential for growth because, well, clearly we think about the data in ecological terms. So you can bring in many different concepts of ecology in the context of microbes, and then think about why these patterns may be emerging.
That is a good point to remind us all that there are these massive publicly available databases that are accessible. And I could imagine, Heather, you know, that the three ideas shared here, you know, could each be a part of undergraduate experience, right? Because they could each sort of tell you something different about microbiology and sort of address different questions. Those are great responses. Thanks, friends. Melissa, we have a question from Krista Longnecker about whether you have hypotheses about which metabolites will show the greatest difference across the two virucells.
I have to admit, when I saw this pop up, I had to quickly write to Morgan, the grad student, like, what do you think? So I know she's watching this. And I can say her response is that, let me read it first. Yeah. So another concept that we're trying to layer into this thinking is the notion of a bottleneck. And actually that becomes much more important when you bring in the idea of the environment and thinking about where is the bottleneck. In the data I showed today, we saw that it was at the level of intracellular resources. That was where the, things were hitting the limit, and that's where you start to see differences manifest because the phage were able to get to the point of transcription translation. But in the case of extremely limiting environment, they may not even be able to get there. And so then the bottleneck would be more at the energetic level from resource limitation. And so bottlenecks are important. And so I can see that's probably inspiring Morgan's response, which is the metabolites that relieve the virucells of the greatest bottlenecks. So the biomolecules in the lysates, such as amino acids, could be a metabolite that would have greatest difference between the two. In the first experiment, though, I'll say we don't have the exometabolome for that. We only have it for the environment.
Well, that's very cool.
I have a question, if I can butt in with one. Arpita, I'm really curious how your lab member managed to delete the essential Rubisco genes.
So that's the cool thing about some of these phototropes, that they can grow aerobically like we do. And so that's how he got the mutants. So he grew them aerobically, and then the cells didn't require the Calvin cycle for autotrophy anymore. And then he would make the mutants, grow them aerobically, and then wash them and test them under the essential conditions of autotrophy. But even then, it took a very long time. It was almost like Rubisco had some function on the aerobic growth, because it was like it took 2 years to get mutants in aerobic conditions. So.
Wow, yeah. Do you feel paranoid about those mutants? Have you sequenced them to make sure that everything else is okay?
Yeah, and they used to get very easily contaminated with wild type, so we had to keep the thing, we had a lot of tests to make sure it was always clean and
sure.
Really.
Yeah, go ahead. Sorry.
No, go ahead.
Yeah, so Arpita, that's interesting. So if I heard you, just to make sure that I'm understanding this and that our audience participants are hearing this correctly. So autotrophy is used primarily under anaerobic conditions. Is that right?
In these organisms.
Now you grow them aerobically and heterotrophically.
Yes.
Right. And that's interesting because so often there is this idea that heterotrophic growth can be so much more energetically favorable, and yet here you are seeing the absence of key parts of the CBB cycle having an impact. Just wild speculation. Any guess on what might be going on there?
Yeah, so I mean, when we were making the mutants with heterotrophic growth and rich medium, like, yeast extract and peptone, and we were having so much trouble for the CBB cycle, we started thinking that maybe when we were growing them heterotrophically under some conditions, they go anoxic. And when they go anoxic, they might require carbon dioxide and for some growth parameters. And for that, to assimilate that, they still require Rubisco. I don't know. I haven't explored, what is going on, but it's possible that, eventually as we grow them, oxygen becomes limiting, and then they're not doing just heterotrophy alone anymore.
Right.
It's almost like mixotrophy.
Right,
and
indeed.
when, it's becoming relevant. But yeah, we learned a lot through the deletion of essential genes in that way.
Right. And of course, there's all the states in between aerobic, sort of oxic and anoxic, right? There's everything that sits between those two. And what's happening under sort of hypoxic conditions and where the tipping point really is also intriguing. That's a really cool point.
Yeah, especially in the environment.
Yeah, right. Absolutely. Cool, thank you. So we have a couple of other questions here. So Hyman Hartman poses a question. Cyanobacteria carry out 1/4 of the photosynthesis on the planet. What is the effect of viruses on this geochemical carbon cycle? Anybody want to take a shot at that?
I can start. We showed what was that? 2009 that some cyanophages do carry photosynthetic genes from photosystem 1, from photosystem 2, and later on we did show that they actually let the infected cyanobacteria carry on photosynthesis. Otherwise, at least photosystem 2 is falling apart during infection. So actually, I would say that infected cells, at least those that I know, cyanobacteria, would keep on doing photosynthesis during infection. And actually, viruses that carry photosystem 1 and photosystem 2 genes are actually also, like the virus cell, they are changing linear photosynthesis into cyclic photosynthesis. So all they care is to make more ATP. They don't care about CO2 fixation, so they change also the photosynthesis of the infected cells. But photosynthesis is going on, at least in the ocean, in the photic zone. This is what I know.
Yeah, that's very cool. The let's see, we have another question that came in. Dr. Duhaime, would you mind answering this one or sort of sharing this out loud? The question was from Karen Farner about your interest in fitness seems based on replication of viral particles. Have you thought about fitness in terms of the ability to lysogenize?
Yeah, so for those of you, I think, can folks see the answer that I just wrote in the broad crowd? So I answered this in the text, including a reference there. So yes, I did forget to explicitly say that this talk was very much synthesis and thinking about the lytic phage infection process, which is, we don't know really relatively, but it's only one of a number of different strategies for infection. And I expect many of the things that I talked about today to differ in scenarios with other infection classes. And an important one is exactly because we do try to frame it in fitness. The definition of fitness is very important, but you don't want a definition that is different for each viral class. And so a lot of good work and thinking has gone into this, redefining and defining viral fitness. And I put a link to a recent review on the topic of kind of the rules of life and how viruses intersect with that. And there they lay out kind of explicit definition of viral fitness that is universal across all infection classes. So hopefully that can help to kind of found that it's really important. So I guess the important thing to recognize is it's definitely not viral particles, I will say. And I tried to make that explicit with a small reference that it wasn't the count of viruses that we used as a fitness measure. It was the number of viruses that were able to carry out infection. So able to move on to the next possible host, but in very lab forced manner. So we eliminated one of the components of fitness, which is that extracellular foraging for a host. And so the fitness we measured is lab fitness, but still it's infective viral particles, not just viral particles. So there are some important nuances there, but fitness, defining fitness is very important.
Excellent, thank you. So folks, we have our question and answer queue there at the bottom of your webinar window. So you can hit on the Q&A button, pose your question. If you'd like me to keep it anonymous, I'm happy to do so. So send those questions in. One thing I would like to ask each of you though, and what we can start, last time we started with Arpita, let's this time start with Oded and then go Oded, Arpita, and Melissa, is to offer a bit of a crystal ball perspective on your area of work. So where do you see your efforts as well as your colleagues headed in the next 5 years or so? And so Oded, you want to kick us off with that?
Yep. So we are looking for new rhodopsins, and then we actually are slowing moving from bacteria and archaea into picoeukarya, because we do find that beside the regular proton pump rhodopsins, they have very exciting new rhodopsins. So enzyme rhodopsins, for example, were found in microbes that are actually eukarya. And then the varieties are enormous. So we're switching slowly. It's harder to do metagenomics on picoeukarya because the genomes are bigger. You have introns, exons. So it's not easy, but we are there. So this is one switch. And I didn't mention the optogenetics field, which is actually now very popular, and they're looking for new rhodopsins all the time. So we are also moving to rhodopsins that would absorb in red and infrared, and there are a couple of those new rhodopsins, because if you would have red and infrared absorbing rhodopsins, then all experiments with optogenetics would be easier because you don't need, to stick the electrodes with the light on those poor animals, if I may say. So I would say those are the directions.
Very cool. Arpita. Oh, sorry. No, we said, yeah, let's do Arpita, then Melissa. Sorry, let's do that. Confused myself.
So I am very excited about extracellular electron uptake. As I was mentioning at the end of the talk, I was surprised how far we have come in just 6 years and how the community finds these organisms everywhere. But being trained as a geneticist, a physiologist, I think that understanding why this is so common and what the underlying mechanisms are is what our lab is focusing on. And we are trying to expand how we go out what kinds of environments are we thinking about. So we are looking at wetlands, but that's not the only environment we can look at. So we are looking at the human body, and we're also thinking about how this process might be important for the generation of the first eukaryotes. So we can co-culture archaea with these organisms that we isolate from Ellis Island and Woods Hole, and they grow beautifully as co-cultures. So and they seem to be exchanging electrons. So, that would be interesting in the next 5 years to see as well.
Very cool. Thank you. And Melissa, what do you think?
Yeah, so I think I made a pitch for this at the last few seconds of the talk, that I think, and this is, future vision is coming from what we struggled with most in interpreting our data, and that was the relative nature of most of the data we collected. So there's been great work and actually, frankly, some of it not new. So I would say some of the first kind of work with quantitative metatranscriptomics spike-ins was back in 2013 and 14 by Mary and Moran's group. But it feels like that notion is coming back now in full force. And I think that's what's going to be necessary to kind of take our observations of pathway changes and think of them more in terms of fluxes. And also combining it then with things like stable isotopes. And I can give one pitch for that I thought of in response to the photosynthesis question. This was work from Jake Waldbauer and Maureen Coleman that through the use of kind of quantitative proteomics and stable isotopes were able to show that in the case of a cyanophage, so infecting a cyanobacterium, the viruses acquired over 41% of their nitrogen from extracellular sources. And so that's when you start to think about what are the ecosystem level impacts, and you can put numbers on that, not just hypotheticals. And so that's a case of one virus and one host. We know that interaction can result in different phenotypes, so different impacts. So now we need to expand that to different virus host pairs, and then hopefully make observations about the universality of some of the things we're seeing, and being able to link the universal properties of traits of infection with data we can collect in high throughput in the field, like possibly virus and host genomes sequences. So getting more quantitative, linking it together and doing it high throughput, making inferences in high throughput.
All right, that is amazing, right? I mean, in terms of actually what all three of you shared in terms of moving forward and being more quantitative, right, and establishing these relationships that that cross-cut the systems we study, from a genome to a transcriptome and a proteome, metabolites and so on, is where I hope we are all headed in microbiology. We have let's see, a question here, which I will share with you, then I'll turn it over to Hannah, who I know has a question she'd like to ask. Someone asked, they recently realized that they hadn't thought much about the bioenergetic differences between bacteria and viruses since high school biology. Are viruses, in fact, defined as alive these days? Like today, do we define viruses as alive? And how do you define life in terms of bioenergetics? And where do viruses fit into this definition?
I'm not going to answer this, but I also just feel the need to thank this person, this anonymous person, for this question, because I don't think it's silly at all. I can tell you that Michael Gilmore and I are working together to teach a graduate level class, a graduate level seminar microbiology class. And we recently spent the better part of a 2-hour class talking about this question, how we conceive of viruses. So I just feel the need to say that and say thanks for an excellent question. It's not silly at all.
Who wants to take that? What are your thoughts on viruses?
I really wanted to punt it to Arpita. I know I talked about viruses, but she did begin her talk with a slide of like, what is life? And I'm really curious, as you were presenting that, I was wondering what your answer to this question might be. Hopefully you don't mind.
I think it's philosophical, right? I feel like it gets into this gray area of what we think is alive versus what isn't. I think that I didn't think about this when I was defining life in my view, but viruses are alive. That's my vote. Go viruses. They're totally alive. I think they're just sort of this gray zone of they have to be in a host to be able to replicate, but viral particles are alive because they can infect, right? I mean, what do you think? What do you think about it, Melissa? Like I always get into this philosophical thing with freshmen, like Hannah was bringing up. They always ask very thoughtful questions like that, and to me, it seems like it'd be unfair to not account for all these viruses and say that they're dead. So I go for alive. As far as bioenergetically goes, they have to be probably inside a host, but many animal viruses have membranes. What if viral membranes had bioenergetic capabilities? I mean, why wouldn't that be possible? Like I know that they have only receptors that allow them to attach to host cells. People don't think about them as energy, but then what if there were viruses that had rhodopsins in their membranes? I don't know. Couldn't they make energy? Just a thought.
I don't want to go. I don't want to go too deep into it, but we are now working on something like this. So, rhodopsins in the membranes, viruses, but not for energy. It's more, to infect the host. But I would say we can easily escape from those questions because we had the term viral cells, and this, I think, why. This was coined, viral cells, because inside the viral cells, those are alive. So, I mean, hey, so I would say alive, but with the concept of viral cell.
Yeah, I think I can expand on this. These nuanced responses. So thinking about them, yes, they can infect, but that infection process, like mechanistically, it relies on, I guess, the potential energy stored in proteins that is then released in certain protein-protein interactions. Without that, without that kind of release of energy, they will stay as these stored inert particles. That's one thing that complicates my idea of them being alive, because they can infect. The other is this notion of self-replication. So they may be able to store energy, but they can't use it for self-replication. They rely on their host. But again, then the action's in the virocell. So then if we start thinking, okay, virocell's alive, and thinking about, okay, yes, the bioenergetics fits that concept of alive, but what about self-replication? What is self-replicating at that point? If you check out the different, like the multitude of viral strategies in the review that I shared there by Adrian Correa and all, you'll see that some viral strategies obligately end in the virocell or cell death, but it's the virus that goes on. So I guess that would still fit. It's using energy and it's self-replicating. Yeah, totally thinking out loud and on the spot.
Yeah, definitely. I think I heard at one point the concept of a viral particle compared to a seed, in the sense that it has the potential to become a a replicating entity, but that the seed itself doesn't replicate other seeds. And so I think that's kind of an interesting, I think about that conceptually a lot too.
I realized I actually had that analogy in my notes, my slide notes I've been making. So I'm very glad that you said that. That's, I think, a useful one in this context.
Thanks, folks. We have one question here, and then I do want to turn it over to Hannah, because I know she has a question in mind. Amanda Fultonhauer asks for Arpita, could the electron transport work your lab is doing contribute to biobattery development?
Sorry if my daughter interrupts with her answer to this question. Yeah, we are. Well, I didn't show any of the sustainable bioproduction work that we are doing. And we are developing different ways of capturing. I'm so sorry. But we are definitely working on things like that. Sorry.
Speaking of lots of energy.
I was just really appreciating what's going on in your shirt. I feel like that's a very sciencey shirt.
That's amazing.
Dinosaurs.
Absolutely.
Cool. Yeah, well, I can come in with a question here that I think also relates to themes we've been thinking about in this class with Mike. But I think that one of the challenges facing maybe any microbiologist today is balancing the desire and need for mechanistic experiments, which relating maybe gene to gene function to phenotype, and the inherent reductionist nature of those, with the incredible power of sequencing for genomics and metagenomics and breadth of data, and the acknowledgement that microbes live in complex environments and communities of other organisms. So I realize that that's a broad question, but whatever comes to mind for each of you in terms of how you in your own work balance those sometimes competing desires to think about microbes in their very complex communities, but also to understand specifics about their molecules or about their genes and gene functions.
Okay, I can start, but yeah, it's a big question. I mean, usually when you do the bioinformatics, then you need to decide whether to focus on individual or to focus on the entire community. It's an open question. I mean, I don't have an answer. I think it also depends on your resources. I mean, how many good bioinformaticians you have. I would say this is the bottleneck nowadays, because we have many data sets waiting for us just to look at. And it depends on how you look at. I mean, it's easier to work on individuals. It's much more complex to work on the interaction of those with others and the environment. So, I mean, we do both, but it's always a question on where to start.
I can go next if no one else is. I had to go give my daughter an ice pop, sorry. So I think that, I struggle with exactly that. And I think that was demonstrated in my talk as well, starting with a background in just isolated organisms, still isolating them, but always trying to think about how they operate in the environment. And I think that the way that I have been managing that is always knowing that what I'm doing is artificial in the lab, appreciating that, always thinking like a microbe. Abigail Saliers was on my committee when I was in grad school in Urbana-Champaign. And she was always telling me that think like a microbe, put your microbial hat on. And I think that that's been a huge help, just thinking about that always. And also, all these omics approaches that Melissa is bringing up, and how integrating them with that same microbial hat on has been also helping me. I'm learning how to, know that there are limitations of both these approaches, a completely systems approach and a completely isolated approach. And marrying them would require crosstalk and lots of open conversations about the limitations of both. So I think that's how I've been growing my own mind.
Mm-hmm. That makes a
Yeah.
lot of sense. Thanks.
Can expand on the notions of both of those, and that I think we need them all to understand our systems. I think that's the reason why they are all parts of this field, is because there's a need for them. And like Oded mentioned, in addition to like skills and bioinformaticians, it's like time. It's our lives, like the time of our lives. How do we choose to spend that? And I always bring that back to what brings you the most joy. And that's different for different people. So it's probably not this is better than this realm to focus your energy in and your vital life source in and your brain power, but follow the things that are fun for you, because there are so many other folks out there working on all those other parts. So find who they are. If you're not going to do that yourself, figure out how to work together, and then continue to do the parts of the puzzle that bring you the most joy and that you can fund, I guess.
Yeah.
Well, I want to thank all of you for those answers, and picking up, if you don't mind, sharing a perspective as well on picking up on where Melissa just took us is, I often hear students ask, how do I, what do I do to increase my chances for success? Do I pick this university over that university? Even graduate students who are applying to work with me saying, oh, well, if I work with you, I get to work at this institution and with your lab. But really, my passion is in this other thing that's a bit outside of your lab, but it's, a smaller liberal arts college or something. And, to echo what Melissa said, I think it's important to recognize that if you do what you're passionate about, right? And that is where you're going to shine. And in many ways, I think our students often assume that studying processes at one scale, for example, they may think that an omic approach is inherently more valuable than an ecosystems approach, or they may say working with a lab, a cultivated microbe is so much better than working with an uncultivated bug because you can do so much more. But the three of you have emphasized that we have to look at our world from these different vantage points, right? And so pick the one that you're going to be most passionate about, because that's where you can make your greatest contributions.
I really feel like each of your works too emphasize the way that these can complement each other and inform each other. And so that feels really like a valuable takeaway for me from each of your talks.
Right. And if I didn't know better, I almost feel like the three of you conspired in presenting such a lovely spectrum of amazing work that cross-cut scales and even different kind of habitats, right? From the photosynthetic world to the chemosynthetic world, from cyanobacteria and pico-eukaryotes to viruses. What a heck of a way to open our symposium. So thank you all. And of course, our scientist artists, who are amazing scientists in their own right, and then do this extraordinary thing that I find so soul nourishing. Do you, any of you three have anything you'd like to share before we move into our next part of our symposium? Any closing remarks? Let's start with Melissa this time and then work backwards. Melissa, Oded, then Arpita.
I was already sitting back and like just reflecting and appreciating, thinking I was done with providing.
No.
So, closing remarks. Nothing, I guess, super profound, but I'm really excited to see the rest of the artwork. And I really appreciate just the format of how you've integrated all these different passions and shining of various talents. So,
art is profound.
People are awesome.
Thank you, Melissa. What about you, Oded? Any parting thoughts?
Mine is very simple. I love doing science. I mean, I'm enjoying, almost every minute, and it's fun. It's fun. It's a hard work because usually you don't get, the experiments do not work, but every now and then that it works, it's wow. So I really love it.
Well said, Oded. Arpita.
My highlight was seeing your home office, Pete.
Oh my.
What's behind your, is that a Millennium Falcon? Or,
There is, that's true. There's a Millennium Falcon here. This is the Apollo, this is the Saturn V rocket that was used for the Apollo moon missions. Behind my head down here is the Wright brothers flyer. And then, of course, there's an Enterprise from Star Trek, and that is the Bell X-1. It was the first plane to break the sound barrier. So it's my little shrine to the amazing ingenuities in aerospace that shows what we can do when we put our differences aside and try and do something extraordinary. So
I have something to tell about Peter. And this is 20 years ago. He used to take my kids and fly rockets.
Oh, that's right.
Remember?
I do now.
How are the rockets powered?
Motors?
I would let Peter answer.
No, they're just the little model rockets that you buy the rocket motors for. And we Oded and I lived in Marina, California, in an area where there were these open fields, and I really enjoyed taking your kids out and showing them the model rocketry. So we apparently need to do this again, I think. I mean, come on. So, well, this was a wonderful morning, everyone. Thank you. And I am going to now introduce our friend and colleague who is going to be running our lunchtime science trivia. Now, before I introduce Dr. Sarah McAnulty, for the next about hour, we're going to be doing trivia. And I would really encourage you to hang out with us, go get your lunch, bring your kids, involve, drag your neighbors into it. This is fun and lighthearted, and it's a great way to just sit around and kick back on a Saturday. Now, Dr. Sarah McAnulty is actually a squid biologist who earned her PhD in 2019 working with Dr. Spencer Nyholm at the University of Connecticut. She's a prolific science communicator with an active presence on Twitter, Instagram, YouTube, and TikTok. Sarah founded Skype a Scientist. It's a nonprofit organization focused on connecting scientists around the world with everyone else via personal connections. Since 2017, her programs have connected over 37,000 groups, not people, but groups with scientists, and all of their kid-facing programming is offered for free. You heard that right. Dr. McAnulty's Skype a Scientist programs for kids are free of charge. Now, Skype a Scientist also runs weekly trivia events for adults every Thursday, and on occasion, Sarah and her colleagues do some other activities like do some more adult sort of oriented evening sessions where we talk science. And in fact, I had the privilege of talking to Sarah and her colleagues at an event from a ship one time while I was out at sea. I have to tease Sarah a little bit because I lent her a giant isopod in a glass jar and made her promise to just take good care of it. And then minutes before the event, Sarah said, oh, well, it got dropped on the sidewalk and the glass jar broke and there's ethanol everywhere, but the isopod is fine. So I got to tease her in front of a bunch of folks, reminding her that she had just one job that night. But Sarah is an extraordinary communicator and brings her passion for science and cephalopods to all of us. You can learn more about Skype a Scientist and sign your family up, or learn about their evening trivia for grown-ups at skypeascientist.com. So let me turn it over to Sarah, and she'll give you a sense of how the next hour is going to run. So Sarah, it's your show.
Awesome. Thank you so much. Thanks for the introduction. Hello, everyone. I'm Sarah, and I'm going to be running our trivia event for the next hour or so. There are going to be 3 rounds of trivia today, all microbe themed. So the first round will be 6 questions. The second round will be a visual round where I will be sending out a link to a bunch of pictures, and you'll tell me what caused what you're looking at. Is it a virus, a bacteria, an archaea, or a fungus? And then we'll do another round of 6 questions. There aren't all that many rules. It's pretty straightforward. After each round, I'll announce the answers. Actually, after each question, I'll announce the answers. You grade yourselves. And then after each round, I will be sending you a Google form. You put in your team name. That can be your name, your family name, or any wacky name that you come up with. And then I'll announce the standings after at halftime, which is the visual round, and then again at the end. For the winning teams today, we have a couple prizes that you can choose between. We have some notebooks that are very science themed. I am admittedly a little partial to the ocean. So we've got some cephalopods on this notebook. It's a lined notebook. And then this one is a collection of invertebrates. We've got some brittle stars, some coral, some crabs. Of course, got to put a cephalopod in there, the nautilus. And then we have some fossils. So you get the choice between one of the notebooks and one of the fossils. And so we have some old ammonites. So that's one type of fossil you have to choose between. Ammonites are ancient cephalopods. They're the animals that came before the squid, the nautilus, the octopus, and the cuttlefish. And so we've got some that in the process of fossilization have gotten what's called opalized. And that means that basically shiny minerals got in there. I admittedly am not a geologist, but love a good fossil. And so these are pretty cool, pretty shiny. The light doesn't always pick up on them, but with your eyeballs, it looks pretty cool. So these are what you have to choose between. Also, we have some, these are called belemnites. They are more squiddy looking as opposed to spirally shell looking. And this is the internalized shell that you would have found inside a belemnite. They looked real squiddy. If you look at a picture of a belemnite, looks like a squid, but the shell was inside the animal's body. So with that, let's do it. Let's get started. This is going, so we're gonna read out a question, and then Hannah is going to release a poll where you get to pick your answer. And then after that poll runs out, after 60 seconds, I will tell you the answer. I'll put up some pond scum for us all to look at. It's not really scum, it's all the organisms that are living inside pond water during that 60 seconds where you have to deliberate with your squad. Or the voices inside your head, as I do living with cats. And then we'll go from there. So I hope you're all gonna have a good time doing this. Feel free to comment in the chat with whatever you'd like. Let's get into it. So our first microbe round starts with some microbe history. The first question here is as follows. The first person to look at bacteria under a microscope was Antoine van Leeuwenhoek. What did he name the microbes? Did he call them A, Kleinenkrittern, B, animalcules, B, pill beings, or D, sphericals? All right. Can you see the pond scum? Can I get a, okay. right, the minute is up. The correct answer, which 90% of you got correct, is animalcules. That is the adorable name that Luna
Nice.
came up with. Yeah, way to go, everybody. Doing well. All right. Hopefully, I'll be able to stump you all at some point here. Here's number 2. Mitochondria are famously the powerhouse of the cell. A long time ago, mitochondria were bacteria that squeezed inside of a bigger cell. Over time, they became part of the cell in a process called endosymbiosis. What other organelle was originally an independent organism? Was it A, the lysosome, B, the chloroplast, C, the ribosome, or D, the Golgi apparatus?
Hello. My big talk. This is like a trivia. You want to listen to it with me? If they like ask questions and you can learn things.
What did you say?
Want to do this next part with me?
Perfect. All right, that's been a minute. Let's go through the answer here. All right, the correct answer is chloroplasts. So chloroplasts used to be external photosynthesizing little critters, and now they're not just an organelle. They are one of the greatest organelles out there, the chloroplast. All right, number 3. MRSA or MRSA is a medicine-resistant strain of bacteria that can be very dangerous for people. The M in MRSA stands for the drug MRSA is resistant to. What drug is that? Is it A, methicillin, B, metronidazole, C, azithromycin, or D, moxicillin?
There's some algae.
I kind of feel like I'm in church with this one. Just music.
Let's move around and see if we can find something else. A lot of algae. Let's back out. Ooh, focus.
This is making me want to go look at some pond scum under my own microscope.
Same.
Here's a dude.
All right. The correct answer, as many of you got, is methicillin. The others were partially made up by me. I took other antibiotics and just slapped an M at the beginning. All right, number 4. Which of these are bacteria not used for? So which of these applications did I just make up? Is it A, bacterial batteries, B, cleaning up toxic waste, C, degrading ocean plastic, or D, moisturizing skin?
What are you? You're more fun. No? Here's something.
All right. Many of you got this one correct too. Moisturizing skin. It's amazing what people use bacteria for. The batteries, I think, is the wildest one I've heard of yet, at least the one that blew my mind the most. I believe that's Geobacter that has that electrical ability. Cleaning up toxic waste is another very useful thing that they do. Degrading ocean plastic is something they basically just eat the plastic. I don't know if they could do it at the scale we'd need them to do it, given the amount of plastic in the oceans, but you know, every little bit helps. And to be fair, I wouldn't be surprised if over time we find a skin moisturizing bacterium, but I haven't found any commercial use of that yet. All right, and let's move on to number 5. That is, which of these foods is not made with the help of bacteria? Is it A, yogurt, B, kimchi, C, ice cream, or D, sauerkraut?
So hard to. Ooh, spiky. Next. Oh, those guys are so quick. Stop it.
All right. All right, so the correct answer is ice cream. Fun fact about ice cream that I recently learned, there are scientists that are studying these antifreeze proteins. And so antifreeze proteins live in all sorts, not live in, but are produced by all sorts of animals, including Arctic fish, a lot of different kinds of fish that live in cold environments. And scientists are looking to see how we can learn about those little proteins and then apply them to different foods. So I don't know if you've ever had ice cream that like maybe you put it in the freezer and then you left the door of the freezer open for a little bit and then it thawed and then it froze again. And then you get these like crunchy ice cream, which is, nobody wants crunchy ice cream. That's unpleasant, except for obviously Oreo bits, which are very important. But having the texture of the ice cream be crunchy is not great. But having the inclusion of antifreeze proteins in the ice cream actually creates this like smoother, nicer texture. And so scientists are trying to figure out how we can learn from Arctic fish to make our ice cream better, which is pretty wild.
I love that. I'm very in favor.
Yeah, so anything that makes my ice cream better, like ice cream is already about as good as it can be. So better ice cream. I mean, thank you, science. Thank you, science. All right, number 6. In the 19th century, beauty marks were very in style. What were they used to disguise? Is it A, chicken pox scars, B, acne scars, C, smallpox scars, or D, birdshot wounds? A lot of hunting happening. All right. What else? What other critters are we going to see here?
Hey.
Sarah, while folks are answering this, do you happen to know whose voice we're hearing?
Who this guy is? No, I don't know who this guy is. I'd love to know. His commentary is something else. The mix of the like disembodied man with the church music is quite the combo. I admit. Protists are so weird.
So many levels of. Ooh, cool.
I think he's German.
Oh, take that one. Where did he go? There you are.
All right. All right, smallpox scars. So this is something that I learned from Pete, that the beauty marks were just like things people put on their smallpox scars to hide them, which is sad but pretty cool. Innovative fashion for sure.
But in fairness, Sarah, I mean, it could go to birdshot wounds, I suppose, or acne.
I guess it's possible. That's perhaps not why they were invented, but who's to stop the innovation of fashion from covering your birdshot wounds?
That's right. That's right.
I don't know how often women were going on bird hunts, but who knows?
Who knows?
Who knows? All right. So now I'm going to send out the scores link. So you can submit your scores for the first half in this. I'm going to stop. There we go. Chat. There we go.
Very cool. So now folks have a chance looking in the chat box there to submit their scores, right, Sarah?
Yes.
Outstanding. And while we're waiting for folks to do that, you know, I was thinking about that microbial products in cosmetics, and I know there are some in cosmetics, but skin moisturizer specifically, I have no idea. So I was sharing with our participants that, you know, Sarah knows a lot. She rocks at trivia, but if they knew something we didn't, to go ahead and share it.
Absolutely.
Right. And Irmgard chimed in that said that there are topical probiotics used for acne and use common oral probiotics. Interesting.
Yeah, I think this is where I confess to you all about the time, you and the very many people watching this, about the time that I tried using a commercially available product that shall remain nameless because I'm not, you know, sponsoring them or anything, in lieu of deodorant. So there's a product that employs soil microbes that are good at breaking down the aromatic compounds that make up the smelly component of our sweat. And in theory, you can spray it on any part of your body, but like particularly the sweaty parts, to in substitution for like a stick deodorant. I tried it for a summer. I got some feedback that it wasn't particularly effective. So I am not currently using it anymore, but that's the closest I've come. No, I don't know of a moisturizer.
I was at a beneficial bacteria conference one time, and there was this guy walking around, interestingly, approaching only the young female graduate students, saying, I have something that will make you more beautiful using bacteria. And we didn't get a lot of the details as to like how that all worked, but it was like something that you spritz on your face that apparently used bacteria, but I didn't get the deep. And I'm not sure how moisturizing that was, it was a little dubious, but he was going for it. It's possible that it works. You know, yeah. Bacteria do amazing things. Who's to say? Bacterial squalene is a thing, using some moisturizer for skin. Interesting.
Yeah, that's so cool. And Claudette was saying that she's questioning the smallpox. I'm very intrigued. My comment about that is sharing that with you, Sarah, comes from reading about beauty marks in Victorian London in the middle of the 1800s. So I'm kind of curious to hear what Claudette can tell us more about that. Bacterial squalene, though. Didn't know that was a thing.
Me either.
Very cool. Thanks, Una.
Yeah, thank you. All right, we've gotten 8 teams in so far. A lot of you acing it.
Uh-oh.
Not everybody, but but a lot of folks.
That's great.
A brainy bunch for sure. Awesome. All right, so in the meantime, we'll give you until the end of the visual round in order to get those scores in. So now I'm going to send you to the visual round. Here we go. The link is going to be in the chat right now. It's going to send you to basically a PowerPoint slide, and you can record your answers wherever. Here we go. Visual round. There you go. Let me know if anybody has trouble accessing that. It should be good. You are going to have about 7 minutes to complete this, during which time I will find a different microbe video on YouTube that doesn't have a delightful German man speaking to us the whole time. And we'll go from there. If anybody has any questions, we're all ears.
Let's see. So just to clarify, Sarah, I'm seeing 8 pictures here, and I'm going to record for myself whether each one was caused by bacteria, archaea, fungi, or viruses. This time I don't have a poll for all of you, so you will need to
scribble it down, text it to yourself, what have you, and then after 7 minutes, we'll go through all the answers.
Okay, awesome.
Wonderful. All right. Do we have any microbes that people would like to see? Let's do Stentor for a start. It's one of my personal faves. But this one's only a minute long.
Put your head on my.
All right, here we go. I might put up my own personal tunes here. I have never found a tardigrade in the wild, although I hear it's not particularly challenging.
I haven't yet myself either, although I do know a person who has done it.
My lab mate in grad school did as well. Yeah. Ugh, Stentor are so cool and so weird.
I don't think I know what that is, a Stentor.
Oh yeah, that's crazy.
Yeah. Truly, truly strange. It just looks like a trumpet, like a weird little cellular trumpet. I like them a lot.
Oh yeah, now we're talking. The tunes, this is the way to go.
We're doing some calming French cafe tunes today.
Right. I'm suddenly feeling the need for an espresso.
I'll put the volume down here, up here. There we go. There's the use of that animal fuel.
With a smile on my now you're alone. You're so sad on your own. The truth is, we ran out of time. Now you're looking for me.
Weird. I've been, this is just a side note, I've been collecting Uuthika, which are like the eggs of mantises for the last couple months out in the woods, specifically the invasive mantis, it's called the Chinese mantis. I'll go get one and show you. They look like this. Almost looks like spun sugar.
Oh, yeah.
This is like the egg of the mantis. They look like little, like, bug lollipops. You'll see them on all sorts of different plants. And my first one hatched this morning, overnight.
Whoa, that's a big deal.
It is a big deal. Can you see the little?
Oh, I can.
I'll see if I can pull one out. I'm gonna have like thousands of mantises in mere days.
When you say the first one hatched, that whole, how many mantises does that one sack yield?
What a good question. I think something in the ballpark of like 200.
You are gonna have a lot of mantises.
I have like 50 Uthika, because you're not supposed to let them just like exist in the woods because they're invasive. I don't know what I'm gonna do with all these mantises. I'm supposed to kill them, but I'm not good at killing animals. And they're just so goofy. They're just, let's see.
Oh yeah.
They're just the littlest silly babies.
Oh, I have a friend that I need to put you in touch with. I'll hook you two up. She also raises mantises.
I have, so I practiced first because I had no faith in my ability to raise bugs. I've raised squid. I can do squid in my sleep at this point, but mantises I figured would be easier than a squid because squid are notoriously a pain in the butt. But I hadn't done it. So I practiced with these mantises called ghost mantises. They're spooky looking, and I have them right here because I bought them from a person on the internet.
Les étoiles, les étoiles, dites-moi étoiles, pourquoi je vous regarde.
These have been with me now for about 2 months.
Whoa.
They're so cool. And they're very easy to take care of. All I have to do is feed them fruit flies. I get flightless fruit flies from just like Petco or whatever.
That's awesome. Whoa.
They're so cool. And all I have to do is spritz their tank down with a little water every day, just with like a little spritz bottle. And our 7 minutes are up. Yeah, and then feed them fruit flies, and they totally take care of themselves.
Wow.
So a fun starter pet. So I mean, like, I want to show you the like profile of them because they're truly so cool. Like,
yeah,
their little faces are just so spooky and cool.
They're very majestic.
They're majestic, and they often like. So in the wild, you know, they're pretending to be leaves, basically. So they'll pretend to like sway with the wind just by like going like this. And like
they're fluttering
in their tank. It's just like, you know, but they're trying to be sneaky. It's very cute. If all goes to plan, I'll have a male and a female. I'll get to breed them.
Whoa.
But we don't know yet because they're not quite fully grown yet.
Are you going to be able to tell once they are full grown?
Yeah, the females have bigger abdomens.
Okay.
I always have to sing in my head the head thorax abdomen song.
You know, so Chinese mantises are native to China, Japan, the Korean Peninsula, Micronesia, and Thailand.
Uh-huh.
I wonder what customs would think if you tried to send back 100,000 Chinese mantises.
Are your mantises back? Please take good care of them. Yeah. I just don't know. That would be a tough, like, conservation project to fund. Hey, we'd like
to a mantis reserve.
The mantis reserve, exactly.
That'd be a heavy lift.
Pretty heavy lift. But here we are. Okay, let's go through the answers. Am I still screen sharing? No, excellent. Okay, So here we go. The first was a salt pond, and the halo archaea that live in those are archaea. The next question, let me pull up our sheet here. Oh, it's a tulip I took a picture of just the other day. That tulip is infected with a virus that doesn't really hurt them. But whenever you see in many different kinds of plants, instead of having like one color on the leaf or the petal, if you see two, that's from a virus. And that's going to be true for number 2 and for number 5. Both of those, if not the same virus, is like in the similar kind of virus. Number 3 is something called a stromatolite. Stromatolites are so cool. They're bacterial boulders that are created by bacteria. These are some of the oldest forms of life that we're aware of and are super, super important for studying ancient life. It's like a little fossil record of ancient bacterial life. Number 4 is smallpox, unfortunately. Glad that's not something that we have to put beauty marks on ourselves for anymore. 5 is a virus again. Of course, smallpox, so number 4 was a virus. Number 6 is a root nodule. Root nodules are little specialized housing containers for a type of bacteria called rhizobia. There's a bunch of different types for different plants, and they fix nitrogen. So they help the plant get the nutrients that it needs from the soil that it's unable to suck out of the soil and air by itself. It just doesn't have the ability to process the type of nitrogen that the bacteria can use and then turn into a usable form by the plant. Number 7 is a fungus that is cordyceps, a type of fungus that infects ants and then gets into their bodies and then sprouts. It alters their brain chemistry such that the ant wants to crawl up a plant quite high. And then once it gets up there, it does what's called a death grip. So they bite really hard onto the plant that they're on. They kind of root them there. And then the fungus sprouts out of their head and sends spores to the forest floor to hopefully for the fungus infect more ants. Pretty brutal. And lastly, number 8 is also a fungus that is called cheatgrass, is the host plant, the plant that's being infected. And that is a type of fungus that infects cheatgrass. Cheatgrass is something that it's another invasive species in some places. And so scientists were trying to use this fungus as a way of controlling the cheatgrass. So there you have it. Those are the answers. So to go through that one more time, the answers in order were number 1, archaea. Number 2, virus. Number 3, bacteria. Number 4, virus. Number 5, also virus. Number 6, bacteria. Number 7, fungus. Number 8, fungus. I posted those answers in order in the chat so you have them for reference. And now let's do the scores as they stand. Here we go. All right, the team who has learned the most this afternoon with 3 points from round 1 is JJX. The team with 4 points, Melanin and Tan. With 5 points, Penny Powell and Team B. And tied for first place after round 1 with 6 points is Alexis, Izzy and Mom, Unabella, Team Alborlins, and West Point. So thank you all for participating so far. Let's get into our 3rd round of trivia. Again, of course, with the theme all about microbes. Alrighty, here we go. Number 1, Jon Snow is more than just a hunky character in Game of Thrones. He was a scientist during a cholera outbreak in 1854. Where did he trace the cholera outbreak to? Was it A, a bakery, B, a well, C, the river, or D, the butcher?
Or Winterfell. Don't forget Winterfell.
Of course, Winterfell and the wall. Beyond the wall, on Tormund, perhaps. Who knows? All right. Now YouTube is automatically playing Ooh, a time lapse of a salamander developing inside of an egg. That's cool. Let's do it. Let's do it. I will do this in the next question because it looks like most people have gotten their answers in. One of my many anxieties and fears is that all of these mantises are going to break out of their enclosure, and I'm going to come into my office one day and I'm just like, the walls are going to be covered in little baby mantises.
Right. And then someone somewhere will come back and say, Sarah, these are invasive and we have to talk.
Yeah, then I'm going to be jailed in invasive species court. I guess better that my cats eat them than they live in the wild of the Pine Barrens where I caught them. But not sure about that. All right, the answer for number one, as 81% of you knew, was a well. So yeah, everyone was just feeding themselves cholera every day. Very sad. Okay, number two. Big blobs of bacteria are thought to be one of the, we already asked about this. Well, you, if you're paying attention, you're really gonna get this question right. Are thought to be one of the earliest life forms. They're basically boulders made by cyanobacteria. What is another name for them? Is it A, cementamites, B, bacterial boulders, C, stromatolites, or D, microbe mops? Luckily, I only said that word once while going through the answers. All right, let's open up this salamander video. Development is such a strange thing to watch.
It still creeps me out that you can actually study something that you can see with your own eye. It still unnerves me.
It's weird. It's weird when, I mean, it's weird the whole time, but it's weird when folks die like one of those first 4 cells, and then you can like watch everything that came from that cell.
Right.
That's just wacky.
That's crazy. It's
Can't
crazy.
believe that works.
Crazy talk.
Crazy talk. All right. So the answer here is stromatolites. Stromatolites are the name for those big blobs of bacteria that turned into stone over many years. All righty. Now, development. Number 3, listeria is a ridiculously cool type of bacteria. It's one of my personal faves. They have the ability to steal parts of the cells that they infect and then use those parts to zoom around the cell. What are those tails that they use to zoom with made of? Is it A, actin, B, cytoplasm, C, mitochondria, or D, fat? I've been spending a lot of time collecting moth cocoons to prepare for the bug zoo that we're doing in Philadelphia this summer. And I've learned that it is really hard to be a moth because they're born, they run around eating all summer long, and then they turn themselves into a cocoon effectively. That's like these particular moths that I'm working with are silk moths. Their cocoons are about yay big. I am sure I have one lying around that I can show you.
Wait, that seems like a giant cocoon. Was that like the size of
It's huge.
Like a Vienna sausage or like a hot dog?
A short sausage, I guess. Here, I'll show you.
Oh, like a breakfast sausage. It's lunchtime, if you can't tell.
Like a duck's egg. They're huge.
What?
It's bananas. Okay. So, but not banana size. Okay, so here's the size of one. So this is my face. This is the cocoon.
Whoa.
Like large, very large. These are the large silk moths. I was trying to really impress the children of Philadelphia this summer. And these moths will hatch. So within this cocoon, there's like a mini cocoon, it's like multi. So these have to survive the winter all the way up through like, I know they live up in Massachusetts. They might go as far as Maine. So within this like blanket, there's another blanket of silk. And then you. The pupa, which, can you see the fact that this legitimately looks like Mothra? Like, this is so wild and cool. You can see the antenna like imprinted on the pupil, like casing.
Yeah, that's crazy.
It's bananas. And then you can see the wings as well here, and then you can see like the butt over here. Okay, so it's a three blanket system. We've got this, and then the actual animal inside. And so my goal this spring has been to find these and then make sure that they're still alive inside the cocoon, because wasps will parasitize these animals. And so while they're just helpless sitting there in their cocoons all winter, the wasps will burrow in, lay their eggs, and then the babies will eat up the just sitting there moth. And so I've collected 13 of these cocoons, which are quite hard to find, and they've all been dead, every single one. And it's just so sad. And so I've collected another species as well. I've collected about 7 of those, and one of those is alive. So I'm currently 1 for 27 for dead moths. These poor moths. I don't know how we have moths anymore, honestly. It's tough to be a moth. Okay, let's go back to bacteria here. Listeria, very, very, very cool. They will propel themselves around the cell by co-opting a bunch of different cell cytoskeletal proteins. And what they use is actin. Basically, the actin will like form on the back of them and push them forward through the cytoplasm. And if you watch, it just looks like they're just like zipping around. It is so, so, so cool. And there are also videos that have the actin like that will like light up in a color, so you can see the little comet tails. Oh, it's good microscopy. It's just good stuff. So feel free to Google that when you can't sleep tonight. If well, that might make you sleep worse because you'd be excited, but that's what I do when I can't sleep. All right, number 4, known as one of the most indestructible micro animals, what kind of tiny multi-celled organism can survive totally drying out, radiation, and even the vacuum of space? Is it A, the amoeba, B, the tardigrade, C, the mini mushroom, or D, the paramecium? Another type of moth that we find around here is called a bagworm moth, and they like to hang out. One of the particular species that I'm about to show you, they live on all sorts of pine trees. So this is called the evergreen bagworm moth, and this is its.
No, but it's a very slow thing. It's why it's a tardigrade. It's always behind.
And I think that they put them in space.
So these moths will put what looks like pine needles on their cocoons in order to look just like a pine cone. And I mean, they do. That looks like a pine cone, but it's a moth. And I'm very impressed with insects on the whole. All right, we're almost done here. All right. So question 4, the indestructible micro animals are called tardigrades. Tardigrades, they're also called water bears. They are very cute. They have these like little feet on either side of them. They're kind of chubby. Their faces are a little spooky, but they're very, very, very cool. Yeah, the tardigrade, tough little guys. All right, one of my favorite things that's come out of microbiology in the last decade-ish are poop transplants. Poop transplants are most commonly used to treat infections caused by what type of microbe? Is it A, the flu, B, COVID, C, Clostridium difficile, which is a type of bacteria, or D, tuberculosis? This week, I also saw, I was in the Pine Barrens looking for these darn moths. And I happened upon a snake eating another snake, and it was the wildest thing I've ever seen. It was a juvenile black racer. So the snake was only about that big, and it was eating another snake that was a red-bellied snake. So it's a black snake with little keel scales, which are like not like a smooth snake, like a kind of little rougher snake. And the belly of the snake is bright red. So they're black on the back and bright red on the belly. And the black and red snake was the one being eaten, but the snakes were the same size. And it was just a shocking thing to come upon. I have that racer snake was really.
How quickly was that progressing? I feel like that might have
not quick. I just sat there and watched the show. The snake had this look on its face, like the one that was eating the other snake. Like, what have I done? Like this, I've done it now.
Questionable decision.
Really questionable decisions here. All right. So poop transplants, just so powerful, so useful, but also inherently funny. Clostridium difficile is what they're used to treat often. There's also a lot of other things that people are working on. With C. diff is the shorter version, and also poop transplants, looking at what else we can help people with by using poop transplants. Just cool, very cool stuff. And our last question for the day, number 6. Puerto Rico has a bay full of bioluminescent organisms. At night, you can swim there and the water lights up. What organisms cause that bioluminescence? Is it A, fish, B, dolphins, C, fungus, or D, dinoflagellates? Our last question for the day. This office is really turning into a bit of a cabinet of wonders over here. Every time I look around, I'm like, oh, I can tell them about this. I don't know. Got a spider in here. That was not on purpose. There was a spider in one of the cocoon moths, and I'm afraid of spiders. And then the spider came out while I was doing trivia for another event. It scared the pants off me. But it's only an itsy bitsy little spider. So I'm going to take some very close-up pictures of this spider, because I found that if I take a blown-up picture of a spider, I'm less afraid of them. So I've been tackling my arachnophobia with macro photography of spiders.
That's impressive and counterintuitive.
Super counterintuitive. A lot of spiders have really cute faces, or at least big googly eyes, and that helps.
Yeah.
So, all right, the answer for Bio Bay, as it's sometimes called in Puerto Rico, it's full of dinoflagellates. And so they are triggered to glow basically when you whack them, when they get hit with something. And so if you put your goggles on, go out at night, turn off your light, and just swim through the water, I mean, it is wild. I did this, I've never been to Puerto Rico, but I did this in Belize one time, and it was super, super magical. There are other places in the world that also have this bioluminescent stuff living in the water, so you can do that too. But yeah, wow, it is. I highly recommend putting that on your bucket list. It's really, really, really cool. All right. So with that wraps up our trivia game. At this point, I'm going to send you a link to the scores sheet so that you can submit your scores. And then we find out who wins. Although frankly, if you've learned anything today, we've all won. Winning is relative. What was number 8 in the visual round? Asked Penny. That was cheatgrass being infected by, it sounds like dead man's fingers. It's something like that, but it's not dead man's fingers. Let me look that up for you. Black fingers of death is what the fungus it's called. It's a plant parasite, a fungal plant parasite infecting cheatgrass. And Tansu, I don't know the answer for the bioinformatics experience question. Let's see how we're doing. Get those scores in for the second half. We'll give you until 12:55. It's currently 12:53, and then we will go through. And if you think that you have really killed it today at trivia, start thinking about which fossil you want. The choices being this opalized ammonite, or the cut through the middle ammonite, so you can see all those chambers inside, or the belemnite, the ancient squid. And then you also have 3 options for the book that goes along with it. You have marine invertebrates, you've got cephalopods, and your third option, which I didn't mention before, but we have right here, is the Incredible Cephalopod coloring book, authored by yours truly. It has, so every letter of the alphabet is a different cephalopod. So we've got B is for the blanket octopus. We've got G is for giant squid. K, of course, is for our buddy the Kraken, which is basically a giant squid. I don't know if anybody's told you, but we think Kraken are the giant squid. Pajama squid. And every page has information about the species that you're looking at, and also has a pencil at the bottom so that you know actually how big is the squid we're looking at, because some squid are incredibly tiny. They're like this big, and some squid are bigger than a school bus. So, you know, it's important to know what we're working with. And then the end of the book also has like all of our squid in size comparisons and compared to a little human as well. And also comes with a glossary to learn about all the squid words that you may have learned. So those are your choices. All right, let's see how we're doing on the scores. The clock has struck 12:55. We've got 9 teams. Here we go. Calculating. All righty, here we go. The team who has learned the most today with 9 points is JJX. With 11 points, Penny Powell. With 14 points, Team B. With 19 points, tied for second place, we've got Claudette and Team Albrolins. It seems like a bit of a portmanteau we got there. And tied for first place with 20 points, which is really, really good, is Izzy and Mom, Unabella, and West Point. So congratulations, everybody who won. At this point, please, if you are one of those 3 teams, private message me on the chat. I am, what am I called? Skype a scientist or Sarah McAnulty?
Sarah McAnulty, yep.
Sarah McAnulty, there you go.
McAnulty.
So you can just type your email address just to me so that everybody doesn't see your email address, and I will send you an email. You can tell me which of the things that you'd like in your address, and we will send those out once you get back to me. Thank you all for joining us for this hour of science and trivia. I hope you all had as much fun as I did talking to Pete and all of you, and Hannah, of course. If you want to know more about Skype a Scientist, please go to skypeascientist.com. We have everything you need. If you have any questions, you can find me on Twitter or any social media platform at Sarah, MACK attack. I'll put that in the chat as well, along with the Skype a Scientist website, skypeascientist.com. I'm Sarah McAttack, and you can also email skypeascientist@gmail.com. I get back to emails pretty quick usually. We're always happy to help and connect people with science all over the place. So hope to hear from all of you.
Sarah, thank you so much. And the prizes that are just like, that's so cool. So I'm glad that you all who participated and hung around get a little bonus. And major thumbs up to Team Duhaime. Look at them. They're all on screen there. A whole bunch of folks that were really rocking it. As Hannah said, we'll hang out for a little bit. Right, Hannah? Are we taking a bit of a breather?
Yes, we will have just a 10-minute break here to transition, stretch, do whatever you need to do. And then Mike will come back with session 2, which will be emceed by Mike Gilmore. So I will put up just a screen, a hold screen here for those 10 minutes, and then we'll be back really shortly.
Outstanding. Thanks. And while Hannah's putting that up, I'll just say that, Sarah, I can't believe I've been pronouncing your name wrong this whole time. You haven't called me out on it.
Oh, no, sometimes you'd get it right, though.
That's even worse.
It changes it around. Well, you don't say it. Everybody says my name wrong.
Very well.
I would say 95% of the time, people butcher it, and that's fine because Irish phonetics make no sense. So,
okay, fair enough. Fair enough.
But folks, actually, you'll say it right, and that'll be great. So,
but Sarah, Dr. Sarah McAnulty is fantastic, and thank you so much for being here. And do check out Skype a Scientist, and again, stretch those legs, and we'll see you in a few minutes. Thanks, folks.
Hi, Shirong. How are you doing? Yep. Muted, I think. Ready to rock and roll? Okay, how do we get started here, Anna?
You can kick it off whenever you're ready. Just introducing the second session and then our first speaker in the lineup.
Yeah, I'm ready to go.
Great.
Okay, well, welcome to the second session on medical microbiology. Specifically, we'll now be talking about interactions between humans and microbes. I'd like to thank again Dr. Sarah for leading our trivia at lunch session. That was really terrific, and I learned a lot of new things, I'll have to say. Before I introduce the speakers for the next session, let me remind the audience that sometime in late September, maybe early October, we'll have another meeting of the Boston Area Antimicrobial Research Network, especially for those interested in human microbe interactions. Be watching for announcements about that. So a few things have garnered the attention and caught the imagination like the microbiome has. We're surrounded by microbes, and we exist only to the extent that we can hold those microbes at bay. Dr. Shirong Liu was the first to discover functional microRNAs in the mammalian gut lumen and feces. He's interested in host microbe interactions, particularly how the host evolves with the symbionts in and around it, how the symbionts were specifically selected for specific hosts, and what the role of that symbiosis is in autoimmune and neurodegenerative diseases. Following Dr. Liu today, we'll have Dr. Christine Faherty. She's enhancing our understanding of gastrointestinal bacterial pathogens, particularly Shigella, with the goal of identifying vaccine candidates that could improve the health of millions around the globe. And then because bacteria aren't the only things we have to deal with, Dr. Damian Krysan is a pediatrician who's interested in understanding how pathogenic fungi cause disease, how the host defends itself against these fungal pathogens, and how to develop new therapies. His work currently focuses on some of the most common fungal causes of human infection, including Candida albicans, which arguably is a host commensal, a human commensal, and Cryptococcus neoformans and Aspergillus fumigatus that arguably are environmental fungi capable of causing severe human disease. Interspersed between those talks, again, we'll have a fantastic opportunity to enjoy the art and hear more about the scientists, artists, including Dr. David Goodsell and Dr. Anna Maria Porras. And then again, like the first session, we'll finish with a 45-minute question and answer period. So if you don't get your questions answered between talks, we'll have plenty of time to discuss the material then. So it's a pleasure today to have Dr. Shirong Liu. And so Shirong, let me turn it over to you then.
Thank you, Mike, for your introduction. And hello, everybody. Thank you for joining us to discuss about the host microbe interaction. This is the, so just a second, let me share the screen. Hello, can everybody see my screen?
That looks good.
Okay. Hi, everybody. Hope you enjoy your lunch and have your bugs in your gut happy. So then I'm going to talk about the interaction between the host and the commensals in the gut, particularly how the microRNAs that we found in the gut involved in this interaction. So my talk will be separated to two parts. The first part, I'm going to, in general, talk about the interactions between the host and the microbe mediated by the microRNAs. And then the second part, I will talk about the specific microRNA. How does this microRNA involved in the specific disease, multiple sclerosis? So as you know, everybody know nowadays that the commensal play a really important role in the health maintenance and involved in disease pathogenesis. So I think it's very, very important to keep the commensals in shape. A very important question at least in my mind, is how these microbes, because the microbe and the host is really specifically really specific. The microbe is really specific for the host, and such relationship is so specific that I think that there should be a chosen selection mechanism that the host can select a specific microbe to the host. So my background is in immunology. So I think about this question, whether the microbes is selected by the innate immunity, adaptive immunity, or something else. So when I look at the literature, I found that actually, if you look at the microbiome data in the mediated knockout mice, this mice actually is mediated is toll-like receptor signaling adapter. So in this mice, basically they knock out the innate signaling, and you can see. Let me just put here the laser point. So here's what the purple ones are white type and red ones are mediated knockout mice, and you still find that they're clustered to each other. So that means they have a really cool core microbiome. It's not something like the loose core microbiome. And if you look at the red one knockout mice, these mice are deficient of adaptive immunity. They don't have a mature T cell and B cells. In these mice, you can also see the core microbiome in the same group. That means adaptive immunity is not really dominate the factor to shape the gut microbiome. I think here just an additional evidence from the literature. Chief is another adaptive molecules in the toll-like receptor signaling pathway. It involved in signaling through downstream toll-like receptor 3 and toll-like receptor 4. And in this double knockout, mediated and achieved double knockout, you still find a core microbiome. And this is just a repeat another additional evidence saying that in the adaptive immunity knockout mice, the microbiome is shifted, but as you can see, it's still clustered to each other. They didn't lose the core microbiome. So we find something interesting in the gut when we look at the gut. When I joined the lab of Dr. Holdweiner's lab, his group were working on the circulating microRNA. So my colleague was looking at the microRNAs in the blood. So I joined the lab to work on the microbiome. So I asked the one question: so if the microRNA can exist in the serum and plasma, why can it exist in the stool? So we look at the stool samples and found a lot of microRNA there. Here just show the top 10 and 15 microRNAs both in the mouse feces and the human feces. These microRNAs come from the gut epithelial cells, because if we generated the intestinal epithelial cell specific of dicer knockout mice. Dicer is an enzyme that is really important in generate the microRNA. If we knock out this dicer in the gut epithelial cells, you find that the microRNA in the feces are dramatically dropped. Those microRNAs also can come from the Hox positive cells. The Hox positive cells divide to the penile cells and the goblet cells in the gut. If you knock out the dicer in these cells, you can also find a geometric drop of the microRNA. But if you look at the microRNA in the red wine knockout mice, this means that the mice are deficient of mature T cell B cells, and you can find a few microRNA changed, but it's not universally dropped. That means most of these microRNAs come from the gut epithelial cells. So, what the relationship with between this microRNA and the gut microbes? We look at the microbiota profile in this gut epithelial cell dicer knockout mice. We found that in the wild type mice, you can really find a really nice clustering of the microbiome profile. But in knockout mice, they're just randomly distributed. They're not clustered. That means the loose core microbiome. If you look at the similarity of the microbiome, you can see in the knockout mice that just don't. Each mouse is different from the other. So in the phenotype, we want to have a look. Okay, so if the microbiome is changed upon knock out the microRNA in the gut, so what's the consequence? We use DSS induced colitis model. We induced colitis in this mice. It found that upon the gut epithelial cell microRNA deficient, and the disease is much more severe, as you can see in the blue one. Interestingly, if you isolate the microRNA from the feces and transfer to the knockout mice, then you can partially recover the microbiome to be more similar to a wild type mice. And when you induce colitis in these recipient mice, the recipient that received the fecal microRNA from the wild type mice, the disease is less severe. So this is kind of interesting. So that means the microRNA is involved in shaping the microbiome. Specifically, can we show that this microRNA can change specific microbe? We get some microbes as an example and some microRNA. We synthesize the microRNA and put in the culture of the bacteria. We can find that this microRNA can get into the bacteria. Here is we use a GFP labeled E. coli. We find that the localization of the microRNA with the bacteria. Here's just another tool to look at the microRNA in the bacteria used in the flow cytometry. So how does this work? We compare the sequence of the microRNA versus the gene of the bacteria. We found some microRNA can really well pair the bacteria gene, and then we synthesize the microRNA and culture with the bacteria, and we find that this microRNA can change the growth of the bacteria. Here's an example of fusobacteria, and then another example of the E. coli. And this is seen to be sequence specific because this one, so this mere 515 can only increase the fusobacteria. It can not increase the abundance of E. coli, and vice versa too. And then the reason, I think, is because the microRNA can regulate gene expression, as we show here. This particular microRNA can increase the gene expression of 16S RNA in the fusobacteria, and this particular microRNA can increase the YGH in the E. coli. So come back to the question. So I just was curious of which system is more important, innate immunity, adaptive immunity, or here the microRNA in the gut. So we use the infectious bacterial infection model. We infect the mice with the Citrobacteria. We compare the mice of the wild type B6 mice or the wild type little mice of the gut epithelial cell Dyson knockout mice. We also compare to the REG1 knockout mice and mediated knockout mice. We found the Dyson epithelial cell knockout mice is the first one, the most severe one, because it really died out before like 10 days after infection. And the other mice, the mediated knockout mice is secondary severe. The REG1 knockout mice is first severe, and the other mice can totally recover after a while. So this evidence suggests that we already identified the microRNA in the gut, and we show that these fecal microRNA are mainly produced by the gut epithelial cells, and this microRNA is able to get into the bacteria, specifically regulate the gene expression of the bacteria, and affect the growth of the bacteria. And we show that by transfer the microRNA to the recipient, you can change the microbiome. So we conclude that this fecal microRNA can shape the gut microbiome. Okay, now we go to the second part. The second part, I'm going to talk about the specific microRNA that we found in the gut of the multiple sclerosis patient, and also in the mouse model of MS-EAE. When we look at the microRNA profile by sequencing, we found a specific microRNA MIR30 that is increased in the EAE model as well as increased in the MS patient. And this is something interesting because when we synthesize this microRNA and give to the mice, give to the animal model of the MS, we found this MIR30 can improve the disease. And when we treat the mice with antibiotics, while we treat the mice, when we treat them with MIR30, we found that this phenotype disappeared. That means the MIR30 is working through regulate the microbiome. This actually is also evidenced by the fecal transplantation. When we take out the feces from the MIR30 treated mice and give to the antibiotics pre-treated mice and then induce EAE, we found that those recipients received the fecal samples from the MIR30 treated mice. treated donors, the disease is less severe. So then this suggests some microbe in the feces mediated the effect. So, and then which bacteria is mediated this effect? Then we look at the microbiome changes in the MS patient as well as in the EAE model. As we published previously that the specific bacteria named Akkermansia muciniphilia is increased in the untreated MS patient. And this is just the qPCR data. Also, another cohort also showed that is increased in the MS patient. And when we look at the animal model, we also find Akkermansia was increased in the peak EAE. So we asked whether this particular microRNA method D can regulate Akkermansia. First, we look at whether this microRNA can get into the bacteria, which, as we show here with the electron microscopy result, this microRNA do get into the bacteria, and it can, by informative analysis, show that this potentially can regulate this particular gene in the Akkermansia. As we show here through the qPCR, it do upregulate the gene expression of the of this particular gene. And then when we culture this Akkermansia with method D, it can increase the abundance of Akkermansia. We give this particular microRNA to the mice and then profile the microbiome. We didn't see the shift of the general microbiome profile, but we do see it increased Akkermansia, particularly Akkermansia. And this is just qPCR data. So it seems method D increased Akkermansia, and through this change the phenotype of EAE. So the next question is, does Akkermansia really improve EAE? Then we did the experiment, and then we induce EAE and then treat these mice with Akkermansia. We found that Akkermansia do reduce the severe of the disease. How does this work? So we observed that in the Akkermansia treated mice, the regular T cells were upregulated. And then we do the in vitro culture. So how does this microbe Akkermansia upregulate Tregs? We culture the bacteria with naive T cells and trigger them to develop to Tregs, THEP17, THEP1. We didn't see it change THEP1, THEP17, but we see that upon co-culture with dendritic cells, Akkermansia can increase Tregs. We didn't see it by pure culture, the naive T cells, it cannot upregulate. Only when we co-culture with dendritic cells, it can upregulate. That means dendritic cells is involved in recognize or in response to Akkermansia stimulation. And then when we look at the dendritic cells, what changed upon Akkermansia stimulation, we found it can increase the secretion of TGF-beta and secretes much less IL-6 and less IL-1. These cytokines are important in the differentiation of T cells, and with less IL-6 and with a high concentration of TGF-beta, you get Tregs. So this, I think, is a mechanism. So here I just want to summarize the second part. We found the microRNA method D is increased in the feces of peak EAE and untreated MS patient. And we found the synthesize the MIA30D by giving orally, it can can improve EAE in the microbiome dependent way. We found MIA30D can enter the Akkermansia, upregulate expression of the lactase in Akkermansia and expanded the bacteria in the gut. And Akkermansia can expand the phosphate and positive Tregs and then improve EAE. Okay, that's the story. I would like to acknowledge my colleagues, Dr. Weiner support the whole study, and a lot of colleagues in the very helpful help that doing the experiment or analyze the data. Thank you for your attention.
Terrific, Dr. Liu. That was fascinating and really stimulating. Just to remind the audience, EAE stands for experimental autoimmune, and then what?
Encephalitis.
Encephalomyelitis. And your readout for that is what?
We read out this clinical score. Sorry, it's a clinical score. Yeah. We also show those data in the histologic data, but when we present, we just put here and the EAE scores, clinical score. The higher the score, that means the disease is more severe.
So really fascinating that these microRNAs somehow look to be involved in not just as a consequence of disease, but in the causation of disease somehow in an autoimmune disease. And they seem to do so by regulating the composition of members of the microbiota.
Yeah. What we found there actually very interesting is we found, so in the gut microbiome profiling study, including ours and a lot of others, cohorts show that Akkermansia actually is more abundant. They found more abundant in the patient, in the disease patient. So people were thinking probably this is a pathogen induced MS or induced in the mice induced EAE, but actually we found it's not. It's just it's a protective mechanism so that a patient, when one get a disease, he or she is going to get like a response. And in this case, most likely it can generate like a MIA30D, and this microRNA can activate this bacteria, and this bacteria play a protective role that these bacteria can just come to recover the disease.
And Amanda asked a question that was crossing my mind as well. So how do these microRNAs then get taken up by bacteria?
The mechanism how they take up the bacteria take up the microRNA, we still don't know. We are working on that. I think it's in general is something like so we know there are a lot of horizontal gene transfer, and probably it's a similar mechanism, but we don't know at the moment.
Fascinating. And do you have any sense for the relative stoichiometry of the amount of microRNAs versus the number of bacteria in proximity to the epithelial boundary to know how saturating is the availability of those microRNAs?
This, you mean the ratios or how about this? Actually, I don't know, but what I can say is the abundance of the microRNA in the feces or in the gut lumen is is very, very high. It's much more higher than those you can find in the blood, in the serum and plasma. But the question you're asking is what the ratio of whether the 1:10 or 100 ratio regarding the gut epithelial cell number, the gut bacterial cell number, or in the microRNA number, and we didn't quantify that. I think it's kind of hard because we can say, okay, we can say how many nanograms or picograms of microRNA we can find in a particular weight of feces or particular weight of gut lumen content, but we cannot say the exact number of the microRNA.
Sure. And one other question, a couple of other questions, but one is we talked about how they that we don't know much about how they get into the microbial cell. How do they get out of the epithelial cells?
We do look at them. So actually, this is related to the protection mechanism, how this microRNA exists in the gut. We look at the microvesicles in the feces in the gut lumen. We found a lot of microvesicles of different sizes, and we found that this microRNA, particular microRNA, a lot of them actually can exist in the microvesicles. Most of them is in the microvesicles of size in around 100 nanometer. And we also find a lot of them actually they're not within the microvesicle. They just can exist in a vesicle-free state. So I think some of them is secreted from the gut epithelial cells in the form of the microvesicles. Others probably just release. I don't know in which form, or because when we profile the microvesicles, we use a size filtered approach. We just go down to 20 nanometer, the size 20 nanometer, and then below 20 nanometer, we say this is vesicle free, but we are not sure whether it's really vesicle free. But yeah, that's what
Great.
I think. Yeah.
And then just in the 1 second or a few minutes, few seconds we have left, is there any known role for these microRNAs? This is a question from Hyman Hartman. Any known role for the microRNAs in the epithelial or other cells themselves, host cells?
Yes. Of course, microRNA endogenously exist in the host cells, but they are playing different role. Yes, and most of them. But
for example, the one that affects Akkermansia. Well, what's known about its role in the cell? Anything or?
Yes, in the circulation, actually, there are already paper talking about this microRNA and the target in the heart tissue, in the cardiovascular side. Sorry, but I cannot. It's like a muscle tissue in the heart.
Okay.
The cardiomyocytes, I think.
Yeah, exactly.
Yeah.
Thank you. So they can regulate the proliferation of these cells. I didn't see it can directly change the behavior of the gut epithelial cells. I didn't see any literature talking about that. But in the case, this is if we come back to a general state, we found some of those microRNAs is specifically secreted to the gut lumen. They are not they have really high concentration in the gut lumen, but much less concentration in the gut epithelial cells. That means they're by purpose secreted to the gut to do something.
Great. Well, thank you again. That was really quite stimulating and a very fresh and interesting perspective on the dynamic between microbes and the host. So thank you again.
Thank you.
With that, Hannah, let me turn it over to you to tell us about David Goodsell's art.
Awesome. Yes, I will be queuing up a feature on Dr. David Goodsell, who is a professor of computational biology at Scripps Research Institute and a research professor at Rutgers State University. His research centers on methods for computational structural biology and their application to drug design, protein function prediction, and modeling of the molecular structure of entire cells. He has also written 4 general interest books on molecular biology, cell biology, and bio-nanotechnology, and has collaborated with science museums, filmmakers, educators, and popular authors on the creation of educational and outreach materials. And you'll see why he's such a good fit for that in this short video. We'll first share about a 3-minute video profile, and then I'll share a looping slideshow of his art for the remaining time slot.
My name is David Goodsell, and I currently divide my time between research in computational biology at the Scripps Research Institute and science education and outreach with the RCSB Protein Data Bank. For many years, I've been creating illustrations that simulate what we might see if we could magnify cells large enough to see individual molecules. This painting is one of the first ones that I did way back in the 1990s when I was a postdoc. It shows a cross-section through an Escherichia coli cell. I chose E. coli as a subject for this painting because it was the only organism that had enough information at the time to support an image like this. I recently decided to update the E. coli painting as part of the 50-year anniversary of the Protein Data Bank. There's an amazing amount of new information on E. coli structure and function, so the painting has a lot of new details. I'll spend a few minutes now and walk you through the process of doing one of these paintings. First of all, I spend most of my time on these paintings before I ever pick up any pencils or brushes. I began by researching the 300 most abundant proteins in a recent proteomics study, as well as a few less abundant wonders of nature that really had to be included. For each of these molecules, I used MolStar at the RCSB Protein Data Bank site to view the molecule and measure the overall size and shape. I then created style sheets for the proteome, drawn in my simple cartoony style, and all at a consistent scale of 2 million times magnification. Using these style sheets, I then built a sketch for the entire painting. I started with the nucleoid, which is really always the hardest part, sketching in all the DNA and the associated proteins, with much use of that eraser. I then filled in the cell wall and the flagellar motor, and finally all the soluble components. And then I was ready to start painting. I transferred the sketch to heavy Arches watercolor paper using carbon paper and started laying in colors one molecule at a time. The foreground is painted with bright colors that highlight the different compartments of the cell. The cell wall is in greens, the nucleoid is in warm yellows and oranges, and the soluble cytoplasm is in blues and purples. Finally, when all the tiny little white spaces are filled and I've added outlines around everything, the painting is finished. These paintings are often used in textbooks and other educational materials to help people understand the crowded nature of cells. I also love working with scientists to develop pictures related to their own research. They're also a lot of fun to do. Each new topic is like a treasure hunt to chase down all the information I need. I've been doing a lot of work with viruses recently and vaccines. These are all freely available at the RCSB Protein Data Bank website and are included in the following slideshow. I hope you enjoy them.
Great, so now I will share a slideshow of some of those images that he's just referenced, and this will loop for the next 10 minutes. So in total, going through the slideshow once is about 2 or 2.5 minutes long, so make sure to stick around to see the slideshow through at least once, and then if you need to walk away for a moment, it'll be looping until 2 p.m. Just a second. Just kidding, false start there. Hang with me here just a moment. Okay, here we go. Okay, Mike, I'll turn it back over to you to introduce our next speaker.
Super. Thank you, Hannah. I neglected to mention it, but our previous speaker, Dr. Shirong Liu, is over at the Brigham. Our next speaker is at Mass General, Christina Faherty, Dr. Christina Faherty. So we have great representation from the new MGB, Mass General Brigham Consortium, that incidentally also includes Mass Eye near where I am. Again, Christina's focus is on bacterial pathogens, especially Shigella. And she's trying to understand it to a degree that would allow the ultimate development of vaccines. Christina, tell us what's new with Shigella.
Sure. Thank you very much for the invitation to speak and the introduction. I'm just sharing my screen right now. Hang on one second. Okay, can everyone see that?
Yep.
Okay, great. So Shigella contributes to the global burden of diarrheal disease. And I'd like to start out with just noting that we are having success in this fight, and primarily due to things like clean water and sanitation, better nutrition, and the rotavirus vaccine. However, we still have a long way to go. Diarrheal disease is the 9th leading cause of death, at least pre-COVID, and accounts for millions of infections and deaths per year, especially in children under the age of 5 years. So the current challenges, especially with bacterial pathogens like Shigella and even E. coli and Salmonella, are malnutrition, antibiotic resistance, and a lack of effective vaccines. And this is despite significant amount of research that has occurred over the last 40 to 50 years, and actually understanding important mechanisms of disease. However, we're unable to really reach that successive therapeutic advantage, especially on the vaccine front. So just to provide a little bit of background about Shigella, it is a gram-negative bacterial pathogen, and it is very human adapted. So it was discovered in the early 1900s, and it has really co-evolved with humans. And that's something to kind of keep in mind as I present my work here today. There's 4 species of Shigella that are pretty much 90 to 95% homologous, and there's multiple serotypes that circulate the globe. For my talk today, we will be focusing on Shigella flexneri. So the pathogen causes watery diarrhea and bacillary dysentery. And to do that, it harbors a 220 kilobase virulence plasmid that is separate from the bacterial chromosome. And encoded on the plasmid are important proteins for infection, including invasion and virulence genes, as well as a type 3 secretion system. Shigella also induces the watery diarrhea by secreting enterotoxins, and then initiates that bloody phase of diarrhea by invading the colonic epithelium. And I would like to note, since Shigella flexneri does predominantly affect the global community, especially in developing countries, it is prevalent as well in industrialized nations in Europe and North America. And currently there's a Shigella flexneri outbreak in Vancouver, Canada. So as I've mentioned, we do understand mechanisms of infection for Shigella. And here is a model of what happens for invasion of the colonic epithelium. The bacteria can't invade on the top or the apical surface, so it needs to find and sort of access these antigen sampling M cells that are present in the epithelium that are normally there to sample the environment and to decide if we should be initiating an immune response. The M cells are actually few and far between in the colon, especially relative to the small intestine. Yet we know the bacteria are able to target these cells and exploit that antigen sampling uptake. Shigella first needs to, once it reaches the basolateral side, first needs to get around the innate immune cells, particularly the macrophages. So Shigella is able to sense once they are engulfed by macrophage and very rapidly induce cellular death. And then they are available to invade the enterocytes at the basolateral pole, and where they can then replicate and spread to cell, from cell to cell without having to re-enter the extracellular environment. So what we do know regarding tissue destruction is that that is mediated mostly by the neutrophils that are sent to the site of infection, and these neutrophils are actually what's responsible for the bloody diarrhea phenotype and the tissue destruction that we see as a result of infection. So as I've mentioned, especially classically, we've really appreciated that as little as 100 organisms are needed to cause infection through the fecal-oral transmission, and that the bacteria can invade in the colonic epithelium. Additionally, what we knew classically when you think about gene regulation and virulence regulation in the host was that the body temperature at 37 degrees Celsius was utilized by the bacteria to turn on the virulence plasmid. So in environmental temperatures, the virulence plasmid is turned off. However, once inside the host, the bacteria is able to sense that and turn on virulence expression. But besides those classic examples, there was limited information about what we really could understand as how well can the bacteria successfully transit the host, and how does the bacteria regulate virulence gene expression, especially pathogens like Shigella that do not want to waste energy expressing virulence genes too early and only like to express them when needed at the right time. So if you think about transit through the human gastrointestinal tract, there's many barriers that need to be overcome, especially low pH of the stomach, for Shigella surviving transit through the small intestine. And then once inside the colon environment, there's an anaerobic environment and needing to overcome things like mucus and the commensal population. So I became very interested in what happens during the small intestine, because that is where the bacteria spends the majority of its time prior to causing infection in the colon. It takes about 12 to 24 hours to transit through the small intestine. And what's interesting about the small intestine is that bile is pretty much localized to this area of the GI tract. It's only 5% enters the colon, so the majority is actually recycled back into circulation and deposited in the gallbladder. And another interesting aspect about bile is that it's bactericidal, so it's a really great innate host immune defense. However, pathogens like Shigella are known to survive it, and these mechanisms were not well appreciated, especially in the early 90s and 2000s. So the overall hypothesis for our lab is that Shigella resists bile and utilizes small intestinal conditions as a host localization signal to help optimize virulence. And today I really want to focus on the virulence aspect of some of the work we've performed. But first I wanted to introduce what we discovered when we exposed the bacteria to bile. So in the lab, we actually predominantly use a mixture of bile salts, since that is about 80% of the composition of bile. And what we can do is supplement bile salts into the laboratory growth media and then do our various investigations. So we found that Shigella can resist the physiological range of bile salts, which is about 0.2 to 2% weight by volume, depending on the person and diet. And we found a significant mechanism involved in this bile salt resistance was the induction of, the ACR-AB efflux pump, which is very much important for antibiotic resistance. It can expel at least 20 different classes of antibiotics outside the bacterial cell. So we found that these genes were induced in the presence of bile salts, and that an ACR-B mutant was unable to resist bile salts. And so this has important implications for antibiotic resistance. It's very much conserved amongst the enteric pathogens. They all utilize this pump to resist bile. And if you think about it for future therapeutic development, it's important because the sheer fact that enteric pathogens are exposed to bile at some point in the infection cycle is actually priming them for reduced antibiotic susceptibility or increased antibiotic resistance, depending on how you want to look at it. We also found that the bacteria in the presence of bile salts forms a biofilm. And this actually turned out to be an interesting story, because when we were utilizing different laboratory media like LB or tryptic soy broth and adding bile salts to that, we were noticing different phenotypes. So in tryptic soy broth, which has added glucose relative to LB, and then in the addition of bile salts, we were finding this biofilm formation happening. And so it turns out these both bile salts and glucose are needed to form a biofilm. And this is really important for the extracellular matrix that's produced by the bacteria when they're exposed in these conditions. So we really believe that we're starting to really understand transit through the small intestine with these mechanisms, and that bacteria will be exposed to bile salts and to glucose as food is being digested, and those simple sugars are being released by the digestion process. And this allows them to sort of transit the small intestine and actually helps to explain why they possibly are not infecting in the small intestine and only in the colon. Now, we knew once we started detecting the biofilm formation that we'd have to understand a dispersion mechanism, because how else is the bacteria going to invade the colonic epithelium if they're inside a biofilm? So we began to investigate, and we found that the removal of the bile salt signal from the biofilm was enough to start a dispersion event. And so that actually mimics what happens as you transition from the small intestine into the colon, as I mentioned, where you only have about 5% of bile entering the colon, and the majority of bile is in fact being recycled. And then finally, we found extensive changes in gene expression through RNA sequencing analysis. So not only did we find changes to help resist bile, but also even things like suggesting an adaptation to the anaerobic environment. And then we saw significant induction of virulence factor expression. So our model at this point was, after the dispersion from the biofilm and resisting bile, that the bacteria are sort of entering the colon in this hypervirulent state relative, especially to growth in laboratory media, and that they're ready to cause infection. So we were kind of really interested in pursuing this hypervirulence, and some work done initially in the 1990s by Shelley Payne's group and Wendy Picking's group demonstrated that if you expose Shigella to bile salts, that actually induces the adherence and invasion phenotype. The invasion phenotype was worked out very well by the Picking's group, in which the bile exposure affected one of the invasion proteins, IPA-D. And then so we knew that this phenotype was happening, and we were really interested in exploring the adherence aspect of it. So combined with the previous literature and what we were seeing on our RNA-seq data, we started to look at performing adherence assays with colonic HT29 cells. So if we grow the bacteria in subculture conditions with glucose and bile salts, we had a significant induction in adherence relative to bacteria grown in media alone. I would just like to note that we do standardize the bacteria to the same inoculum for these assays. And then also we are only exposing the bacteria during the subculturing conditions. And so what we do is we grow the bacteria in these conditions, wash and resuspend in tissue culture media, then that sort of helps to mimic that small intestinal to colon transition, and then applying the bacteria to the cells. So there's no bile salts or glucose added to the actual epithelial cells. So we saw this really significant induction in adherence factor expression. And then so that led us to the question, to adherence. So that led us to the question, does Shigella make adherence factors? And this is actually an interesting question when you look at the Shigella literature, because this consensus was that the bacteria do not express adherence factors. And that was primarily due to the fact that when you grow the bacteria in standard LB laboratory media, you do not see any adherence factors being expressed. The bacteria look completely bald. However, if you then compare that to E. coli under the same conditions, you'll see very much significant expression of the adherence factors. So coupled with these observations, and depending on which species of Shigella you're looking at, the genes are either not present to encode for the adherence factors, or they're present, but they're annotated as pseudogenes, either due to various mechanisms, which I'll get into in a minute. So we knew we had a little bit of a hurdle to overcome with this, especially with what's been established in the literature. So our first approach was to look for the adherence factors from both the biofilm assay and then also adherence to epithelial cells, and then go in and look at the genetics behind the expression. I just want to note that at this point, we started to move away from the traditional cell lines like the HT29 cells, and then started utilizing organoid cultures, which have become very popular in the past 5 years. They're a more physiological relevant model of different sites of tissues, especially for the gastrointestinal tract and for the lung. And so what's really an advantage to them as well is that you can get the expression of the different cellular types that are present in your organ of interest. And then also it's usually in a non-cancerous cell line. So at the center, with our collaboration with Dr. Stefania Senger, she was able to utilize this modeling for her studies with celiac disease. And so basically, if you can isolate the crypt cells, the stem cells from the crypts of the intestinal epithelium, you can propagate them in culture and develop these spheroid or organoid cultures. And others in the literature, as well as Stefania, found that you can then trypsinize these cultures and seed them onto a transwell system. It's very similar to polarized TD4 cell lines and things like that. And so this actually was really helpful for us from a pathogenesis perspective, because we can then look at applying the bacteria directly to the apical surface. The problem with the spheroid cultures is the apical surface is on the inside of the spheroids. So absent of any type of micro injection of bacteria into the cultures, this transwell system offered a really nice format in a multi-well format to do our analyses. And then when we do things like add differentiation reagents to the media, we can make sure we get expression of enterocytes, mucus-producing goblet cells, and those antigen sampling M cells that are really important for Shigella. So here we're just showing a top-down SEM view of the monolayer, where you can see that you get that same single cell that lines the intestinal tract. Anything fuzzy here are enterocytes expressing microvilli. These darker cells are cells that don't express microvilli, which are typically the M cells. If you do some transmission electron microscopy, we see really nice microvilli expression, as well as the nice tight junction and barrier formation that is important and an important function of this model for your intestine. And then, so we also looked at other cellular markers to make sure that we did in fact have expression of the M cells. And here we're showing we get very great mucus production from the goblet cells. So we really had a nice physiological model. We went ahead and characterized Shigella infection on the model, as well as some other enteropathogens like Salmonella and E. coli. And so we found, for the most part, especially when we utilized our subculturing conditions, we had great adherence to the enterocytes. As you can see here, again, the microvilli are on the top, and we saw nice adherence. And then where there were areas of cells that were not expressing microvilli, so most likely the M cells, we saw areas upon SEM analysis where it looked like the bacteria were starting to penetrate those cells. So then we went on and further characterized, and we found that the best invasion rates occurred when we had conditions that induced the M cells. And then comparing different samples along the gastrointestinal tract, especially relative to the small intestine and colon, we were getting the best invasion rates in the colon, indicating we had a nice model that replicated the tissue tropism of Shigella, and that also we can infect in a physiological relevant manner by exposing them to the apical surface, as opposed to directly exposing the bacteria to the basolateral surface. So this model is very helpful for our studies in the lab. And so back to the adherence story, when we looked at the bacteria grown in the biofilm or following adherence to the organoid model, after we subcultured the bacteria in bile salts and glucose, we could see great expression of adherence factors, or what looked like adherence factors to us, in both conditions. And so again, we are, if the bacteria are from the biofilm, the bile salts and glucose are present in the media. However, if there's adherence to the model, again, this is only during the subculture and not during the actual infection. And again, we can see the really nice adherence factor expression relative to growth in laboratory media alone. Again, we did need both the bile salts and the glucose signals, because if we grew the bacteria in LB plus glucose or just LB plus bile salts, we did not see the appearance of the structure. So we did need to have that combination. And so for the most part, we saw three types of structures, these thicker structures that you can see here, thinner structures, and then these protein aggregates that were very evident and disorganized. but still present. So we not only detected expression under these conditions in our laboratory strain of Shigella flexneri 2457T, but we also detected in other strains and clinical isolates of Shigella that we had in the lab. So we knew we weren't working with some fluke with our lab strain. And so just looking at the binding again, the three structures seemed evident for two different cell types, with the thin structures binding to the microvilli, and then the thicker structures and those protein aggregates binding to cells that lack the microvilli, most likely the M cells. So now we had a justification to genetically go in and look at the genes and expression of these factors. And a very brief crash course in adherence factors. Many genes are needed to actually express and secrete these factors onto the outer surface of bacterial. So for the most part, you have structural genes that are assembled on the outer surface that can be divided between major structural subunits and minor structural subunits. And then there's also the pores or the ushers that are needed for secretion. So you have the pore in the outer membrane, and then the chaperones that bring the individual subunits through the pore to allow for this assembly on the outer surface. So common types include type 1 Fimbriae and curli, especially in E. coli. Now, I know this slide's a little busy, but basically what it's showing is that for the most part in Shigella, the genes were intact and transcribed. So based on the appearance of the structures, we thought the thicker structures looked like long polar fimbriae, the thinner structures looked like type 1 fimbriae, and those protein aggregates looked like curli to us. And so we were able to utilize our RNA-seq data and map transcription to these operons, and then also confirm the expression of either the single genes or the operons by RT-PCR analysis. So for the most part, LPF, the genes looked very much intact for the expression. And to zoom in and focus a little bit on the type 1 fimbriae, the reason type 1 fimbriae were thought not to be expressed was because there was a transposon insertion in the usher or pore gene of FimD. But as you can see from our data that we do get significant expression, and this is about 90% of the gene relative to E. coli. So we were able to get expression of all the important components and segments of the operon, even though the operon itself is not co-transcribed together. And then for curli, we had the same problem with the transposon insertion, except it occurred for the structural gene CSGA. But again, we were getting about 90% expression of the gene, and then other expression of either the individual genes or the actual operon components. Also, the problem with curli was in the pore, there is a point mutation that leads to a stop codon. So we were able to confirm that, and that we would see expression up until the stop codon, but then not after that. So the annotations were correct. It's just that the annotations didn't mean that these genes were not being at least transcribed. And then one more experiment that we did to confirm at least transcription was to see if we can induce transcription. And so in the literature, glucose is a great signal to induce these major subunits. And so we did some qPCR analysis of bacteria grown in media with or without glucose, standardized the bacteria, of course, to the same amount. And so as you can see with our control, the glucose didn't affect expression of this gene. However, for all of these structural subunits and some homologs, we saw significant induction of the genes with glucose. So at this point, we were very confident that the genes were in fact being at least transcribed. And so we went ahead and made mutations in the major subunits of LPFA, FimA, and CSGA. And you can see we had significant reductions in both biofilm formation and adherence to the organoids. For the adherence to the organoids, especially FimA, was a significant reduction. We lost the structures upon TEM analysis as well. And then since we're mostly a bacterial genetics lab, we went through and complemented the mutations. We went ahead and made C-terminal histidine tagged fusions that would help us with some protein work. But basically, we wanted to see if these constructs would then restore function in the mutants and if the tag would interfere with function. So we did this for all three. I'll just show one example here with type 1 fimbriae. The mutant again loses those thin structures, as you can see here. The complement very much nicely restored the structures, and we got nice expression on the outer surface of these structures, and you can really appreciate that we did in fact were getting the right appearance as we suspected from earlier in the slides. And then most importantly, the construct with the tag was able to restore the biofilm phenotype. So just to kind of appreciate the appearance of the structures, looking at the complements, again, the long polar fimbriae really appeared to be those thicker structures, the type 1 fimbriae, those thinner structures. And then the curli, despite disorganization and a lack of assembly, especially relative to E. coli, they're there and it is functional. So we're really confident that we've identified 3 structures utilized by Shigella that are important for biofilm formation in the small intestinal conditions, but then also adherence and making that first contact with the surface of the epithelial cells. We went ahead and did confirm that the proteins, especially the tagged versions of the proteins, can be secreted and present on the outer surface of the bacteria. So utilizing the biofilm culture supernatants, we were able to detect the tagged protein secretion by mass spectrometry and immunoprecipitation analysis. So we have a model where we feel like we have 3 structures. Again, as I mentioned, we feel LPF is for the most part intact. And then type 1 fimbriae, despite the truncation to the usher, we feel is being assembled and secreted appropriately. And then for curli, it is present, it is functional, however, it is disorganized. And that disorganization could be due to the fact that the pore is almost at close to a 50% truncation, or that assembly is not happening due to the truncation on the major subunit of CSGA. So we're hoping to really dive into those mechanisms in the future. But with that, I hope to convince you that Shigella is in fact utilizing adherence factors and really has an interesting lifestyle to consider when you expose it to the in vivo environment or the small intestinal conditions. So with that, I'm happy to take any questions. I'd first like to thank members of my lab, both past and present, that have taken all of our work forward and have contributed with all their hard work. For this adherence project in particular, soon to be PhD Rachel Shannon, who is about to defend at UT Southwestern, really got us started, and Dr. Courtney Nickerson really took over as she transitioned to grad school. And then we had additional help with Alejandro for the adherence with the model. And we've had a lot of interns come through the lab that have helped us, both undergraduates and master's students and even high school students. And particularly Jessica Ding and Kelvin Lee, Harvard undergrad students, helped us with the complementation analyses, and Sneha Dagapathori and Benjamin Kuzbar that helped us with the RT-PCR analysis. Our collaborators at the center, Dr. Senger and Fasano, especially for the use of the model. And then for the RNA sequencing, Dr. Rasko and Sistrunk at the University of Maryland. Ms. Ann Tidsdale and Dr. Kumar have helped us with both the scanning and transmission electron microscopy. And then for the mass spec analysis, Dr. Auclair and De La Parra at Northeastern University. So with that, I will very much be happy to take any questions.
That was really terrific, Christina. And as we've announced before, if you have questions, you can use the Q&A function. I have just a couple. So Shigella, so as you know, I work on the gram-positive side of the fence. And so Shigella to me is sort of like a rogue band of E. coli's, right?
Yeah.
And so would you help me understand a little more clearly the ecology? You mentioned they're highly human adapted. Are there any other hosts for Shigella? Do they always cause disease in humans, or is there a high rate of asymptomatic carriage?
Right.
What do we know?
Yeah, so they cause disease in humans, and then they do infect higher primates, but that's it. So there's never been a really great animal model for Shigella, which has also hindered vaccine development and things like that. So it's pretty much restricted to humans and higher primates. And It depends on the species, but it does seem like it causes infections. And there are starting to be some questions if there's any sort of asymptomatic carriage of Shigella, especially in endemic regions like in Asia and Africa. So we're actually interested in pursuing that to see if, what's going on with any sort of asymptomatic carriage for sure. But yeah, it's like I mentioned, it's very human adaptive, and that if you think about the human adaptation, you can really appreciate that it co-evolved with humans. And so it's going to utilize these signals to express virulence and prepare for infection, especially without wanting to waste energy once inside the host. Yeah.
So unlike Salmonella, it doesn't occur asymptomatically in turtles or anything like that.
No, not that we know of. So,
okay,
yeah.
no reservoirs. Let's see, we've got a question here from Dr. Chen. Fantastic research. I'm wondering if your lab has tested if any of those adherence factors play a role for colonization in a in vivo model, rabbit, mice, etc. Also, I'm curious to know, how does Shigella move between, I think, epithelial cells, but it says endothelial cells. Is that right? Or would it be epithelial?
Yeah, it would be epithelial. So we haven't, because of the human specificity, we've looked at the organoid models. There is a couple new rabbit and mouse models that are out there that look a little bit more promising to replicate human infection. So we would be interested in looking at adherence in that and help diving into the mechanisms. And then for the intracellular spread between epithelial cells, they utilize the actin polymerization like Listeria does, as Sarah mentioned during the lunch break. So that actually is really nice. It allows them to spread from cell to cell without having to re-enter the extracellular environment. So
terrific. Well, that's the time we have. I'm still dying to do some Enterococcus and organoid experiments, so we got to put that together. But with that, Hannah, tell us about the art of Anna Maria Porras, and then we'll get back to questions for Christina and the others in our 45-minute post question and answer period. Thanks again, Christina.
Yeah, so I'm going to now queue up our final SciArt feature of the day. And I'll repeat what Pete said earlier, which is to note that each of these folks that we're highlighting for their work in science communication and outreach through SciArt are also researchers in their own right. And so it's really neat to see how talented they are in multiple areas. And I'll also mention that each of the videos that you're seeing, these 3-minute video features of each individual SciArt feature, have been produced by Jennifer Berglund, who we've worked with through the Harvard Museums of Science and Culture. And so we're very thankful to her for helping us put these video features together. But with that, I'll introduce Dr. Anna Maria Porras, who is an assistant professor at the University of Florida. She's a biomedical engineer with experience in both tissue engineering and the human gut microbiome, and is particularly interested in understanding how geographic differences in gut microbiota composition impact host health and specificity, and specifically susceptibility to infectious disease. She believes in the power of art to communicate across cultural and national barriers, and at the same time, she's an advocate for and practitioner of multilingual science communication.
Soy la doctora Ana María Porras, soy ingeniera biomédica y uso microbios hechos en crochet para enseñar microbiología en inglés y español en redes sociales. Los primeros 5 microbios que hice los hice para el Festival Nacional de Ciencia e Ingeniería de Estados Unidos en el 2018 y fueron tan exitosos que después los seguí haciendo primero en inglés y luego un mes después en español en redes sociales. Yo no podría hacer lo que hago ahora sin los miles de mujeres en todo el mundo que han revitalizado y revivido las manualidades como arte. También me inspiran mucho otras artistas científicas que han traído las manualidades precisamente a la ciencia, la tecnología y la ingeniería. Yo creo arte para mis micromartes más o menos semanalmente. Todo empieza con una historia chévere que he escuchado en algún lugar. voy, busco mi personaje principal o el microbio, veo cómo se ve bajo un microscopio, dibujo un bosquejo de lo que yo creo puedo hacer con mis propias manos, cojo mi ganchillo de crochet y hago el microbio usando lana. En esto me demoro más o menos 3 a 4 horas. Lo tengo que fotografiar y después también hacer mucha investigación para asegurarme de contar la historia adecuada y completa en mis publicaciones en redes sociales que hago en inglés y en español. Las publico y después también tengo que seguir interactuando con el público para asegurarse que todos estemos aprendiendo. Hay millones de microorganismos en nuestro planeta y gracias a esto pues son una fuente de inspiración infinita para mí en mi arte. Al mismo tiempo, el arte me permite comunicarme visualmente sin importar las barreras del lenguaje. Y además, el arte me ha ayudado a apreciar la diversidad de los microorganismos que nos rodean y que están dentro de nosotros. He creado más o menos 60 microbios distintos en crochet. Sé que es difícil elegir a mi favorito, pero me gusta mucho esta Helicobacter pylori, me parece que me quedó muy tierna. Aunque últimamente lo que más he estado usando es esta representación de una nanopartícula de la vacuna del ARN mensajero. En redes sociales interactúo principalmente con mujeres adultas, pero también he usado mis microbios en otros eventos donde también han sido muy populares con niños y adolescentes. Mi idea en general es llamar la atención de la gente con la ternura de estos microorganismos y luego entregarles un montón de ciencia.
All right, Mike, I'll turn it back over to you.
Great. Oh, that was awesome. Those are amazing artwork. I really enjoyed that. Also enjoyed the guitar music that was kind of like game show guitar. I think that's a whole genre.
I think so too. I really appreciated this file from the royalty-free music archive.
Oh, there you go. Good. Well, thanks for that. Let me say again before I introduce our next speaker, in addition to the upcoming Boston area antimicrobial research network meeting, or BARN meeting, which will be toward the end of September, our next speaker reminds me that there's also the Boston bacterial meeting upcoming July 10th and 11th. I think, as most of you are aware, that's a meeting that's put on by grad students and postdocs, very grad student and postdoc centric, well worth attending for everybody, however. And their keynote speaker will be a mutual friend of mine and our next speaker, Arturo Casadevall. He's a terrific thinker, a very imaginative guy, and a great speaker. So put that on your list. The registration for the Boston Bacterial Meeting is right now, so look into that. Contact Hannah if you need more info on how to connect up with that. Again, our next speaker is Dr. Krysan, Damian Krysan. As I mentioned, he's a pediatrician with part of his time when he's not investigating fungal infections. And sometimes those things actually mix. He's looking at fungi that spend a lot of time in association with humans, as well as fungi that are more unusually intimately associated with humans. And there's some fascinating comparison and contrast. But what's common to all of those is treating fungal diseases is extremely difficult because fungi, as everybody is aware, are eukaryotes, and targeting one eukaryote and sparing the other, which is the host, can be a challenge. So this is extremely important work and an area where, we talk about antibiotic resistance, and usually gram-negative bacteria come up these days. But this has always been a problem with fungi and continues to be a problem with fungal infection. So Damian, let me turn it over to you. Thanks for joining us.
All right, thank you so much for the invitation. It's really a pleasure to be part of this very kind of unique meeting. So hello from the middle of the country in Iowa. And what I'm going to do here, let's see, is, can we see this screen here?
We can. Mm-hmm.
Oh,
yeah.
Yep. All right. So.
Oh, sorry to interrupt you, but just as an FYI, we're seeing your presenter view, so we can see, your notes in the next slide. Just so you're aware.
Damian, if you go to that circle with 3 dots underneath the picture, and then hide present. There you go. Okay, and now go there. Yep.
This is why we should have had our rehearsal, isn't it? Oh, why isn't it? Let me try this. Let me try this again. Go.
Yeah,
here.
try that. Yeah.
Still over there.
Yeah, but that's okay. It's a 2-screen problem, and Zoom doesn't handle that very well.
Just a second. Let me at least.
This will work unless you've got some animations.
No animation. I'm not that fancy.
Okay, well otherwise this is pretty much
I guess
fine.
we'll just have to go with it. I apologize. All right.
No big deal.
So, as Michael was pointing out, serious fungal infections are really a worldwide global health problem. And there are estimated to be about 300 million serious infections per year. And the slide I'm showing you here comes from an organization called the Global Action Fund for Fungal Infections. And the black circles are the conditions that predispose people to fungal infections. And then the colored ones represent a sampling of the most prevalent of the fungal infections that we have. And they really span a whole wide range of conditions, including, cancer, chemotherapy. The one we usually think of really quickly when we think of immunocompromising conditions that put one at risk for opportunistic infections is HIV. But we sometimes don't think of COPD, emphysema, asthma, which is actually extremely common in the world, can also put you at risk for fungal infections. And in fact, you can get fungal infections that can cause significant morbidity, in particular, with no underlying diseases. And these are fairly common unfortunately, in the developing world. And these are things like fungal keratitis, mycotomas, which actually involve direct inoculation of environmental fungi into people. And as Michael alluded to, one of the problems we have with these infections is we really only have 3 classes of antifungal drugs that are useful in treating serious invasive fungal infections. You compare that to things like the number of drugs we have for bacterial infections, it's huge actually. There have actually been more drugs developed for hepatitis C in the last 5 years than there are classes of drugs developed by humanity for the treatment of serious fungal infections. And so we really need to be able to develop new therapies, which is one of the things my lab is interested in. But sort of what we need to do maybe to improve that is understand a little bit more about the biology, the pathobiology of fungal infections, and in particular, how do they interact with the host? And that's sort of going to be the focus of this talk. Starting off from where do fungi come from? Well, how do we get a fungal infection? And in general, there are 2 sources of fungi. One is the ones that live within us. This is the commensal microbiome, if you will. And that includes mainly Candida species. We also have some like Malassezia, but Candida is really the driver here, and it colonizes our GI tract and our genitourinary tracts. And everyone is colonized with it. We have Candida within our bodies right now. If you alter the immune system, that's when we end up with disease, and the disease spans sort of the gamut from very minor things like thrush, babies, like the white plaques in babies' mouths in the first few months of their lives, to life-threatening, almost untreatable meningitis. So it's a very broad spectrum of disease that can come along with this type of infection. The other type of, the other source we have is the environment itself. And so these are, just what we come in contact as being members of this planet. This includes things like aspergillus, a common mold that we are all right now breathing. We're breathing the spores of that. In my part of the country of Iowa near the Mississippi River, histoplasmosis is a very important cause of fungal infections, but doesn't really lead to a lot of disease. And we'll talk about that later. A more famous one would be valley fever from the southwest part of the United States, which is California and Arizona. And so these are our 2 sources of fungal infections. And the types of, there are basically, estimated to be up to 5 million types of fungi on this planet. And however, out of those 5 million, we only have about 300 that have been documented to cause human disease. And so we are in contact with fungi all the time, but only rarely do we come across those that can cause harm. And this evolutionary tree I'm showing you here has the pathogenic fungi, a sampling of them in red. And you can see they actually span a fairly large sort of evolutionary space, if you will. Down at the bottom here, we have Candida species, which are, again, part of our own microbiome. Up at the near the top, these are all among the Ascomycota. We have Histoplasma coccidioides, which is the valley fever. Aspergillus was a mold that we breathe almost every day. Cryptococcus, and then some Zygomycota rhizopus infections, which are actually becoming a very big problem in our, particularly in our bone marrow transplant and immunocompromised patients. So a wide range of different fungi can cause disease. Probably the major bottleneck that selects the pathogens from the non-pathogens is the ability to grow at the human body temperature. Mammalian body temperatures is 37 degrees. There aren't that many fungi that can actually make the transition from the environment into the human host, unless that's their starting niche to begin with, such as Candida. And so when you talk about fungal pathogenesis, you have to think about the host. It is inextricably linked to the host. And because, as I sort of alluded to, essentially all the pathogens are either commensals, part of us, are organisms that we come into contact with every single day, such as Aspergillus, or are organisms that we get, many people with totally normal immune systems will get infected with and never get sick. And that's really like Histoplasma, Cryptococcus neoformans, actually Arturo, Casa de Val's work showed that in New York City, by the time you're 2 or 3 years of age, you have antibodies to Cryptococcus neoformans. And so these are things that we live with and live around. And generally when disease comes, it's because we mess up the homeostasis. We have, in general, over evolutionary time, established a détente, if you will, with these organisms. And the way you get sick is you either get too much fungi or you don't have enough immune system. Pretty simple as that. And there's a famous experiment from right after the Second World War, which is when, at that time, Candida albicans wasn't thought to actually be a pathogen. People just thought it was a contaminant in blood cultures and wasn't really causing disease. And so an English physician decided to drink basically a milkshake of what it amounted to a stationary phase culture of Candida albicans. He got sick, and he survived to write a paper about it. So that was a perfect example of too much Candida will make you sick. Out here in the Midwest, we don't have a lot. We have fairly sporadic cases of histoplasma, but in Indianapolis about 10 years ago, they ripped down this big amusement park in the middle of the town and had 100,000 cases over a 6-month period of time. And that's where you basically just took dust full of histoplasma and bathed the town in it and got a lot of disease. As I mentioned before, we more frequently think of fungal disease coming from changes in the immune system. So cancer chemotherapy, bone marrow transplantation. But actually, if you take an inhaler for asthma, which many people use, a controller medication where you take an inhaled steroid, if you don't brush your teeth right away, you get fungi in the corner of your mouth within about 2 or 3 days. And so it doesn't take much of a change in the immune system to have some kind of disease. We also have primary immune disorders that put you at risk, as well as HIV. And so the ability of fungi to actually cause disease and pathogenesis, the cycle of pathogenesis, if you will, there's a required step for where they move from their normal niche to a new niche. And that transition has to be navigated. For commensal fungi, that usually means going from the luminal side of, or the mucosal side of an organ, such as the GI tract or the GU tract. There's some change, they are able to translocate. Under normal conditions, we have dendritic cells and resident macrophages that clean them up right away. Or the barrier really keeps the amount of translocation fairly low. If you disrupt the barrier or you mess up the immune status of the patient, now you have translocation into deep tissue in the submucosal area. From there, they replicate, they disseminate to the rest of the body under some circumstances, in particular with profound immunosuppression. The transition, the adaptation, if you will, in environmental fungi is a lot more dramatic. They're coming from ground, air, compost heaps. They're breathed into the lungs of humans. They then, most of the time, go right back out and cause no problem. In the right conditions, they're able to get into the blood, into the tissue, establish infection, and then cause disease. And so this adaptation is really part and parcel of the pathogenic cycle. And so what I'm going to focus the rest of the talk on is questions about how does the environmental fungal pathogen Cryptococcus neoformans adjust to life in the human mammalian host. And I was really heartened to see Anna Maria have Cryptococcus introduced there in our last art segment. And I'll have one last thing to say about that in a couple slides. She was right on. She got her biology perfectly good. So Cryptococcus is a basidiomycete. It is a haploid organism. So it is sort of like Saccharomyces cerevisiae in that manner. And it also undergoes mating like Saccharomyces cerevisiae. It is, however, an environmental yeast. And when we're taking our board exams for infectious disease, the answer to the question is, where is its niche? Everyone puts down pigeon guano, but it's everywhere in the environment. And there are basically three pathogenic species. Neoformans variant Grubbii, which causes about 85 to 90% of the disease in the world. A less virulent form is Neoformans variant Neoformans. And then there is an emerging Cryptococcus species called Gaddii, which actually causes more disease in immunocompetent people than the other two. And there was an ongoing outbreak in the Pacific Northwest around Vancouver Island in the mid-2000s, early 2000 teens. One of the unusual parts of Cryptococcus is it has a capsule. So it has a polysaccharide capsule, something like the pneumococcus or Haemophilus capsule, which is a virulent trait. And so the pathogenic cycle is spores are breathed into the lung by people. Once it gets to the lung, 99.9% of the time it's completely cleared. It probably establishes a small latent infection, something like latent tuberculosis. And then if the immune system, particularly T cells in this case, are affected, it can then disseminate, and it is neurotrophic. So it goes to the central nervous system, and patients present typically with cryptococcal meningitis, which is invariably fatal unless it is treated. And it is a sort of a very indolent meningitis, not like Neisseria meningitidis or pneumococcal meningitis. And many patients who have HIV will only learn of their diagnosis in resource limited parts of the world by going to the doctor with a really bad headache, getting a lumbar puncture and finding out they have Cryptococcus, and then subsequently are diagnosed with HIV. And so the HIV really, this is a disease of HIV globally. About 250 to 300,000 people die every year in the world from cryptococcal meningitis. And the vast majority is in sub-Saharan Africa and Southeast Asia, where the rates of HIV are fairly high. And so many people have to survive their cryptococcal neoformans meningitis if they're going to take advantage of our advances in HIV therapy. So it's a critical disease to be able to treat if we're going to manage this and keep as many people as we can living with HIV. And as I mentioned before, in general, only a minority of fungal species will cause human disease. And when we look at isolates of Cryptococcus, for most fungal pathogens, the environmental isolates and the clinical isolates are kind of a completely overlapping Venn diagram. And what has been clear over the years is that's not the case for Cryptococcus. There seem to be distinct pools of strains, which very little overlap. And so one of the questions is what drives that and what makes a strain pathogenic and what are its characteristics? And the three big virulence traits, I wouldn't call these virulence factors in the traditional bacterial sense because their presence doesn't cause disease. We're talking about an opportunistic pathogen. But ability to grow at 37 degrees, ability to form this polysaccharide capsule is critical for the ability to cause disease. And then the reason that nice little round guy with the goggles that Maria was making is black is because they melanize. And so the cell wall has melanin. And if they don't melanize well, they don't cause disease as well. And so a lot of work has shown that capsule and melanin formation actually correlates with patient outcome. And so these are the big three traits. And if you take isolates that have all of these big three traits, they can grow 37, they can form capsule, they can melanize. They have very, very similar genetics. So here we have some sequence types listed at the bottom. On the axis here is pretty much is survival and mortality. And within these strain groups, you can see there's a wide range of disease formation. And you can't explain this by invoking differences in immune function, because all these patients in this particular study had T cell counts well below 50. And so there's something else going on here is impacting on a strain's ability to cause disease and the virulence. And so that was basically one of the questions we have are, what are the virulence traits are we missing? And from that kind of led me back to think about what is the transition like? This is starting with an organism that is out in ambient air, in a eucalyptus tree, if you will, and now it has to go to the bronchial tree. And one of the things, I actually was trained as a chemist, so I'm all about chemistry. And if you think about the CO2 concentrations, even though they are going up with global warming, that is true, they're still pretty low. And when you go from 0.04% CO2 to the lungs of a human, which is 5 or higher percent CO2, that's a heck of a change. There's a lot of orders of magnitude in that transition. For humans, if we start to breathe 8% CO2, we start to go to sleep really quickly, and then we pretty much forget to breathe. So that's a really big change. And I thought, and actually my colleague and I, this is a collaborative project, thought maybe there's something going on there. Maybe we should take some environmental isolates and some clinical isolates and ask if they both can grow in the presence of CO2 very well. And we were actually astonished to see that for once, a really good idea actually had some data that didn't destroy the great idea. And what I have here on the left in this figure A are a set of strains. H99 on the top is our laboratory strain. A's are environmental strains and C's are clinical strains. And if you go from air to CO2, you can see that the growth of these strains drops pretty significantly for many of them. Unless they're a C, they pretty much don't match. They fail the Sesame Street test. One of these doesn't look like the other. And this is done in RPMI media that is buffered. So this is not a pH effect. And we've done a bunch of other control experiments. The CO2 certainly can change the pH. When we went through a number of these and did a Fisher's test, basically we had mammalian data, this is mouse model data, and everyone that could grow in CO2, except for one pretty much, could kill a mouse, and the ones that couldn't grow in CO2 could not kill a mouse. And we have expanded this set even further now to really, and over hundreds of strains, and this is held. And they all make melanin, they all make capsule, and they are all growing at 37. So something is up with CO2. And we did a little more quantitative assays here using a competitive growth assay where we labeled H99 with GFP, and we do a 1-to-1 mixture and compete them both in ambient and 5% CO2. And again, the A's don't do so well and the C's do pretty well in the competition. So these strains have reduced ability to grow in the host at levels of CO, at host relevant levels of CO2. And when we look at the virulence, as an example here, this is H99 that's coming down and pretty much everybody's gone by 9 days. The next one over here is a an environmental isolate that actually was CO2 tolerant. And you can see not quite as virulent as H99, but does a pretty good job. Whereas the environmental strain, everybody's running around happy, drinking their water, looking unruffled. And when we took samples of brain tissue just before the H99 infected strains were dying, we could see that the brain burden here is pretty similar. Whereas in the environmental strain, it's over a log, almost 2 logs lower. And so it appears to be either an overall reduction in the burden in the lung or a failure to disseminate to the brain. And one of the things we're working on right now is trying to tease out exactly which of those mechanisms is working. The other thing we noticed is that host concentrations of CO2 affect antifungal drug susceptibility. And so here we have fluconazole strips laid down on lawns of Cryptococcus over a variety of different isolates. And you can basically see the zone of clearing is much bigger in the CO2 exposed strains than they are in the air. And that's true for fluconazole, one of the most widely used antifungals. It is not true for amphotericin B, on the other hand, which is another antifungal we use to treat Cryptococcus. And so host concentrations of CO2 are affecting drug susceptibility, and which suggests that maybe we should start to think about using more host-like conditions to look for our antifungals, because there's possibility there are ones that we never find in the absence of CO2. And that's one of the things we're also interested in. So what's going on? How does CO2, how does the tolerant strains respond to the host CO2 concentrations? And so one of the things we do, I mean, like everybody else, RNA-seq has become cheap and easy for the most part. And so what we looked at was a transcriptional response to exposure to host CO2 at 37 degrees. So this is like host conditions. And we were kind of surprised to see that not much happens after 4 hours. And a little bit happens at 8, and it really takes 24 hours before we see a significant transcriptional response to CO2. If you took Cryptococcus, for instance, and shifted them into like serum levels of salt, just a salt test, you see transcriptional changes within 30 minutes. Hell, even probably 15 minutes if you could get your cells harvested that quickly. So this is very different than a lot of other stress conditions. And when we looked at the transcriptional adaptation, it also has a relatively unusual pattern. And so here we have 4, 8, and 24 hours, and we're using Venn diagrams here. And we see almost nothing happening at 4 hours, like the dot plot showed. At 21 hours, there's a fairly significant set of down-regulated genes. Hardly anything's been upregulated yet. And then when we get to 24 hours, we have kind of a, now there's a transcriptional response I can do something with. I can do go terms, I can do everything. This other stuff is just kind of, what's going on? The interesting thing is when we look at the downregulated genes, they're completely different. And so there's a initial there's this stepwise transition in the way the cells are responding at the transcriptional level. Many of the genes that are downregulated at 8 hours are actually genes involved in cell cycle and DNA replication and protein translation. So it looks like, and our growth curves look fit this. There's almost like an extended lag. They don't replicate very well, and then once they do, they start going. And so there's this sort of stepwise adaptation that we're still trying to work into. These are completely unpublished data and really aren't that old. So this is a new story that we're trying to work on here. When we do a go term analysis on the upregulated genes at 24 hours, the one that really stands out from everyone else was these carbon-related redox genes. And we got 28 of them that are upregulated at this stage. And these are things like alternative oxidase, succinate dehydrogenase, a lot of genes that are involved in energy metabolism, mitochondrial rewiring, and also basically reducing equivalent homeostasis. And so the CO2 appears to be inducing basically a metabolic rewiring. And it appears to take a while to do that. And so we had a really simple experiment after that is, okay, well, the RPMI is a very low carbon glucose media. It's pretty much a starvation media for Cryptococcus. They don't like to grow in it all that well. What if we give them some glucose? What will happen? And it turns out when we use rich nutrient media, this increases CO2 tolerance dramatically in otherwise intolerant strains. And so YPD is kind of the LB plus. If we think of Christina's media, it'd be like taking LB and dumping glucose in. And so this is 2% glucose, and it's basically just peptone. So it's chewed up amino acids. And as you look here, this environmental strain near the bottom here is very sensitive in RPMI, but it's much more able to grow in the presence of this rich media. And so it seems as though carbon metabolism is being rearranged. And as a result, there's almost a synthetic effect between the high CO2 concentrations and the relatively low glucose, low nutrient conditions that the Cryptococcus runs into in the host. And so the model we're working on right now, our working hypothesis is that there's another bottleneck here. And from going from the eucalyptus tree to the bronchial tree, the CO2 is very high in the lung, but also the glucose is abnormally low compared to serum. It's 1/50th the serum concentrations of glucose. So it's a low glucose, high CO2 environment. And we think that this is putting up basically a selective barrier, and only certain environmental strains are able to pass that and actually cause disease. And so to finish up here, what we've come across is that we really have to broaden our ideas about what it means to transition an environmental pathogen into a mammalian host. And we have found that host CO2 concentrations appear to be a barrier and a significant stress in and of themselves, and also may synergize with the low nutrient status. And this could be have implications for the pathogenesis of things like histoplasma, coccidioides, other primary environmental organisms that are going through the lung as part of their pathogenic cycle. And then we found that there's this stepwise kind of delayed transcriptional response to CO2 stress. And we've identified one transcription factor already that seems to modulate that. And that's what we're really focusing on in that part of the project right now. And the one thing that gets me excited as a pediatrician, an infectious disease doctor, is it looks like Cryptococcus in particular might be an organism where we really want to think more about figuring out more host-like conditions to do our antifungal drug screening, and that may lead to new compounds that we can go ahead and transition and start to work on. And I have over here the improvised CO2 scrubber from Apollo 13. So if you remember right, CO2 was an important thing, and if they weren't able to cobble that together from duct tape and their cardboard bags and everything, Apollo 13 would not have been a very good movie for Tom Hanks at all. And so with that, I have to acknowledge the folks who did the work. This is a collaboration actually that has a connection to Boston. So Shirong Liu and I spent 5 years as the course directors for the medical mycology course at Marine Biological Laboratory in Woods Hole. And so over those 5 years, we kept having all these conversations, and finally we decided, okay, let's try this. And it turned into a really nice project. In my lab, Andrew Jazuski and Lori Risto have been the prime drivers. In Jerron's lab, it's been Ben Chadwick, and before him, Zing Din B. And then the funding, we were very fortunate to get from the NIAID. And I thank you for your attention. Sorry about the technical fiasco here, but I'd be happy to take any questions.
Terrific, Damian. Thank you very much. I had the same problem myself a year ago. And I still haven't entirely figured it out, but that didn't compromise it at all. It's perfectly legible in that format. That was terrific. So now technically we're into our question and answer period for the second half of today. And it works out perfect. We'll start with a few questions for Damian. Damian, as you were talking, and again, we brought up Arturo, and a number of things made me think about him. Two things. One, his ideas about why endotherms have 37-degree body temperatures, which I think is terrific. I love that. Yeah. And I'll turn it over to you just to explain that to the audience just a little bit and talk about the relevance to fungal adaptation temperatures. And then the second thing that he and Lisanne Porofsky put on a very nice thought paper about, and I don't recall the exact name, but it was something about the continuum.
The infection damage. Yeah, the infection damage continuum.
Exactly. Yeah, so let me ask you to explain Arturo's idea on the temperature and the dinosaurs and that, and how that might impact also some of the selectivity of fungi that can cause infections.
Right. So Arturo he's an incredibly creative and imaginative person. And one of his sort of pet ideas that he's been working on a long time and thinking about a lot is that the transition from so dinosaurs basically, went extinct as the world kind of warmed up, and the poikilotherms, which were not very conducive so they were fairly, conducive to fungal infections because they were all at about the same temperature. And after the transition and the mammal age started to come, the fungi basically lost their, if you will, big ticket items in which they could affect, and it reduced the mammalian body temperature, greatly reduced the number of fungi that could actually infect humans. And so, alternatively, recently, he's now taken that same idea with another fungus, which is Candida auris, which has become right before COVID came. It was sort of one of the most prominent new pathogens in the news. It is a highly drug-resistant Candida species, and he's proposing the idea that our global warming is actually letting these more fungi actually get back onto, in particular, skin colonization. So Candida auris was originally described in the middle 2000s from an ear. That's why it's got the name auris. And so our interactions with the environment, if you want to put it on a more general term, and we are interacting with the environment. As the environment changes, as we change, the distribution of fungi that we come in contact with and could cause disease is likely to change. So then the disease sort of infection damage paradigm has to do with trying to understand, it's a little bit, I kind of did a less elegant version of too much fungi, not enough immune system. Those are the extremes on Arturo's much more elegantly argued continuum. And in the middle is where for instance, in histoplasma, where many of us inhaled histoplasma, we had an immune response. That immune response was adaptive. It led to us to either contain or eliminate the fungal infection. If your immune response is too far to one side, you can end up with damage, as in collateral damage from the immune system trying to destroy the fungi. That probably happens in Aspergillus in the setting of a thing called allergic bronchopulmonary aspergillosis, where you don't actually have invasive disease, but the immune system keeps hammering away trying to get rid of it, causes inflammation in the airways, and now you have asthma-like symptoms. You can go the other way then, if you have a very aggressive fungi such as mucor. So mucor in the right patient, now our immune system is down. It's not immune driven, it's actually bug driven, because there's no immune system to contain this thing. Mucor will basically penetrate through tissue planes. It expresses proteases, lipases, all kinds of things that as bacteriologists you would think of as virulence factors, and it will destroy the face. It will go right back into the brain. So it is literally just like this flesh-eating bacteria. It is a flesh-eating fungi in the right patient. And so the relationship, as I said in the beginning of my talk, you have to talk about the host. And really that comes from Arturo. That I should have had a reference on that, because that is, the host and the pathogen are not two sides of the same coin. They're the same side of the same coin in how you think about pathogenesis.
Yeah, I think I really think it's fascinating that that relatively few fungi are able to grow and divide at 37 degrees, and they seem to start to drop off sharply at about 34 degrees.
Yep.
And again, Arturo's argument is that endotherms have a higher temperature, have been selected because of their higher temperature, which precludes fungal infection. And it's also worth reflecting on the ecology of fungi. They're almost exclusively terrestrial organisms. There's very few marine fungi.
Almost, right.
And they're all highly aerobic, exactly as you point out. They're even intolerant to, relatively modest levels of CO2 in many cases.
Right. There are a few. The only really facultative anaerobic fungi, if you will, are Candida. Candida is perfectly happy, but it also is a colonizer of our GI tract and in the colon. So it is very able to. They can tolerate a fair bit of what we would call hypoxia. So, for instance, Aspergillus, which its primary niche is a compost pile, where deep in that biomass, there is relatively low oxygen. They can do just fine there. However, you go to like low oxygen, like anaerobic one, then they die. Cryptococcus is a completely aerobic organism. It has to have a mitochondrial function and doing oxidative respiration, or it won't survive. And so, yeah, they're the sort of the physiology of us, the endothermic part, and then other parts of our physiology really put up a barrier to a number of these. However, what we are doing is altering our immune status a great deal. I mean, the number of new drugs that we have to have these really important disease modifying effects on things like rheumatoid arthritis, inflammatory bowel disease, many of the other therapies we're using for things like MS and are modulating the immune system. And that puts us at risk for things that we normally would be able to just pass off. And so it is there are more and more people becoming at risk for fungal infections in ways that aren't just HIV. That's kind of your knee-jerk response to, oh, that's an immune compromising. But you know, there are other conditions that do that.
Great. Let me bring. Go ahead, Hannah.
Oh, I think we were going to do the same thing, Mike. I was just going to invite both Christina and Shirong to join us also, and remind folks that you can either submit questions through the Q&A chat feature, or if you'd like to ask them aloud, you're very welcome and encouraged to just virtually raise your hand, and we will invite you to unmute and ask it aloud.
Terrific. Thanks. Well, to bring Christina and Shirong back into this, the Arturo's damage response framework reminds me of my attempts to understand Enterococcus and its behavior. And when I started off, I thought about that microbe as if it were a pathogen. And as I matured in my perspective, I pretty quickly realized that it's not a pathogen. It causes disease, but the number of enterococci that benefit from causing disease are infinitesimally small compared to the total global population of enterococcus. So that's not its thing. Christina, you're working with something that it sounds like it's more obligatorily pathogenic, and Shirong is looking at ways that the host maintains that dynamic. Christina, how do you view it?
Yeah, I mean, I think it's bacterial or pathogen dependent. You know, something like Shigella, a lot of people argue are just basically like E. coli. I do feel like it's not just genetics that you have to take into account, but you know, what's actually expressed and the different mechanisms by the bacteria. But something with Shigella, where it's very specific, causing disease, they really haven't found a reservoir outside of humans or apes, versus even comparing to something like Enterococcus or Vibrio, where you have this sort of accidental pathogen that's mostly in the environment, but then it ends up in humans and either wants to stay or wants to try to get out, especially something like Vibrio, where they're inducing that secretory diarrhea phase and just they want out. So I think it's variable in that regard.
Yeah. Now, for Shirong, one of the things that I think about a lot are antibiotic-resistant bacteria that are causing a lot of hospital problems these days. And for the most part, these are microbes. The hospital pathogens are microbes that can accumulate in the gut of an antibiotic-treated patient. And that accumulation, I suspect, leads to translocation into the bloodstream. And these microbes tend to be just very hardy, sort of cockroachy type bacteria. That certainly is the case for Enterococcus, but it's also the case for Acinetobacter and some of the others. They're just very hard to kill. And my sense is, whereas usually translocation involves representative normal flora of the gut, which often are exquisitely sensitive to oxygen and to neutrophil killing mechanisms, these hospital pathogens aren't, and as a result begin to flourish in the bloodstream and in the filtration organs. Shirong, is that your sense of the dynamic at that mucosal boundary, that's holding normal flora in check, but it may not be tuned to aberrations in the composition of that flora due to antibiotic treatment? How do you view it?
I would say so the host has a mechanism to define where the microbes, including the commensals and the pathogens, locate in a specific niche. And in the case of a hear what you're asking the antibiotic resistant bugs. Actually, I think there are multiple mechanisms that can escape so antimicrobe activity, and potentially they just have a different mechanism to get into the different site. In this, I think from my perspective, microRNA potentially in the gut, they can regulate the community as a whole, and through these commensals and through the other commensals can interact with the potential pathogens and restrain them to the gut instead of getting to the circulation. Whether there are microRNA or other mechanisms that will attract the potential pathogens get into the circulation, I don't know, but this is also a really good question. Yeah.
Yeah, you know, specifically, I was thinking, is there any evidence that there are microRNAs that affect the growth of some of these multi-drug resistant strains? Are they aberrant in their response to microRNA? So, for example, maybe there's a microRNA that affects the growth of E. coli. If we look at some of the extended spectrum beta-lactamase resistant E. coli's, do they still respond to that microRNA, or do we know that?
No, not yet. We don't know yet. But
that would be interesting.
We don't know yet. But actually, it's not related to the antibiotic resistance, but it's related to the pathogens when bacteria is translocated to a different site than original site. For example, the fusobacteria is a commensal in the mouth, in the oral cavity. In some cases, if it increase in the gut lumen, then there are accumulating evidence that yes, this is going to cause colon cancer. And yeah, this is something we show actually in our first cell host micro paper, that some microRNA potentially can promote the growth of the fusobacteria. This actually probably will be a potential mechanism explaining why some people get rectal cancer, although many other people have this fusobacteria in the oral cavity, but just a few population get the cancer. Yeah.
Great. So there's a very interesting question from Oni Basu asking about, so all of the things that we've been talking about are microbial interactions at the wet mucosal surfaces of humans. And the question is to what extent do pathogenic bacteria, are they able to capitalize on the carbohydrates of mucins, which are abundant in that environment? To what extent are they able to utilize those? And maybe, Damian, starting with the fungi, but Christina, does Shigella also do that? Damian, do any of the fungal pathogens utilize those carbohydrates?
So there actually has been some work from Sandy Johnson's lab at UCSF that shows that Candida albicans, which is again a mucosal commensal, its a potential to form biofilms is increased in the presence of mucin. And so the biofilm from Candida albicans and other fungi is made up of a highly carbohydrate rich extracellular matrix. And the presence of mucin actually will increase the ability of bacteria or Candida to form biofilms, in such to the fact that you'll have a strain that's very poorly biofilm ogenic, if you will, in vitro, and you add mucin to that, and then they form a biofilm just as well as another very proficient biofilm forming strain. And so it can use it certainly as a matrix. Whether they metabolize it is an interesting open question, and it's not entirely clear. They don't. Most fungi don't have any really large transporters that can import large carbohydrates. But in terms of the mucin, it is definitely part of Candida albicans' ability to interact with mucosal surfaces.
Yeah. Actually, if I could jump in real quick.
Go ahead, Pete.
Yeah.
I just wanted to ask a question on that note, Damian, because I think this is a really interesting, mucins are a really interesting focal point to elaborate a bit on the complexity of this relationship that fungi have with mucins. Am I correct in remembering, Damian, that there are also sort of some strains of albicans that their virulence traits are suppressed in the presence of mucins? And I wonder if there's a kind of ecological competition there.
Right. Yeah. So one of the key sort of Candida albicans virulence traits is the ability to transition from yeast to hyphae. And that seems to be reduced a bit in the presence of mucins. And in fact, Suzanne Noble's lab has shown recently through some really nice competitive assays that strains that are unable to form hyphae, and actually Carol Kumamoto from Boston is also into Tufts has shown this as well, are more fit. They will outcompete hyphae forming strains in the GI tract. Huh. And so they won't elaborate the adhesins and these other things, but somehow they're sticking, and actually they're sticking better and replicating better. And so, one of the things that especially, my group is also interested in is how you go from one niche to the next. These guys have to they can't just play one or two songs. They got to have a big repertoire in order to make it through all the stages in the infection. Yeah. And in addition, right, there's also bacteria there. And so it's just now starting to have having work by a number of investigators asking, how do these bacterial fungal communities, how do they modulate each other's biology to be successful?
Yeah, I think this is really a great point. Yeah, and there's already evidence that so some bacteria, for example, the bacteria I mentioned, the Akkermansia, is a mucin degrading bacteria. And I think in this community that they have some balance. So yeah, it's very interesting.
Yeah. And one of the prime risk factors for having an invasive infection is particularly, for instance, in neonates, is to have prolonged antibiotic exposure that increases your risk for invasive fungal disease like 20 or 30 fold. So changing that community really affects the dynamics and how the fungi, in particular Candida and other in Candida species, their pathogenic potential goes way up when you've altered that.
Christina, last word. How does Shigella contend with the mucosal boundary?
Yeah, so the idea is that the enterotoxins to induce the watery diarrhea actually helps to remove the mucus and commensal layer to help expose the epithelium. It looks like they do have some putative large autotransporter proteins that can degrade the mucin to help penetrate the layer of that. Also pathogenic E. coli like enteroaggregative E. coli have the same thing. So a lot of that work has been looking at how are these bacteria getting through the mucus layer to get to the epithelial surface. But I think certainly if they're going through the trouble of degrading the mucin, I don't see why they wouldn't then be able to utilize any of those nutrient sources and to grow in the mucus. And something else I'd just like to add, it's really been appreciated now that there seems to be a virome and bacteriophage present in the mucus layers to help keep, commensals closer to the lumen and the mucus more at least bacterial free and pathogen free. So I think those are some interesting dynamics that are going to come out in the future as well.
Absolutely. Absolutely. How about that? On her last word, the bell's going off at right on time. You can't get more precise than that. As the chime rings, let me thank all of the speakers in the second session. Like the first, they were all outstanding talks and really fascinating and nicely complimentary. I thoroughly enjoyed it, and thank you everybody for joining us. Although we don't have the opportunity to get together and raise a glass together after this, we'll do it virtually. But one of the advantages is that we could have speakers like Damian join us from Iowa and kind of broaden our usual symposium net. So with that, thank you everybody again. And let me turn it back to Hannah for some more final comments of sponsors and the happy hour.
Sure, yeah, thank you, Mike. Okay, so I'm going to alert you all to a few things. The first is that I have just put the information for the separate happy hour Zoom call. If you're available for that, we will adjourn here in a couple of minutes from this meeting and move into a different meeting. And that's so that if there are enough of us, we can have breakout rooms and chat in different rooms. And if there are just a few, we can all stay together in the same room and chat there as well. So we will move over there. That same information that's in the chat is also available at the end of the reminder email that you received about this event this morning. So that's the happy hour. I also have two more partner spotlights to read here. One of them is from Adventure Scientists. And if you have questions and want sort of a vignette about interacting with Adventure Scientists, I know you can ask Mike, who has done so. But Adventure Scientists aims to amplify and accelerate scientific innovation and decision-making by providing access to field data collected at scale from around the world. We are seeking partnerships where access to an unlimited army of trained boots on the ground would benefit your research. Imagine if you could collect data from every mountain above 20,000 feet, or if you could have soil samples collected from every biome. These are the examples of types of missions we are seeking. And you can contact either Greg, who is the founder and director of that organization. You can contact him directly, and that email address is available on our website, or you can get to it indirectly and read more about the organization at adventurescientist.org. Also, a plug for New England Biolabs. Established in the mid-1970s, New England Biolabs, or NEB, is the industry leader in the discovery and production of enzymes for molecular biology applications, and now offers the largest selection of recombinant and native enzymes for genomic research. NEB continues to expand its product offerings into areas related to PCR, gene expression, sample preparation for next-gen sequencing, synthetic biology, glycobiology, epigenetics, and RNA analysis. Right now, NEB has a number of job opportunities available. So for more information on that, visit www.neb.com/aboutneb/careers. You can also find that on the annual symposium page on our own website. Okay, I think those are my technical reminders, but to wrap us up here, I will mention that the 2021 symposium, MSI Symposium here, marks the 18th annual symposium, 18 years since the birth of MSI. It also marks more than a year now since the rise of the COVID-19 pandemic and all the ways that it has changed the way we communicate, work, and exist in the world. I know that for me, the pandemic and all the interwoven social and racial inequities highlighted by numerous events of the past year have at times made work feel wildly unimportant in the broader scheme of life. But in most moments, I'm also infused with a big feeling of gratitude for the ways that scientific research can support human health from all the ways we've heard today, especially in the second session, and also looking forward to a future vaccine moment for myself. And I'm also grateful for the individuals and communities with whom I'm connected through the microbial sciences. So I hope that what you've seen and heard and participated in throughout today's symposium leaves you feeling inspired, maybe by an enhanced understanding of some new microbiology topic, a new favorite trivia fact that you'd like to share with a friend, a new lens through which to view your own research, or a new way you might apply skills in an art or craft that you have to the microbial sciences, communication and outreach. If you would like to join us for the social virtual happy hour immediately following, use that Zoom link that I have noted. And it's also in the email that I mentioned. In the few minutes that it's going to take us to close out this webinar and start up the other Zoom meeting, you can grab your favorite beverage and snack, maybe your child or cat or dog, and then meet us there. I need to send a huge shout out to the entire MSI leadership team, all of whom you have heard from today, the co-directors, Peter Girguis and Michael Gilmore, and also our program assistant, Julia Worcester, and some folks who are working behind the scenes who maybe aren't technically paid staff, but who have been very helpful today. Thank you, Sophia, and many others who have provided support. If this was your first MSI event, I certainly hope it won't be your last. I am so grateful to all of you and the larger MSI community for your active participation. And I look forward to interacting with the MSI community, both virtually and in person in the future. Best wishes for the rest of your weekend and your summer and your year ahead.
Thank you, Hannah. Thanks, everyone. Hopefully you'll see you in the happy hour.